ఎంటర్ గ్రూప్ లో ఖచ్చితంగా జాయిన్ అవ్వమని చెప్పండి. ఇప్పుడు మీరు 6 మెంబర్స్ ఉన్నారు. 6 into 120 6 into 15 90 మెంబర్స్ దాకా వచ్చేస్తారు. పిల్లలందరూ ట్రావెల్ లో ఉన్నారు.
ఉంటే ట్రావెల్ లేకపోతే ఊర్లో సిగ్నల్స్ మీది మ్యూట్ లో అన్మ్యూట్ లో లేదు మేడం. మీది అన్మ్యూట్ లో లేదు. మ్యూట్ లో
Good morning, everyone.
Hello? Yeah, madam.
Yes, sir. Good morning, sir.
Can you call me later? I'm actually in a meeting. I'll call you after 1 hour. Actually, I'm in a seminar. Right? Yeah. Thank you. Yeah. Good morning, madam.
Good morning, sir. Good morning.
Yeah, madam.
Good morning, madam.
Okay. Am I visible, madam?
Not yet. Only some green screen we are able to see, sir.
I think I need to change this. Okay. How to change this?
Madam, good morning, madam. Chandra sir, good morning.
Good morning, madam. Hello, madam.
Hello.
Okay, ma'am. Got it.
Start.
Good morning, madam.
Good morning, Masunita.
Morning, madam. Good morning, madam.
Good morning, Vijaya.
Madam, shall I start?
Yeah, shall we start, sir?
Madam, I'm just trying to change my background.
I know.
I'm just trying to, earlier it was a green background for some recording purpose, we kept it. I may have to change this. Just give me a few seconds.
No problem, sir. No problem, sir.
Yeah.
Absolutely no problem.
Are you there? Okay. Still.
It's a black background now, sir.
Yeah. I'm just trying to change that out.
Correct. Vijaya, did you send to Pisa Party, sir, today's link?
Mailed, madam.
Mail to him also, no?
Now I will share in WhatsApp.
Okay.
How to remove this?
అదే చూస్తున్నాను నేను అనుకుంటాను.
I'll try to see in case my other laptop works. This is the new laptop where I was using.
Recording in progress.
I think I can use other laptop, madam.
Okay, sir. Okay, sir. This is pretty clear, sir.
Just a second, madam. Hello?
Yes, sir. Yeah, we are able to hear you, sir.
Okay, madam. Sorry for the inconvenience.
Absolutely. These are the hitches which we are these days expecting only, no, sir.
Yeah.
Madam, shall I start?
Please start, Sunita.
Okay. Warm welcome and good morning to all. I am Sunita, Associate Professor and Placement in charge of Tigala Krishna Reddy College of Pharmacy. Welcome you all for the second day national webinar on approaches and challenges in pharmaceutical sciences, sponsored by Royal Society of Chemistry, Sura Labs, and St. Paul Clinicare. It's my pleasure to introduce our today's speaker, plenary lecture by Dr. G. Chandraya Garu, currently working as an assistant professor, Department of Regulatory Toxicology, Naipur, Hyderabad. He is having more than 10 years of teaching experience, guided 55 master's students and 10 PhD students. He did his B Pharmacy from Kalia University, MS in Pharmacology and Toxicology, Naipur, Hyderabad, PhD in Pharmacology and Toxicology, Naipur, Mohali. He did his postdoctorate from Florida A&M University, USA. He has published more than 136 papers in peer-reviewed journals. He has presented more than 80 posters. He received grants from various funding departments, Government of India, towards extramural projects. He is skillful in multidisciplinary research areas like pharmacology, toxicology, drug delivery, and nanotoxicology, with special focus on organ fibrosis and respiratory diseases. He is listed in Stanford's World's 2% Scientists in Pharmacology and Pharmacy category. He is a trained GLP inspector for DST NGCMA, holder of 1 US patent. He is a recipient of DAC Fellowship. He is a member of Board of Studies in JNTH and Anurag University. Now, I invite Dr. G. Chandraya Garu to present the webinar on recent trends in pharmacology and toxicology. Over to Dr. G. Chandraya Garu. Good morning, sir.
Yeah, good morning, madam. Thank you very much for the kind introduction. Hope you can listen to me, right?
Yes, sir.
Voice is clear?
Yes, sir.
Wonderful. Okay. So nice to be, the part of this particular national recent challenges and approaches in pharmaceutical sciences. And I must thank Dr. P. Spati sir for making me part of this particular event, and also I must thank Radhika madam, Sunita madam, and also the Vijaya madam for inviting me to this particular event. In fact, they are my seniors in Kakatiya University, Warangal.
Yeah.
And proud to be part of ECPSC all the time, and also I'm a part of Naipur Mohali. So after my B Pharmacy, I went to Mohali for my master's and PhD. I did my postdoctorate and came back to Naipur, Hyderabad as a faculty here. Before going into the seminar, I just wanted to tell you about Naipur, Hyderabad, which is sixth rank six in the country in the NIR ranking in pharmacy category. All the infrastructure required for the people to pursue their research activities in different fields, pharmacology, pharmaceutics, all the aspects of pharmaceutical sciences. We have all the infrastructure, equipments, and highly funded. So the people who are actually attending this are the people who are actually doing PGs can and to write the Naipur JE exam to get a seat in Naipur, Hyderabad, and they'll be also given a fellowship, which will be very handsome fellowship also been given. So I also must greet all the participants who might have joined from across the country, and all the other my colleague, fellow speakers, Dr. Jayant, Dr. Narendra, who was my ex-colleague in Hyderabad during stay here. I hope you all doing well, and I wish you all the very happy New Year, and also advance Pongal wishes to all of you.
Thank you, sir.
I'm going to talk about recent advance in pharmacology and toxicology. Pharmacology, toxicology, as you know, everybody knows about what is pharmacology as a definition. However, what are the recent trends and how a pharmacologist can actually contribute, or what are the challenges we are facing in pharmacology as well as toxicology? Because pharmacology, everybody knows, we read a lot, but in toxicology somehow is not been well taught during B Pharmacy or M Pharmacy, because when you take the drug, the more than the pharmacological activity, the safety comes first. 1 in 1 lakh person is suffering from any kind of toxic, is showing any toxic concerns. That's where we don't approve the drug. The US FDA will be very keen on how to prevent drug-induced toxic effects, mainly carcinogenic effects. That's why we have a genotoxic impurities. If the component genotoxic, which is suspected to be carcinogenic, will never be allowed into the market. Even drugs which are approved will be withdrawn in case if you find that they may have, genotoxicity, carcinogenicity, and other effects. So I will be touching upon some of the aspects of toxicology, some of the safety pharmacology aspects, as well as the biologics. Nowadays it has come up how the biologics are produced, and also omics tools, how different omics tools, genomics, proteomics can be part of pharmacology or pharmaceutical research for the drug discovery and development, as well as for the drug repurposing approaches. These all will be covered in my seminar. I have made 80 slides. I know it takes a little longer time, but actually I will try to be a little faster to finish 1 hour time. So let me share the screen, and then we'll proceed further. Is my screen is
Visible. It is visible, sir.
Is it changing to next slide?
No. First slide only it is there still. And it is
I want to see.
not in the
Is it not
slide
moving?
share.
Is it changing?
No, sir.
Or not?
No.
No, right?
No.
Let me reshare again.
Yes, it is changing, sir.
Now it has changed, sir.
But I have removed from the slideshow to normal mode. That's why it's showing. I need to keep this in the slideshow.
Okay. But for us, it is not even, it's not on slide share, sir. Previously also. It was not on the slide share.
I have not kept in the slideshow. Now I kept in now on the slideshow.
But it is not showing us, sir. It's not showing as if it's on the slide share.
Let me just try to share.
But this is also clear enough, sir, for us.
Do you think? Okay. Do you think it is, that's clear enough?
Yeah, it's clear enough, sir.
Sure. Okay. Let me see. Did it move to the next one now?
Yeah, now it has come on slide share, sir. It's much clearer.
Wonderful. Okay.
Very good. It is really clear, sir, now.
Okay. So I hope how to remove this the top one.
Which one, sir? That doesn't go, sir. View options and your screen, no, sir. That doesn't go.
Hi, madam. Yeah. So without wasting much time, I will proceed into my talk. As mentioned, I'm a faculty in regulatory toxicology. Is the voice clear to madam?
Yes, it is clear, sir.
Wonderful. Okay. As I mentioned, I'm working in the regulatory toxicology, a unique subject in pharmacy field where toxicology, which is studied or dealt in regulatory aspects. That means you have to perform all the toxic studies as per the regulatory guidelines provided by the ICH and the OECD, so that you perform the toxic studies properly, so that when you plan the clinical trials, we save the patients or the participants of the clinical trials. Otherwise, what will happen is, if you are not properly doing the toxic studies during animal experimentation for the new drugs, there is a possibility that you might be missing some of the toxicity effects induced by the drugs, like that the thalidomide tragedy which you have seen earlier can also happen. So based on the thalidomide strategy, we have felt that we need to have very strict guidelines on the regulatory guidelines of toxicology. That's why regulatory guidelines have come. And ICH as well as the OECD have come up with a different type of safety guidelines to perform for the new chemical entities to enter into the clinical trials. And then based on the animal toxicity data, you will be performing clinical trials. Again, you have to prove the safety as well as efficacy in the clinical, and only that will be approved, which is a long-term process, which is taking 12 to 15 years of time, which I'm going to talk about in some of the slides. So the topic is recent trends in pharmacology and toxicology, right? So if you see this pharmacology, everybody knows about what pharmacology. This slide changing.
Yes, we can see.
Okay, next slide, right? Second slide.
Yes, sir. Pharmacology.
Wonderful. Okay. The definition of pharmacology, the pharmacology broadly defines as a how the drugs are performing in the body, how the body is dealing with the drug. That means pharmacokinetics and pharmacodynamics broadly. So any pharmacokinetic aspects any pharmacodynamic aspects, pharmacodynamic in the sense efficacy as well as the safety will come under pharmacology. So there's the broad area where we can actually do the different type of multidisciplinary activities, in vitro or in vivo, or clinical versus pre studies. These are all possible in pharmacology to contribute in the drug discovery and development as well as the clinical pharmacology, where you will be trying to optimize the optimal use of the therapeutics after approval, after approval during the clinical trials, right? So broadly, the pharmacology will be pharmacologists will be playing a role in the screening of the NCEs, new chemical entities, where thousands of molecules will be screened in the in vitro in vivo models to find what are the lead molecules, what are the molecules which can become a new drugs later. We'll be doing the pharmacokinetic and pharmacodynamic studies. And one of the very important area where pharmacologists will be contributing is animal model development and evaluate the efficacy studies. Because for every disease, you need to develop the model. Suppose if you want to study anti-cancer activity of the new drug or the new formulation, you need to have the animal model. How do you use animal model? Again, you have to select the right model. It can be a nude mice model, it can be a chemical induced cancer model, it can be some other model. So you need to mimic the human condition in the animals, which is very which requires a lot of skill, a lot of optimization, and you need to select the right species, the right model, right duration, and right parameters will be required for you to evaluate the model, whether that is developing or not, whether the compound is protecting from the particular disease condition or not. So as a pharmacologist, we'll be also identifying the new targets. New targets, why we need a new targets? Because the moment we have a new target, that's where we can explore the possible chemistry, we can explore the drug designing, we can explore the drug discovery process, so that we'll be coming up with a new NCEs, or we coming with the new drugs, which will be very selective to the new target. Why we need new targets? Because what are the existing targets have a problem. We still have a problem, the existing drugs, because the safety issues, mainly the anti-cancer drugs, or the anti-fibrotic drugs, the anti-hypertensive drugs, or the anti-diabetic drugs. That's why you always have to find the new target. How do you find the new targets? By studying the normal versus disease conditions, how the biochemical or the particular protein or the particular gene is altering in the disease condition will be identified. Based on that, we'll be trying to know how a new target can be a novel one. Recently, we have a COVID-19 COVID virus. We know that it is having a spike protein. Can that become a new target? That's how you may have to see what are the ways we can actually inhibit the spike protein or some other component present in the virus, or can we inhibit the host cell receptor which is receiving the particular virus. So you have to see what can be the suitable target. If you are selecting the target should be very selectively present in the diseased tissue and not present in the healthy tissue. So that's how we can be only targeting the diseased tissue, not the healthy tissue. You can also work in the dose response curves, the concentration response curve. Nothing but pharmacology is nothing but dose response curve, the drug receptor binding studies, where you'll be talking about how increase in the concentration is resulting in the different responses. It can be contraction, it can be relaxation, it can be any other parameters. You have to have the generate the dose response curve in pharmacology. Again, this is required for all the new drugs before proceeding for further development. And we know clinical safety and drug interactions are also very important in pharmacology, which we all might have been studying during your B pharmacy. But some of the new things where the recent developments probably we are not aware of unless you enter into the research areas, right? So some of the animal models where you can actually cite the disease pathology, like type 1 diabetes. How will you induce type 1 diabetes? We know type 1 diabetes is more of a insulin deficient diabetes. How can you induce that particular diabetes? You have to induce a chemical to the animal, a rat or mice, so that it's selectively destroying the pancreas. The pancreatic beta cells are the ones producing insulin. So you have to selectively destroy the pancreas without causing damage to the rest of the body. That means you have to find the experimental agents which can be used as a disease-inducing agents. And what model to be worked upon will be decided by on what area you have expertise and what kind of support you will be requiring to develop that particular animal model. So you will be understanding the human physiology and pathophysiology. You have to have knowledge in the biochemistry, enzymes, and the hormones. You need to have a detailed understanding the different receptors and the pathways. Why different receptors and pathways? Because receptors are the ones, the main targets. You know all the G protein coupled receptors like beta receptors, alpha receptors, or angiotensin receptors. Based on the receptors only we have developed the drugs. That means you need to have the knowledge on the receptors. What are the new receptors? What is the downstream or upstream pathways linked to this particular receptors? How can we alter this particular receptor in case that particular receptor is playing a role in the particular disease condition? You need to have a thorough knowledge on the receptors. Again, this comes under the pharmacology and toxicology area. As many PK and PD is are important for you. You can also perform a lot of in vitro cell culture experiments, in vitro tissue experiments. Earlier, we used to do a lot of in vitro experiments where you do you take the rat heart or frog heart. You can also take the ileum. You can also take the rat thoracic aorta. I mean, the moment you take the tissue, you can actually study how the tissue is responding to the particular the experimental compound. The experimental compound can be a natural source. It can be a synthetic chemical. Suppose if you want to study the calcium channel antagonist, how can you study? There are different ways to study the calcium channel blockers activities. You can simply study the muscle contraction or relaxation. You know that the calcium contributes to the contraction of the muscle cell. If you can actually able to record the contraction on the chymograph or the tissue the recording systems, you can able to screen the compounds at the with very minimal expertise. That means you can actually have isolated experiments to screen the molecules. Or you can later, based on the performance of the molecules, the best molecule which is having a good activity can be taken further into the animal models. That's where you can actually do the detailed evaluation by using a right parameters to evaluate it. Parameters can be what are the parameters? It can be hypertension, it can be a insulin release, or whatever it is. And the people need to have a good animal handling. So you need to have a good in vitro skills as well as the good in vivo skills. In vitro means it can be cell lines, it can be a isolated experiments. In vivo, different animal, mostly the rat and mice will be using, because these are the models most suitable for the pharmacological research and are easily available, and they have been found to be suitable for developing a different disease conditions. And you should also have a knowledge in the molecular biology techniques, which is one of the difficult tasks for many of the people who are working in the private organizations. Molecular biology like Western blotting, immunohistochemistry, confocal microscopy, these are the techniques nowadays required for you if you want to publish your paper in a good journal. Yeah, you may be sure that your compound is very good. It is inhibiting the calcium channels, it may be inhibiting the spike protein. But how to prove that at the molecular level by using the different tools like RT-PCR or the Western blot or the immunohistochemistry or immunofluorescence, flow cytometry. These are the tools actually are required. Then only your paper can go in a big impact factor journals. Because until unless you prove the mechanism how that's happening, generally this may not be accepted. Yes, it's very important to have initial screening, but you need to have a molecular tool. That's where you should collaborate with the people who are having this particular expertise. Similarly, like a pharmacology, you also need to have a thorough knowledge in toxicology. Because why? Because safety is important than the therapeutic activity of the drugs. Safety comes first, then only activity. Yeah, okay, if the drug is not producing anti-hypertensive, there is a debate on that whether the statins are producing a required therapeutic benefit or not. However, if the statins are having a serious concern of the toxicity, you will not be allowing the drug to be used by the patients. Safety always comes first, then only the efficacy, right? The toxicology studies of the adverse effect of the drugs, the toxic effects of the drug on the living systems. Living systems in the sense, of course, we cannot study the toxicity on the humans. That's why you're using the mice or the rats as a models. Or you can also use the smaller models like zebrafish or the lower animals. Sometimes, in case if you want to study at the larger scale level. So that means you'll be using different models to study the toxic effects. And if you are doing the toxic studies as a regulatory requirements, we have to do as per the OECD and ICS guidelines. OECD, Organization for Economic Cooperation and Development. ICS, you must be all knowing. International Council on Harmonization, which will be giving a guidelines how to perform the different type of toxic studies, other efficacy studies to get the approval for the particular molecules. There's a very famous statement given by Paracelsus. It says that all the substances are poisons. Even common salt can be a sodium chloride can be toxic if you take higher concentrations or higher doses. There's nothing free from the toxicity. The dose is the one which will be deciding. That's how you'll be doing the dose response studies in the pharmacology and the toxicology. Then understand that, okay, this is the dose, is the effective dose, this is the dose, is the toxic dose. Based on that, you'll be calculating the different parameters. NOEL, LOEL, therapeutic index, margin of safety, these all will be calculated based on the doses you're using, the dose responses curve you are generating for pharmacological activity as well as toxicology activities. If you see the toxicology, if you have a range of toxic studies needs to be done, the number of studies you do in the toxicology are much higher than the pharmacology. In pharmacology, probably you may be using 1 or 2 animal models to prove that compound is effective or not. Whereas in the toxicology, it's not in your hand. You have to follow the regulatory requirements. What are the regulatory requirements? If you see the example, you have to do the single dose toxic studies, acute toxic studies, chronic toxic studies, genotoxic studies, carcinogenic studies, local toxic studies to the skin as well as the ocular, phototoxic studies, reproductive toxic studies to understand the teratogenic effect, developmental toxic studies, immunogenicity studies mainly for the biologics, if they're causing immune system activation or immune suppression, both are problematic. Safety pharmacological studies like QTO prolongation and so on. That's why when you do that, when you talk about the toxicology, you have to follow the guidelines. What are the guidelines you can follow? You can follow the OECD or you can also follow the ICH guidelines. Both the guidelines. Broadly, pharmacology R&D activities can be done in the pharmacology of different models, mostly oncology or pain or inflammation, metabolic disorders, respiratory diseases. Nowadays, it's very common due to COVID. Psychopharmacology, the neuropharmacology. You can also do the in vivo toxicology of different types of toxic studies, as the previous slide is talking about. You can do the in vitro toxicology where you can use the in vitro model to do the toxic studies like genotoxicity or the carcinogenicity, or you can use the human cell lines to study how the compounds are causing toxicity to different organs like liver or heart. You can study safety pharmacology studies, mainly CNS system, cardiovascular system, and respiratory evaluations. Because if you see the safety pharmacology studies are done in the not at the higher doses. The safety pharmacology studies are done at the dose which are close to the pharmacologically active, therapeutically active dose. That's why safety pharmacology studies are called the secondary pharmacodynamic effects, where how this particular undesirable pharmacological secondary pharmacological effects are causing a physiological alteration to the animal. Because there are the drugs which have caused mortalities in the patients after they're used for a long time, like terfenadine, which is a drug withdrawn from the market in 1998 due to QTO prolongation. That is also called the Tarset points. Pharmacologists will be also playing a role in the DMPK, drug metabolism, pharmacokinetics, how the drugs are metabolized, how the liver microsomal enzymes are playing role, type 1, type 2 enzymes, how drug induction or inhibition is important, how the plasma protein binding, these all the aspects will be done. That's all. If you see the industry, there is a special the division called the DMPK, where one of the part will be done by the pharmacologist, analysis part will be done by analysis per people. So it's a group of people. And the people will be doing a lot of histopathology studies and the biochemistry studies to understand after giving a compound whether this particular drug is safe or not. How will you come to know? You'll be doing a biochemistry of the to understand how the liver parameters, kidney parameters, heart parameters, or the lung parameters are altered, as well as the histopathology to understand what how the at histological levels how this particular compounds are altering it. And pharmacologists play a role in everywhere drug discovery and development. At the initial stage where the lead identification, lead validation, you'll be the molecular pharmacologist. That means the pharmacologist will be having a knowledge molecular pharmacology to understand new target and how the disease will be playing. Once they know the molecular pharmacologist identify the new target, the new lead molecule, now you need to do the preclinical studies in the animal models. That's where the safety pharmacology and toxicology will come into picture. Once you perform the safety pharmacology studies or the toxicology studies, as per the battery of the toxic studies required, you'll be sending the molecule to the clinical trials. That's where the clinical pharmacology will come into picture. That means a molecular pharmacology, toxicology, or the safety pharmacology and toxicology, then clinical pharmacology. These are the three areas where the pharmacologists will be contributing it in association with the toxicologist to get the drug approved. This is the process, you already know it, where you're starting with 10,000 compounds at the early stage. and you will be narrowing the compounds to maybe 200 and 250 compounds during the preclinical trials. Then only 5 to 10 compounds will be going to clinical trials, and only one drug may be approved. That it takes more than 12 to 15 years. You need 2 billion US dollars, which is equivalent to $15,000. 15,000 crores. 15,000 crores is the cost required to bring a drug. Convert 2 billion into USD, it will be 15,000 rupees, which is high. It's highly risky affair, because since it takes 12 to 15 years of time, and it needs almost 15,000 crores of rupees. If a company is only depending on that particular compound, if that is failing at the latest stages and then phase of the particular new compound, the company will not be able to really recover well. That's why the companies, only larger organizations are able to do the drug discovery, not the smaller organization, because they need to have other economic sources to survive in case if they fail. So it's a highly risky affair, where you need to ensure that, or you need to make sure that the chance of failure will be lesser by doing a different tools. What are the different tools you'll be using? You'll be using a different pharmacology and toxicology tools, saying that yes, my molecules very thoroughly studied for their safety, and I don't suspect any safety issues will come later. That's how you need to do a very thorough thorough risk assessment and the risk evaluation. Clinical risk assessment will be done. If you see now the stepwise target verification, where you'll be understanding the new target at the the existing target, you'll be trying to validate. How do you validate it? If I inhibit this particular target receptor, whether I'm able to reduce particular pathological condition or not. If I activate this particular target, whether I'm able to get my required pharmacological benefit or not. So you'll be validating the target. Once you validate the target by using the tools like siRNA, small interfering RNA, or any other knockdown experiments, now you need to understand, okay, now I understand that this target is suitable. What's next is, now I need to identify the lead compounds or lead optimization. What could be the suitable moiety? What is the right heterocyclic molecule which will be binding to the particular target? So you need to do the lead optimization. Once lead optimization is done, where we found that group of compounds, these compounds are having a very good binding to this particular target. It can be receptor, it can be enzyme, it can be any other protein. Then you can actually do the preclinical testing in both in vitro, in vivo, to prove the efficacy as well as safety. First, you'll be doing the efficacy to prove that yes, this molecule is behaving in vivo conditions also, and it is inhibiting the particular pathological condition. So once you prove the efficacy, then your next consideration will be on how safe the molecule is. Then we will be doing this different range of toxic studies, and we will be doing the GLP as well as the non-GLP toxic studies. Why GLP studies? GLP studies are more controlled studies where some of the regulatory bodies will be requiring a GLP toxic data only. If you are not doing the toxic studies in the GLP conditions, your data may not be accepted. That's why, again, GLP, non-GLP. But in a GLP takes a lot more time, as well as the more cost is involved in that. The companies, again, will strategize. Initially, they will be doing the non-GLP toxic studies to just for their safety, that the compound is safer. Once they are sure that the compound is safe, then only they will be moving the molecule to the GLP toxic studies. Based on the toxicology data, and IND will be filed. IND is filed based on the information of all the preclinical data generated, also physical chemical properties generated for this particular molecule, and indicating that yes, we have done all the required toxic studies as per the guidelines, and we say that this molecule is safer. This is the data say that this molecule is safer. Kindly allow us to do the clinical trials. Accordingly, they will be doing the clinical trials, do the clinical trials, efficacy and safety, again reconfirm with the clinical settings. And based on the data generated during the phase 1 to phase 3 to phase 2, phase 3, and phase 3 clinical trials, you will be generating the data. The data will be submitted as the new drug application. Once the NDA goes as a new drug application, the review body will be seeing whether whatever the data been generated during the clinical trial is proving to be effective compared to the existing drug or not, and what are the safety concerns. And once they are satisfied with the safety concern, then the drug will be will be approved as a drug. Again, this process takes 12 to 15 years time. That's how it is very costly, right? And the drug, why you call the drug discovery development? Because drug discovery is talking about initial stages where the target validation as well as the lead optimization. Whereas starting with the preclinical testing to clinical trials to NDA approval, it's called development. Development means we already narrowed down a molecule. Now we are trying to develop as a drug. So discovery is different, development is different. Discovery is the initial stage of the identifying the right target and right lead molecules, and discovery is narrowing the molecule and further taking it. So these are the aspects where, initially target selection, you'll be using a different cellular genetic targets, genomes, proteomics, and bioinformatics tools will be. Why bioinformatics? Bioinformatics is nothing but a computer supported data where connections, how the particular receptor, how the particular protein, how the gene is connected to different pathways will be able to understand better by using the informatics. So once it is target selection, target is selected, you'll be doing the lead discovery by using your chemistry and different assays. Then you'll be doing the medicinal chemistry to optimize what side chain is required. Hydrophilicity, lipophilicity, lipid rules, all will be coming into picture doing the medicinal chemistry. Structural active relationships, these all will be done. Once you develop the group of molecules, you do the in vitro studies, then you'll be doing the in vivo studies, both pharmacology and toxicology. Then you'll be going for clinical trials to get it approved. So this is the process. Now, so this is overall how the pharmacologist as the toxicologist is actually having a role here. But if you take one of the very common diseases of the cancer, how the cancer is different than the cells. What are the features of the cancer? What are the features of the cancer? Uncontrolled growth. They are escaping from the cell death. Right? These are some of the features. If you see the cancer, self-sufficiency in growth signals. They don't look for all the signals for them to survive. What are the minimal you give it? They are highly adjustable cells. If they're not getting some of the requirements, some cells may not be able to grow or may not be able to proliferate. Whereas the cancer cells, self-sufficiency, whatever they have it, they will be able to produce the daughter cells, they will be able to proliferate. Insensitive to the anti-growth signals, our body has anti-growth signals. That means once we have number of cells are achieved, your body will be sending a autocrine and paracrine signaling, indicating that you have enough number of the cells, please don't proliferate. That is there in our body as in the normal tissue. Whereas the cancer cells and the tumor cells, insensitive to anti-growth signals. They are not sensitive to the anti-growth signals. So they will be not caring what are the messages being given by your body to stop the proliferation. They keep on proliferating it. Tissue invasion and metastasis, another problem. Cancer in one particular area is fine. You can actually always can remove it. But what if it goes to other parts of the body? You cannot remove every organ, right? If it's in the liver, certain part of the liver, you can always remove. But the problem here is that cancer, the highly metastatic. They can actually go to different parts of the body. They can go to.
All the others, other than the speaker, please mute your mics. Chaitanya? Sorry, sir. Sorry for the disturbance. Please continue. Sir, you are muted, sir.
Okay, I'm fine.
Thank you. Thank you, sir. Sorry for the disturbance.
Am I going slow? Am I going slow?
No, sir. Your speed is perfect.
Okay, fine. Tissue invasion and metastasis is mentioned one of the main concerns why we are not able to really combat the cancer, right? Limitless predictive replicative potential. They get the kind of immortality. They never die actually. There is no senescence to the cancer cells. They keep on developing, they keep on replicating. That's why your normal tissue will be occupied with the cancer cells, cancer tissue. So that's one of the problem with the cancer cells. Sustained angiogenesis, that when the tissue is growing, which is additional tissue which is not part of your body, you need nutrients and oxygen supply to the tissue. That's how the cancer or the tumor cells will be developing angiogenesis. New blood vessels will be formed. So that can be one of the target where you can actually inhibit the angiogenesis by angiogenesis inhibitors, VEGF inhibitors, many other things. That means angiogenesis is found to be increased. Angiogenesis is also seen in the cancer. Evading apoptosis. Apoptosis is nothing but programmed cell death. Your body has a property, any cell which is having abnormal behavior with respect to genetic material or with respect to the cell membrane alteration will be prone to by the programmed cell death. But what is happening in the cancer cells? They don't care for the cell death signal. They don't even undergo apoptosis, reduced apoptosis. These are the features make the cancer cells is unique to survive in the body. Not only that, it is also spread to the rest of the body. That's why what are the drugs we have it, they're not really very, very effective. So how can the cancer can be targeted? How cancer can be treated? Multiple targets are there. Different approaches where you can actually use the EGFR inhibitors, the tyrosine kinase receptors. You can also the cyclin dependent kinase inhibitors where these are involved in the cell proliferation. You can target that. Immune activating immune system is found to be very compromised in the cancer. Your immune system will be trying to fight against the cancer. But what happens is the cancer cells will be developing a immune suppressive suppressive property. That means they don't respond to the immune system in your body. So immune compromised system will be there. So you can actually use some of the immune activating things through the monoclonal antibodies. You can actually do the telomerase inhibitors, which are telomerase is the enzyme which is involved in the longevity of the cell. So you can actually inhibit the telomerase. You can also do the anti-inflammatory drugs. Like celecoxib has been highly discussed 10-20 years back. It can be a very good anti-cancer drug. A lot of clinical trials happen because based on the anti-inflammatory. Because initial stages of the cancer is inflammation. So you can actually use the anti-inflammatory agents. You can also have the different HGF signals for the invasion and metastasis inhibition. You can also have the VEGF signaling for the angiogenesis. PARP inhibitors for the to promote the genome instability. We don't want the cancer cells to be stable. We want to make the cancer cells to be unstable by causing gene instability through the PPR PARP inhibitors. So you can also produce apoptosis to the pro-apoptotic signaling activation and so on. So you can actually target the cancer cell by using different modes. However, what are the mode you are doing, what are the way you are doing, still you are not able to get the exact effective therapy. The reason is the cancer cells, though they are actually having all the signals present, some of the signals are also found to be available in the healthy cell. That is the problem where you are still not getting a effective cancer therapy. You are still seeing a safety concern. You are still seeing a drug-induced toxicity appears, mainly the cancer. That's how it's very commonly, very widely believed that it's not the cancer which is killing the patients, the cancer drug. If you go back and see also some of the people who unfortunately might have been succumbed to this particular cancer, person might be suffering from cancer for a longer time. They may be surviving if they're not diagnosed. I'm sorry to say this statement, but what is happening is the moment the person has been diagnosed, we want to take whatever the care we can do it, we start giving a chemotherapy. The chemotherapy never discriminates the healthy cell and normal cell, healthy cell and the cancer cell. It start causing damage, kidney, heart, the bone marrow, immune system, everything is getting affected. That's how the chemotherapy is the one which is responsible for the mortality, not the cancer. But the cancer to kill the patient, what is required? It has to go to the vital organs. It has to occupy the normal organ by the abnormal malignant tissue. Then only it will be malfunctioning. Whereas the drugs which are having a no discrimination, they will be also damaging the healthy system. That's why the chemotherapy is still problematic, and there is no cure. That's why we are trying to develop the targeted therapy. Or you have to screen the patients properly. What could be the right drug for the patient? Because cancer, again, simple breast cancer can be of different types. Simple lung cancer can be of the different types. What receptors present? You have to do a lot of profiling. You need to segregate the patients. Okay, these are the drug. These are the people sensitive to this particular tyrosine kinase inhibitors. These are patients maybe suitable for the HCR2 receptin type of receptors. Like that, if you can segregate it, you can actually better give the treatment. How that can be happening? You have to do a lot of molecular profiling. Molecular profiling in the sense you have to use the tools where you understand what are the proteins, what are the genes are found to be present in the particular tissue compared to the rest of the other tumors. So that, this is the particular target where I should use this particular drug. So like that, you need to actually do the profiling of the patients and categorize them into different types, then give the appropriate drug. The same is the case even for the antibiotics also, but I'm talking about the cancer here. Now, if other way of targeting the cancer is antibody drug conjugates. What do you mean by antibody drug conjugates? a drug is conjugated with antibody. What is the benefit of conjugating antibody? You know that antibodies antibodies are actually are binding to the antigen. You have to select the antigen which is present only on the tumor tissue. If you can do that, okay, my tumor, the tumor tissue have seen by the molecular profiling, I found that these are the particular antigens present in the tumor cells. Now, what I can do, I will try to develop the antibody against the particular the antigen, so that this particular antibody will be conjugated with the drug. Now, what will happen? Because antibody has a selective binding to the antigen, while traveling to the antigen after injection, it will be also carrying a drug which is anti-cancer drug to the tumor type tissue. That's how I will be able to selectively deliver the anti-cancer drug to the cancer tissue based on the antigen antibody drug conjugates, which is a very promising area. Many companies are working on it. The one of the drawback of this particular technique is antibodies production is not very easy. It's a biological product, right? So you don't have a lot of antibodies. You have Herceptin for the breast cancer, but you don't have a lot of antibodies. So this a lot of area needs, a lot of research needs to be done to find the right antibody can be used. To produce the right antibody, you need to have the right antigen identified as the cancer cells. How do you identify the antigen present in the cancer cells? You have to do a lot of molecular tools. You have to do the omics, genomics, proteomics to understand what particular antibodies. So now, since antibody drug conjugates are injected to the patient, so it will be traveling throughout the body. And since the antigens are present on the cancer cells, it will be going and sending the drug, the chemotherapeutic drug, to the tumor cell, so that you can selectively inhibit it. And you can also promote how the releasing the drug. You know that cancer cells are having acidic in nature. So that means you can make a linker. When we make this the conjugation, you will be having a linker. The linker will be sensitive to certain conditions. It can be a pH sensitive. It can be a protease sensitive. It can be a reduction condition. So you can use the linker. Again, there's a lot of chemistry needs to be involved here. It's not just a molecular pharmacology or the molecular biology. You need to have a chemist who can develop the proper linker. You need to have the right drug. Then only it is possible to produce it. Similarly, a cancer can be targeted by using different receptors. If you see that cancer, there is a folate receptors are overexpressed, transferrin receptors are overexpressed, mucin receptors are overexpressed, EPH2 receptors are overexpressed, integrin receptors also found to be overexpressed in the cancer stem cells, which are one of the biggest problem in the cancer therapy. So can be targeted with CD44 ligands. That means these are some of the receptors I'm talking. These are some of the antigens found to be overexpressed in the tumor cells. Maybe you can develop the drug and this particular ligands together, so that you can deliver the drug selectively to the particular receptor. That means you need to understand the biology of the tumor, biology of the cancer cell, what is the receptor overexpressed. The same is also the targeted drug delivery in the nanotechnology, where instead of the drug conjugation to the antibody, they will be surface decorating the nanoparticle with the ligand or the monoclonal antibody, which is a monoclonal which is having a surface decorated with this particular targeting agents, so that when inject this particular nanoparticle at the drug, they will be very selective to only go to. That's how we can maybe able to reduce the safety concerns, so that effective. There is a recent trend in the CAR therapy, chimeric antigen receptor therapy. Chimeric antigen receptor therapy, CAR therapy. This is also called the living drugs. Giving patients a living drugs. Living drugs in the sense, this is not a chemical drug. This is not even a biological product. This is a cell which is acting like a living drug. So when you can inject the cell will be roaming around the body and only find the cancer cells, which are this particular cell say having a receptor. This cell has a receptor which is having a receptor which will be bind to the cancer cell. When the T cell, because the cells are T cells, you know that T cells are cytotoxic T cells, T lymphocytes. What they do is they will go and attack the cancer cells. But inbuilt, your body may not be having a sufficient T cells which will be attacking your cancer cells. That's why you can genetically engineer them. You can actually program them in such a way that I will isolate the T cells from the patient, from the bone marrow or the blood. Then I'll try to modify the T cells On a surface, they will be expressing a receptor called the CART receptors, chimeric antigen receptors. These receptors having a special affinity towards the cancer cell. Now, once I develop this particular specific CART cells, then I will try to pull up, make the more number of cells, because you need to have sufficient number of the cells to inject into the body. That means you take the T cells, you modify them to express the receptors, CART receptors, followed by you proliferate them to get enough sufficient number, followed by you give the cells back to the same patient. There will be no immunogenicity, because the cells have come from the same patient, and you have made them to attack the cancer cells and inject back to the patients. This can be very safe strategy. People are trying, and this is found to be a very revolutionary technology. Of course, it's very expensive, but the technique looks very effective, where it's not the primary site of the cancer sites can be affected. You can attack every part of the body, because the cells, wherever it is, it will go and police them, they will identify them. That's how I will able to even attack the metastatic tumor cells. Circulating tumor cells, these all can be attacked by using the CART therapy, which is very promising. Some of your keen should go and read on more on the CART therapy, how this can actually make a lot of sense. And since we can actually alter the expression of the receptors on the surface of the T cells, you can also use the CRISPR-Cas technology, which is again the recent trend, where I'm not covering here. The CRISPR-Cas technology can be part of to produce the CART cells. So another the aspect is cancer stem cells. Stem cells are generally good cells. We know that they produce multiple organs due to the pluripotency, and they promote the repair of the damaged tissues, or the cells which are died due to the senescence or the olding. But in a cancer, if we is having a cancer stem cells called the CSCs, it's a problem. We don't want the cancer stem cells to present. Stem cells are generally not sensitive to the chemotherapy. That means when you give the chemotherapy, the cancer stem cells will be survive. Why they are surviving? Because they have a surface efflux pumps, drug efflux pumps present in the membrane. They will be effluxing the drug out of the cell, so that chemotherapy drug is not going to the cancer stem cells. As you know, the stem cells having property to again regrow as a rest of the tissue, they can relapse. Tumor recurrence can be possible. This is one of the reasons why the chemotherapy might be failing. That means 1% of the tumor is having a cancer stem cells which are resistant to the chemotherapy. Even you are removing the 99% of the tumor, the mass, the 1% of the stem cells which can regrow as a tumor relapse. That's the problem. That's why you need to target the cancer stem cells. How to target the cancer stem cells? What are the receptors present on that? What are the particular biochemical pathways I can inhibit the cancer stem cells to produce complete eradication of the tumors without any relapse? This is the strategy. That means you have to target the 99% of the tumor mass as well as the cancer stem cells. You have to target both of them together, not just one. The right strategy should be targeting both the things. If you only target the conventional traditional cancer therapy, it's only target killing the bulk tumor. A red one, the red one is the cancer stem cell will be producing relapse. If you are only targeting the stem cell, we are only removing the 1% of the tumor. The bulk tumor can again be promoting the cancer stem cell reoccur. The best way of eradicating that complete removal of the cancer is possible through the combination therapy, where you are actually targeting the 99% of the mixed population cells as well as the 1% of the cancer stem cells together. That means you may have to have a combination therapy. Because not all the chemotherapy drugs will be targeting the cancer stem cells. The cancer stem cell is a very widely researched area where people are developing a different type of drugs, nano formulations to inhibit it. That means the different ways you can target it. So if you see the cell, on cancer stem cells, different types of receptors are there. Some of them multi-drug resistant efflux pumps are there. They have a very good DNA repair mechanism. Generally, DNA repair mechanism is well controlled. But what happens in the cancer stem cells? They have a well-controlled DNA repair mechanism. In case the chemotherapy drug is going and damaging the kind of DNA, again the stem cell will be again repairing to normal. Because if you remember the anti-cancer drugs damage, which is required to produce anti-cancer therapy. Whereas the cancer stem cells has a very good DNA repairing mechanism. They will repair the DNA whatever has been damaged by the chemotherapy. And like that, you have different biochemical pathways, different intrinsic extension pathways, which will be actually contributing for the cancer stem cells property. That means you can inhibit some of the this particular moieties, produce it. This is the table talking about targeting strategies to cancer stem cells. You can target the surface markers, you can target signaling cascades, you can target the microenvironment, you can also target epigenetic mechanisms, you can also target other strategies. Likewise, you can actually need to have a therapy which is also taking care of the cancer stem cells. This is one aspect. That means you need to have a right combination approach. The aspect in the cancer therapy is the chemotherapy, which may not be properly distributing it. If you see the left side figure, this figure is talking about the yellow colored ones are the drugs. Due to the tumor is very hard in nature, the pressure is very high in the intratumoral pressure is high in the tumors. That's why tumors are harder in nature. They may not be allowing the drug, the drug to permeate throughout the tumor. That means that may not be going everywhere, every part of the tumor. You are giving a chemotherapy, but the problem is the tumor is not allowing the drug to intratumoral distribution. If that is not happening, what will happen? Drug only going to certain part of the body, certain part of the tumor, and remaining part of the tumor is not having any drug. Then what will happen? You are assuming that you are given the chemotherapy, but problem here is the chemotherapy has not reached to some part of the tumor. What is the specialty of that particular tumor? If they are survived, they are going to again grow further. That's how even 1% of the tumor is not getting the drug that can regrow again as a tumor. It can be cancer stem cell, even the normal cells. That means there is condition called the fibrosis. Tumor fibrosis. That means a tumor has a extracellular matrix highly dense and making a certain kind of barriers. It's making a extracellular matrix very thick so that drug is not able to diffuse through the barrier to reach to the some part of the tumor. That's how a tumor will be able not getting exposed to the chemotherapy. So what will be the right strategy? You can use the anti-fibrotic agents. If you can give the anti-fibrotic agents like TGF-beta inhibitors or any other anti-fibrotic agents, so that you can destroy the fibrotic situation in the tumor. Now followed by you give the chemotherapy. The right side figure is talking about that. Once you give the anti-fibrotic therapy, the collagen matrix or extracellular matrix has been removed. Now the cancer drug chemotherapy drug can distribute throughout the tumor. Then you can expect a better distribution of the drug. It can be free drug, it can be nanoparticle. The same experiments we have done in the lung cancer. Where orthotopic model as well as the metastatic model. Cancer can be the animal models, orthotopic model, the metastatic model. If you see the first figure, this is a controlled lung tumor where you can see the collagen. This is nothing but the blue color is nothing but the collagen. You see the kind of compartmentalization. There is a compartmentalization taking place. Some part of the tumor is having a bundle of the collagens forming a compartment. Once you have an isolated compartment, which is very very tight in the barrier, what can happen? Drug may not be going to that particular compartment. Then we can imagine the drug is not reaching there. Then the tumor will be able to survive, and after chemotherapy is over, these drugs are the particular the small part which is left due to the collagen accumulation in the form of tumor fibrosis will be regrowing again. So we have used a telmisartan and losartan, because telmisartan and losartan are the FDA-approved antihypertensive drugs. But if you see the particular pathway of the angiotensin receptor, AT1 receptor, they also activate the TGF-beta, transforming growth factor beta. TGF-beta is the main factor involved in the fibrosis development. So based on that, we have used telmisartan and losartan. We have removed the fibrosis present in the lung cancer. This I have done in my postdoctoral work, where we can see the blue color has been reduced in the losartan treated group as well as the telmisartan. That means you could able to reduce the collagen deposition in the tumors by giving a TGF-beta inhibitors like losartan. Then followed by, you can imagine that if you give the chemotherapy, that will perform better. We have demonstrated, in fact, that particular activity where We have seen the improved distribution of the drug into the tumor and resulted in the better anti-cancer activity. We have published this particular work in the Journal of Control Release during my postdoctoral work. Similarly, lysyl oxidase 2 is the enzyme, lysyl oxidase. There is a monoclonal antibody developed against this particular enzyme. Lysyl oxidase is the enzyme which is responsible for the collagen monomers to form the polymers. The collagen monomer, all the monomers will be forming bundles to make the polymer, the collagen polymer. The collagen polymer is nothing but your extracellular matrix of the tumor fibrosis. You know that there is a very good link to the fibrosis as well as the metastasis. The more fibrosis the tumor is, more metastasis will be the tumor will be. So there is a monoclonal antibody developed against the lysyl oxidase, where they have evaluated how metastasis can be reduced by inhibiting the lysyl oxidase. The clinical trials are going on. So similarly, though they developed the monoclonal antibody, we have used the shRNA, small hairpin RNA, which is the RNA which will be inhibiting the specific sequence of the lysyl oxidase 2, so that I will be able to knock down the lysyl oxidase 2 and see what is happening to the tumor. When we did this experiment, what happened was we could see the increased, when I give the shRNA, we could see the protein expressions are altered, collagen expression is altered. All the markers which are in the control tumors are higher compared to the lysyl oxidase shRNA, where we could see the reduced expression on these particular markers. Quantitatively, we have done it with this Western blot data. When we did the distribution of the drug, a nanoparticle, we could see in the control tumor, TNBC means triple negative breast cancer. In the triple negative breast cancer, which is very aggressive, we don't have any proper therapy. Then again, the drug is not distributing in the tumors. You see the red fluorescence is less here because the drug is not distributing it. The nanoparticle is not distributing it. How did you see this? By fluorescence nature. So when we give the lysyl oxidase shRNA, we could see the improved distribution. This is the collagen staining by the Magnum tricon, the picoserous red staining, and also after treatment, we can see the collagen, the extracellular matrix has been reduced. What I'm trying to indicate here is, when you remove the certain, when you inhibit the lysyl oxidase 2, which is contributing to the collagen deposition, we could see the increased penetration of the drug into the tumor as well as the better anti-cancer effect has been seen. This is the breast cancer model where I have done again my postdoctorate. The tumor volumes are much well controlled. If I remove the lysyl oxidase 2 initially, followed by if I give the chemotherapy, doxorubicin liposomal formulation, I could see almost complete eradication of the tumor from the nude mice model. This is study done by me during my postdoctorate. So what I'm saying is, you have to use the right approach. You can actually prime the animal or the diseased tissue condition to a certain therapy where it is going to normalize the tumor, so that whatever the hurdles are there, you remove the hurdles first, followed by give the chemotherapy. Just give the chemotherapy, that may not be really effective. So you have to have a combination therapy, a segment, a stepwise approach where you prime the tumor, you pre-treatment, treat the particular condition with a strategy where you are trying to reduce the cancer stem cells, or you're trying to reduce the fibrosis, or you do some other things, followed by the conventional chemotherapy that may be more effective here, in the form of tumor volumes as well as the tumor images here. So this is some ways where we can actually fight against the cancer. Some of the approaches have been, these are the approaches I have tried during my postdoctorate. So now I'm actually shifting to another important area where I have been working in the nimbolide. Even this has come in the Times of India during my research work, cancer as well as the inflammatory models. You all know that nimbolide, neem is a sacred plant. We use it for different the reasons, like curcumin. But in compared to curcumin, the neem tree is having more activity. Curcumin, which is having a 20 micromolar, is the IC50 value to kill the cancer cell, whereas nimbolide only needs 2 micromolar to kill the cancer cell. That means it is 10 times more potent than the curcumin to produce anti-cancer activity as well as anti-inflammatory activity. Again, it's a multi-pharmacological, it is having multi-pharmacological properties, where this molecule has been not well studied. Though we all know that nimbolide, the neem is very good. We have been using for some of the agricultural purpose, where your urea has been coated with the neem oil, but that is for some other reason. But we are not having to explore them properly. And we will not be surprised that the patents goes to other countries if they develop the new derivatives of the new formulations. With the background, what we have done the Nimbolide, how that can be used in the different pharmacological tools. That's where we have done on our experiments. What we have done was Nimbolide, we have studied in the inflammatory models. The model is nothing but acute respiratory distress syndrome, or SARS, severe acute respiratory syndrome. We all know that this is common in the COVID. Nothing but your lung will be highly inflammation. So due to that, you will be having a breathing difficulties. That's even can result in the lung function failure. This is the common reason. That's called ARDS, acute respiratory distress syndrome. What is the pathology involved in that? One of the pathologies, the IL-6 pathway is involved in that, as well as TNF alpha pathway is also involved in that. So what we identified was, when we had done the docking studies, molecular docking studies with the Nimbolide, with the TNF alpha, or different inflammatory mediators, we found that Nimbolide is very strongly binding to the TNF alpha. That means it is inhibiting TNF alpha. And we have done this before COVID, in fact. And then once we understand that this is binding to the TNF alpha, then we have done the experiments in the in vitro cell lines, in the macrophages, where we have induced the inflammation through the LPS, followed by we have given how the Nimbolide is protecting it. And we found that actually Nimbolide is actually reducing the expression of TNF alpha. That means it is acting like an anti-inflammatory agent by inhibiting the TNF alpha pathway, which is very potential, which can be a potential in the COVID-induced lung inflammation conditions. After doing the in vitro experiments, we have also done the in vivo experiment. How do we do in vivo experiments? We are getting lipopolysaccharide, which is an inflammatory inducing agent, lipopolysaccharide, bacterial endotoxin. When we give this particular LPS to the mice, it is causing a severe TNF alpha accumulation in the lungs. That indicating that, okay, lungs are highly inflammation in nature, and also there can be the chance of developing a liver lung failure condition. When we give the Nimbolide at different concentrations, 0.3, 1 and 3 mg per kg by intraperitoneal route administration. See, the dose is only 3 mg per kg. We could be able to reduce the TNF alpha expression in the lungs. That itself is indicating that, okay, it's protective. So what we have proven was Nimbolide is inhibiting the TNF alpha. It is also inhibiting the histone deacetylases to prevent the different inflammatory cytokines, inflammatory cytokine cascade. It is also inhibiting the reactive oxygen species, so that through anti-inflammatory and through antioxidant and anti-TNF alpha property, it is able to protect from the acute lung injury, which is seen in the LPS model in particular condition. But the same can be the same is the pathology observed in the ARDS, COVID-induced ARDS conditions. So this is what the paper we have published before COVID. Nimbolide protects against endotoxin-induced acute respiratory distress syndrome by inhibiting TNF alpha mediated NF-kappaB ester nuclear translocation. So we have published this particular work in Cell Death and Disease, which is a Nature Publishing Group journal. So this is one of the nice experiment we have done with the Nimbolide, where we have proven that anti-inflammatory property of Nimbolide. We all know that Nimbolide is anti-inflammatory, but how it is needs to prove it. No? We have done it. Again, the success not ended there. The problem we were facing was Nimbolide is not orally bioavailable. It's poor solubility. It is not. It's only active if you give the parental route administration. You have to give IP or IV. It is not. It is easily penetrated in the aqueous condition. That means there is a need to develop the formulation. We have developed the IRGD conjugate liposome formulation. What we have done, since Nimbolide is needs to be given intravenous administration, peritoneal route administration, we made a liposomes, and we made the liposomes conjugate with IRGD. The IRGD is a peptide which is selectively taking my liposomes to the inflammatory sites, lung inflammatory sites. So what we have done, so we found that IRGD increased the distribution of the nanoparticles into the inflammatory sites, and we have given this particular formulation by inhalation route. That means you do the Nimbolide, we want to improve the activities through the liposomes, conjugate with the IRGD peptide, and we have given inhalation. So by doing this, we could see that it is better protecting in the ARDS conditions upon inhalation route of administration compared to the free drug. Again, this is the histological studies to say that in the LPS there is more inflammation, more neutrophils and other mast cells accumulation, whereas IRGD formulations have reduced it. Compared to dexamethasone, which is used as a standard anti-inflammatory in the different acute inflammatory. So if you see this particular paper we have published in Nanomedicine, Nanotechnology Biology, that if you see the title, IgG conjugated nimbolide liposomes protect against endotoxin acute respiratory distress syndrome. So we didn't stop there. We have continued improving the nimbolide properties by making a formulation. So now we can say that maybe this formulation, if we can do further studies, can be a potential strategy to treat different inflammatory conditions, even ARDS. What is the consequence of the COVID? If you see the COVID is a lung condition where you see the severe inflammation in the lung, which can be causing a lung fibrosis. Also, fibrosis, extracellular matrix accumulation. The normal lung should supposed to be like this, where good CO2 and O2 exchange should be taking place. Whereas in the fibrosis, you can see, due to deposition of the extracellular matrix, the gas exchange is also not taking place. And the survival of the patients due to pulmonary fibrosis is less than 5 years. People will not survive. You only have 2 drugs which are also not very effective. That means pulmonary fibrosis is a very serious concern reported post-COVID. However, the pulmonary fibrosis is not a new one. We have been seeing the pulmonary fibrosis from earlier also. That means there is increased chance to develop pulmonary fibrosis due to the COVID, as well as the different environmental factors, as well as the tuberculosis, COPD. These all can be causing increased pulmonary fibrosis. So what we are proposing here is, can we use the same nimbolide, which is anti-inflammatory in nature, can to develop a meta-dose inhalers at the dry powder inhalers at the nebulizers, so that we can able to send this particular drug into the lungs, so that localized delivery to treat pulmonary fibrosis at the ARDS can be possible, so that we can have best lung exposure. No systemic exposure can be possible, so that a dose will be much lesser, and you can also get the sustained effect. Since we are sending the drug directly into the lung, it can be a more sustained effect compared to the intravenous administration. The problem is, if you take a tablet, if you take injection, I'm not sure whether 5% of the drug is going to the lung or not. One of the best way of sending a drug, if we can, is through inhalation route of administration. That's why even the COVID vaccines, people are trying to develop now the inhalation exposure. Vaccine is fine because the memory will be developed. Whereas what are the small molecules, what are the chemical drugs, they don't have any memory. They don't have any certain amplification system. So that's why you need to have a right delivery needs to be happened. That's why it can be inhalation delivery can be a potential approach to treat the ARDS, even for the lung cancer or the pulmonary arterial hypertension, many conditions. So one of the area I'm working is on the pulmonary diseases is through the inhalation routes of exposure, where we are trying to see how nimbolide can be delivered. These are some of the things where my research experience, as well as the recent trends I'm talking about. Now I'm shifting to the safety aspects of it. There is ICH is giving a guidelines in the form of quality, safety, efficacy, and multidisciplinary. Every aspect is important. ICH QSEM is very important, right? The safety guidelines are most important than the rest of the guidelines. Number of preclinical studies required for establishing the safety are much larger than establishing pharmacological efficacy studies. That means you need to sacrifice thousands of more animals than the just to prove the pharmacological efficacy. If you have to say the anti-diabetic activity compound, what will you do? You develop the 2 models or 3 models, done. Whereas whether the compound is safe or not, you have to do all the battery and the toxic studies, acute, chronic, reproductive, local toxicity, genotoxicity, and so on. You will be doing it. One of the important requirement is, if you are using a drug for more than 6 months, like anti-diabetic and anti-hypertensive, you have to, any compound which is used more than 6 months, you have to do the cancer testing. That means the carcinogenic potential of the compound is studied. Earlier we are talking about how the cancer is developed, how you treat it. Here we are with respect to toxicology, you have to see how a drug which is being approved is being under the investigation can be a carcinogenic or not. Any drug which is carcinogenic cannot be approved. That's why you have to do the carcinogenicity studies in animal models. Generally, it takes 2 years. Just imagine, just one essay. Hello? Hello? How much time I have, madam?
Have, sir, because we
Madam?
started a hello, sir.
We have enough time, right?
You can continue, sir.
I have a lot of slides. That's what I'm telling you. I'm
How many slides are there, sir?
I guess I am halfway.
No problem.
I can rush it. I can rush it a little faster. Okay. No problem. I'll skip some of the slides. Don't worry. I'll skip some of the slides. Don't worry, madam. Yeah. So just imagine you have to do the 2 years rat study. Just one experiment is 2 years. Then only after 2 years only you'll come to know whether the compound is having any risk of causing a cancer or not. And you have to keep in each group 50 animals. Why 50 animals? Within 2 years time, there may be many animals dying. So you have to sufficient number of the animals to see whether the compound is a carcinogenic or not. That means it's very costly. It's very lengthy in procedure, and it may be taking a lot of time. And after 2 years, if you found the compound is carcinogenic, what will you do? Your 2 years is already lost. The amount you have spent, the people who work on the particular molecule, you have to wait for 2 years. That's the problem. That's why you can develop the models like short-term models. There is a cell transformation assay, in vitro based prediction of the carcinogenicity by the cell transformation assay. Cell transformation assay itself tells what is cell transformation. The cell which is normal will be becoming a malignant due to transformation. Can we do that? That means I don't need to wait for 2 months or 2 years. I can just do the in vitro study where I will incubate my compound with the cell line, suitable cell line. The cell line is nothing but the Syrian hamster embryo cells. And this particular cell, due to transformation induced by my suspected molecule, will be forming a transformed cell, which will be having a abnormal growth properties compared to control cell.
మెయిన్ 11:30 చెప్తే ఇంకో స్పీకర్ ఉందా?
That's how it will be telling. It will be possible that a short-term assay is possible in the form of cell transformation assay, so that you can predict whether my molecule or the new drug is having any risk of causing a cancer or not. So that within 1 month time, I will be able to know positive or negative. If the negative, I will go to negative another way. So there is a link between the carcinogenicity and genotoxicity impurities. Nitrosamine impurities. Why recently there are drugs been withdrawn from the or recalled from the some of the batches have been recalled because some of the batches are found to be having a genotoxicity impurities. Why genotoxicity is so important? Any compound which is causing a genetic toxicity, genome damage can cause the abnormal cell. Due to abnormality, they will be causing a cancer. That's why you have to study the genotoxicity. What is the link with the genotoxicity to cancer? A compound, a new drug, which is a direct DNA damage and indirect DNA damage, can cause a cancer because any genetic instability due to genotoxicity can cause a cancer. So there is a link. We have to study carcinogenicity, you have to study the genotoxicity, and compound which are found to be negative only. These molecules will be only taken further development. They never be taken to the clinical trials until unless you prove that they are free from genotoxicity. They are also free from the carcinogenicity. Another important area is the safety pharmacology, where these are the secondary pharmacodynamic effects which will be impacting the physiological condition functions. We don't want the physiological functions to be altered. Your heart rate is supposed to be this much. Your respiratory function is supposed to be this much. Any drug which is altering the physiological function is problematic. Some of the drugs are found to be having this particular property, where the main systems are CNS system, cardiovascular system, and the respiratory system. That's why the safety of pharmacology has come in the form of ICH S7A. ICH S7A guideline. S7A guideline is talking about safety pharmacology, where 3 vital organs needs to be evaluated whether the drugs are actually causing any abnormality in the physiological function of this particular organs or not. And there is S7B guideline, which is exclusively talking about how the drugs are cardiotoxic on the ECG pattern, electrocardiogram. You know that the QT wave, which is supposed to be certain the duration, can be extended. That's called the QT wave interval prolongation. If the interval is prolonged, the rhythm of the heart will be altered. If the rhythm of the heart is altered, that can cause a condition called the torsade de pointis. Torsade de pointis is nothing but a ventricular arrhythmia due to Q2T interval, which is prolonged. The interval is prolonged, which is causing a arrhythmia. The arrhythmia can cause the abnormal heart rhythm that is causing the torsade de pointis. which can be identified through the ECG. You can also study the by using. So example of the drugs causing is terfenadine. Terfenadine is a antihistamine which is converted to fexofenadine. However, if you are using a drug combination drug with the ketoconazole or the any CYP3A4 inhibitors, so the conversion of the terfenadine into fexofenadine will be inhibited. Unfortunately, the terfenadine is having a inhibitory property in IKR potassium channels. IKR potassium channels are the ones which are responsible for the QTO prolongation. If you see the action potential of the cardiac myocytes, this is supposed to be like this left curve, whereas it is getting extended because the ion channel, which is IKR potassium channels, that are also called the delayed rectified potassium channels, are found to be inhibited by the terfenadine. Due to that, the QT wave syndrome is happening, and where you have to do the heart rate correction, because heart rate may be altering from patient to patient, from person to person. That's why corrected QT wave, that means heart rate corrected QT wave, needs to be understood. So there are many drugs have been withdrawn. There is a link with the QT wave prolongation and the CYP3A4 inhibitors, because all the drugs which are inhibiting CYP3A4 also found to be having a QT wave prolonging effect. So these are the drugs been withdrawn due to TDP. What is TDP? Torsade de Pointes, which is a French word. Terfenadine, cetendol, astamazole, these all drugs are not available, and these all been withdrawn from the market after their approval for the clinical use, due to the only reason is torsade de Pointes. Now all these drugs have IKR channel blockers, which are the potassium channels which are involved in the prolongation of the repolarization of the cardiac myocytes, and these are all the CYP3A4 substrates. That means either metabolism is altered or the renal function is altered. That means while these drugs are used, if the renal function is altered, drug levels will be increased. The CYP3A4 inhibitors or substrates are there may be a increased levels of the drug because the inhibition of the drug metabolism. Based on this, there is a ICS S78B guideline has come. They have developed they have made it mandatory to develop a assay called the patch clamp technique. Patch clamp technique is nothing but where you have to study the potassium currents, how the potassium currents through the IKR potassium channels are altered. That means all the NCEs which you are developing it, you need to do the patch clamp assay, which is electrophysiological assay, where you will be studying how the potassium currents are altered in presence of the particular NCE and in absence of the NCE. In case any compound which is inhibiting the potassium currents, that means it is inhibiting the IKR potassium channels. You can do the particular assay, and only compound which are found to be not inhibiting the IKR potassium channels will be taken further for the parallel. That's a patch clamp assay. It's an inhibitor assay where it will be done to perform it. The one of the another assay, this is something about the safety guideline. So we can also talk about the episkin models, episkin, epiderma, the skin ethnic. These are the different models where you know that cosmetics cannot be studied on animals. Cosmetics are not life-saving drugs. Similarly, you can have the 3D models. 3D models in the sense skin can be produced in outside the body, where you can take the keratinocytes from the skin, dead skin or the surgically removed skin. Keratinocytes can be grown on the artificial membranes, and they will be growing as this. In fact, this figure is HNE staining of the in vitro skin, where you have grown the skin outside, and the skin can be used for different applications, for the skin irritation, skin toxicity, skin corrosion, transdermal drug delivery. This all can be done by using a skin. So this is another alternative model. This is a recently come up area. So this can be very important for the cosmetic safety evaluation, but you can also use for different other conditions. Not only for the skin, if you are taking the airway cells, it can be epi-airway. If you are taking from eye, epithelial cells, it can be epiocular, epicorneal, epivaginal, epigingival, epioral. Like that, you can actually have any kind of the cells from where you are taken. You can grow them outside the body and a membrane in sterile conditions. They'll be growing as a tissue. The tissue can be used for experimentation. Maybe you can use even for the COVID infections and all. So while talking about the pharmacology and also while talking about the toxicology, if you see that though you are doing hundreds of molecules in the preclinical studies, only 8% of the molecules, less than 8% of the molecules can be translating into the clinical. That means clinically approved. That means 92 molecules are actually getting failed. That's why we cannot wait for 12 years to see the molecules getting failed. Early killing will be the right of the molecule is very important. You have to kill the molecules early. That means you do all the assays in the early stage, say that, okay, this molecule is not suitable because there is a suspected moiety, there is suspected the profile of this particular drug on the safety. This can be causing a damage later. That's why early killing is very, by killing early, what we can do, you can save the time so that you are not performing unnecessary experiments which are taking a lot of time and you are unnecessarily investing your time and effort to develop a wrong molecule. That's why early killing is very important. So you can save the time, you can save the clinical trial participants, you can also benefit the organization. That's why the organizations and the people will be using a different tools to predict whether my molecule is safe or not. That's where you have the tools, software tools. In silico predictive toxicology is there, where you input the structure, the chemical structure into the computer. It can predict whether it can be mutagenic or it can be carcinogenic or it can be reprotoxic. So that I know that these molecules, only the moieties which are having a no alerts, no toxicity concerns here only will be allowed to further execute. That means you're not even performing the experiment, you're only designing the molecules in the computer, and based on that you're predicting it. The molecules are having a safer prediction profiles will be only taken for the further experiment. We all know the thalidomide strategy. This could have been avoided had it been we have a good toxicity guidelines. We have the good toxicology programs. Earlier, the toxicology programs were done in a very casual manner, where there were no reproductive toxins required at the time, and there was no proper documentation of the studies done on the animal models. If you don't have any documentation, I don't know what kind of safety of the data generated. You can say that I have done it, I don't know where my documentation is. That's why we have we have that thalidomide, which has caused more than 5,000 people have born without limbs, called the phocomelia. And later it was found that when they did the reproductive toxic studies with the same molecule, they found that yes, this drug is causing a fatal toxic effect. That's why the regulatory the agencies have made it more mandatory to develop the stringent toxicity guidelines. And that too, the toxicity needs to be done in the good laboratory practices. Why good laboratory practices? What is the definition of the good laboratory practice? Where the toxic studies is a quality system concerned with the organizational process, where non-clinical and environmental safety studies are planned, performed, monitored, recorded, and reported as per the study plan. That means every step of the toxic study, doing animal studies, will be done as per what you have planned, what you have performed, everything. That means everything is controlled. Nothing can come out of the particular study. You cannot remove one base, you cannot remove a test compound, you cannot add extra animal. Everything is controlled properly. So how the test item has come, how the compound has come, how the animals have come, how we have treated it, everything is documented. Raw data will be properly stored for the next 9 years after the study. So that even after 9 years, if somebody having a suspect on the particular data, they can always come back to the particular GLP lab and say that, can you show me this data? That's how GLP has really become very vital for the non-clinical safety evaluation of the compound, so that data authenticity and data reproducibility will be improved. So otherwise, there is a possibility that you can have a tragedies like thalidomide can happen. So there's always a link between the toxicology and the GLP, and we have a lot of GLP labs in India where the compounds will be evaluated for the safety by using this. While doing the toxic studies, we'll be trying to develop the different parameters. We'll be developing the therapeutic index as well as the margin of safety, as well as the NOEL and LOEL. What is a NOEL and LOEL? NOEL is nothing but no observable adverse effect level. If you give the dose response curve by toxicity concern, the no adverse effect is the one where the concentration up to that you are able to tolerate it. That is a safer dose, you can use it. Based on the safer dose, you will be able to calculate the human dose. That's what how do you calculate the human dose? First time human dose, human equivalent dose will be calculated based on the data you are generating the animal models. That means you are doing the animal toxic studies. You found that this is the dose found to be safer. Based on the safe dose, you will be calculating it. How can you calculate the human dose? Human equivalent dose in milligram per kg is equal to NOEL of the animal multiplied by animal km by human km. What is the animal km? There are some km values. Our humans is 37, and for the mouse it's 3, for the rat it's 6, for the guinea pig is 8, and for rabbits 12. Whether you have the human km values, based on the different km values, you will able just have to keep this particular the NOEL value of the data you have generated from the animal toxicity data, and you will be able to calculate the human safer dose, or the human equivalent dose can be calculated. This is important for us to understand it. You can able to search also this one, how the human equivalent dose is calculated from the animal toxicology data. First time human dose calculation can be done because that compound might have been not given earlier to the patients. Similarly, I'm talking about the omics. the genomics, proteomics, the metabolomics and all. The omics is nothing but as a whole. If you are studying a particular aspect, you are not studying one parameter. You will be studying a group of proteins, or the group of genes, or group of metabolomes together. What will happen? So that you will have a clear. Your sample size is larger. The parameters you are evaluating are larger, so that you will be able to better understand it. Similarly, you have the genomics, you have the proteomics, where you will be you have the transcriptomics, how the messenger RNA is altered can be studied to understand how the disease is altering the particular gene expression, how the particular compound is causing a damage, the toxicity can be studied. Genomics or proteomics or metabolomics can be used for you to understand the new targets as well as to understand how the drug is effective. Just only depending on the animal mortality may not be sufficient. You may have to study at the molecular level, at the gene level, to see that what kind of the alteration happening at the largest set of parameters. So again, proteomics, where you can actually take the protein. The protein can be categorized into normal control and disease control. If you see that in the particular disease control, this particular proteins are altered, that means okay, this protein will be a suitable target. So you can understand the protein patterns, gene patterns as metabolomics patterns. Certain, you have almost 2,800 metabolomes are there. Metabolomes will also give you to understand the target as well as the safety. So so you can integrate basically. You can integrate the transcriptomics, proteomics, metabolomics, functional genomics, structural genomics, and lipidomics. Lipidomics in the sense what are how the lipids are altered in the disease condition, how the lipids are altered due to treatment. So you can integrate them to understand or predict that okay, compound is safer or not safer. So that means omics tools can be also used for us to understand, so that if we, by using omics, we will be able to say that compound may be not toxic. Once I have a more set up the biochemical study to understand whether the compound is toxic or not, so that I have more confidence that my molecule will not be failing later due to the clinical. Since drug discovery is very risky, you can always do the drug repurposing. What is drug repurposing? Is the finding an unknown known drug for the new therapeutic activity. Repurposing, metformin is anti-diabetic. You can say that it's anti-cancer through your experiment. That's repurposing it, so that you can save that time, because drug is already known to the people, so that it's a safe, effective drug and safer because this can already been used by people. It can be a new therapeutic effect to the known target. It can be a new target. So the drug, which metformin, may be binding the same target and causing a new therapeutic effect, or it may be binding a new target. So how did you identify the new target? You might have identified the new target based on the omics you have done it. That means you have given the metformin, and without metformin, you have seen that after metformin, I have seen a particular target is altering. That means it's binding to altering the new target, so that this target is found to be having a role in the disease. Right? See that drug repurposing can be much effective, where you don't need to do all the lead optimization and all the studies. You only have to do some clinical trials and the registration, you can get approved. So time is much lesser. Risk is lesser. However, the reward also will be lesser, because you're just repurposing. You will not get a lot of reward, but you will get some profit. But there is no risk involved in that, because the drug is already safer. That's how the repurposing can be a safer strategy. Sometimes if you want to bring the drug immediately, like COVID conditions where you need a drug immediately. These are the examples of the drug where people have done the repurposing. You all must be knowing these examples. Thalidomide, which is the teratogenic, but now it's used for the multiple myeloma and leprosy and all. Similarly, we have done a niclosamide, which is an anti-helminthic drug. We have used in the psoriasis, where we have done the psoriasis studies. The psoriasis can be very nicely treated by using a niclosamide, which is an anti-helminthic drug. So it's again strategy we have used here to see that how psoriasis can be treated by using a already existing drug, niclosamide. We have done that. So my area is biologics. What are the biologics? Nowadays we are talking about the biotherapeutics, the biologics. So far we have been talking about the chemical drugs, chemically synthesized drugs, but these are having a lot of off-target effects. They are having drug-drug interactions and so on. So nowadays we are having biologics which are very selective, because biologics are developed to specific particular protein. It can be monoclonal antibody, it can be any other agent, so that biologics can be more specific to bind to the disease side, so that less side effects can be expected. Only the problem with the biologics is the immunogenic potential, because the biologists are the biological therapeutics, they may be having immunogenic. Otherwise, biologists are much safer in nature compared to that. What is the problem with biologics? Biologics are very costly. You cannot able to afford it. Example is these are some of the biologics. Of course, insulin is cheaper nowadays, but in the rest of the biologics are very expensive. Erythropoietin, which is used for the chronic kidney disease, and herceptin, which is anti breast cancer drug, streptokinase, adalimumab, and so on. We have different types of biologics. How the biologics are produced is nothing but you can actually use the recombinant DNA technology, where the gene of interest will be expressed in the suitable vector. You are hijacking a host cell with a gene of your interest, so that that host cell will be producing a gene which will be converted into protein. That means you are making a recombinant cell which will be expressing a gene which will be producing a therapeutic of your interest. It can be a monoclonal antibody. You can use different type of systems for the recombinant protein production, unicellular system as well as multicellular systems. How that is produced is you will be developing the cell bank, fermenting them to produce a large scale. Then after you ferment them and for the larger number of the cell production, then you will be purifying them by using a different chromatography technique, affinity chromatography, size excision chromatography, and so on. Based on that, you'll be after separating that, you'll be purifying them. Otherwise, if you are not purifying the protein, you'll not able to get the exact therapeutic benefit, as well as you'll be also having a lot of immunogenicity, because the protein which is not purified can cause a lot of immunogenicity. That's why it's very important for us to properly purify the protein before going them. That's why one of the biggest problems in biologics is the purification step is very extensive. Similarly, the vaccines can be produced by using a same way, where you will be vaccines can be produced in the chicken eggs or the cell cultures, where the virus can be used, or the attenuated vaccine can be used, or you can also use the recombinant technology, where you can only spike protein which is having a gene can be into plasmid that can be used into a suitable vector, and they can use them to produce the vaccine. So different ways a vaccine can be produced here. I just given the slide here, where you have to grow the virus where you want to which you want to develop the vaccine. Then virus will be grown in the suitable cells. Then the cells will be broken. The viral particles will be collected. I will be using them. You will be killing them. I will be making the fragmented. Even the fragmented one also will be having a particular moiety, which will be also immunogenic. That's how you'll be able to produce immunity. Or whatever fragment is having a gene, you can actually produce through the recombinant vaccine can be also possible. Similarly, monoclonal antibodies produced through the hybridoma technology, which is a very promising technology, because all the biologists are produced through the hybridoma technology. Most of the biologists are monoclonal antibodies, and they are very expensive. The reason is they are very difficult to develop. However, techniques used is the hybridoma technology, where B cells which are producing the monoclonal antibodies will be hybridized with the multiple myeloma cells, so that myeloma cells will be highly proliferating cells, and the B cells will be the cells which are producing the monoclonal antibody. If you combine both of them, the cells which are having a property to produce the monoclonal antibody as well as the rapidly proliferating will able to produce more amount of the monoclonal antibodies, and you can purify this monoclonal antibodies, can use them. This is the second, where you are immunizing the animal through the antigen, so that B cells are isolated. B cells which are producing particular monoclonal antibody, then you will be hybridizing with the myeloma cells, so that hybridized cells will be selecting them which are producing the spleen cells will be having different types of B cells. You have to select only cells which are hybridized and properly produce the monoclonal antibodies. So this is a very important technique, where many of the pharmaceutical companies in India are worldwide are using a different type of biological productions. One of the hybridoma technology, where you can use it. These are some of the examples of the monoclonal antibodies where being used for different disease conditions, mainly for the cancer, autoimmune diseases, as well as other infectious diseases. Example is tocilizumab. We all know that tocilizumab, how much we are struggling. Why we are struggling with the tocilizumab? Because this was not available in the country. It's a simple biologics. How this can be produced? You have to use hybridoma technology. We don't have hybridoma technology properly developed, and we don't have patent rights for that. That's why we are struggling during the COVID time. And it found to be more than 50,000 for a while for single administration. That's where we are lagging here. So since sometimes
If you don't mind, shall we Can you wind up in a few minutes, sir?
Yeah, madam. 5 minutes, madam.
Oh, yes, sir. Thank you. Thank you so much.
You can also use the mouse models, the humanized mouse models, because the humanized mouse models are the better models compared to the just mouse models. What are the humanized mouse models? Humanized mouse model nothing but the mouse which will be engineered to get a gene of the human gene into the mouse, so that they'll be producing the antibodies, or what are the particular the proteins which are of the human nature, so that we'll be able to better predict how the mouse is behaving to the particular biologics, particular drug, compared to the simple mouse model. That's how humanized mouse model. That means a mouse which is trying to become a humanized. So you'll be trying to express some of the genes or the proteins which are specific to the humans, so that you can better study them. Right. So that's what is called the wild mice, which is producing a certain protein, can be made into humanized mouse by altering the particular by the modulating the expression of that particular gene of human interest into the mouse by using a different transgenic approaches, so that you will be getting a protein of your interest, the protein of interest, antibody, or whatever it is. With this, I'm just closing this up, my one, and I must thank my director, Dr. Shishbala Singh Madam, for all the support and for our leadership to motivate us to do out of the box kind of research. I must thank all the DOP and other funding agencies like DST, DBT, and all. DOP is nothing but a Department of Pharmaceuticals, which is funding us our research. And this is NIPER Hyderabad campus, where we have a very beautiful lab. We have the sites where you can visit, and sometimes you can come. Yesterday we have conducted Academia Connect, where many of the pharmacy colleges have attended our institute. Try to see how the facilities can be explored or extended for different people for the practice, as well as we are also supporting internships for the candidates to come to NIPER, because we have state-of-the-art infrastructure. However, they are not reachable to some other people, where we wanted to at least some of the people who are highly motivated can come here and do the research activities in NIPER Hyderabad as internships. With this, I'm closing again. I wish you all very happy Sankranti. I'm very sorry if you have taken more time. If you have any questions, I will be able to answer to this. Thank you so much, madam. Thank you for the opportunity. I'm very sorry for crossing my limit.
Sunita, take over.
Okay, ma'am. Any queries from audience?
Chandra, sir,
Yes, madam.
I have a doubt.
Yes, madam.
So this terfenadine and fexofenadine have same cardiac toxicity?
They cause the QT of prolongation. Actually, terfenadine is becoming a fexofenadine.
Okay.
So what that is converted in the body. But what happens is, if the conversion is not taking place because of CYP3A4 inhibition by ketoconazole or any azithromycin category of the compounds, so terfenadine is building in the body. The terfenadine is inhibiting the IKR potassium channels. So that's why they have banned terfenadine. Fexofenadine is available.
Yes, fexofenadine is available in Allegra. Yes, with Allegra.
I don't know the brand name. It's available, but terfenadine.
Is it safe to use fexofenadine?
Yes,
alone?
it's safe to use fexofenadine, but not terfenadine. The reason is terfenadine is actually is inhibiting IKR potassium channels, but not fexofenadine.
Okay.
The conversion is not taking place in the body.
This Allegra, that's a doubt.
That is safe. That is safe. No problem.
Okay, fexofenadine is safe.
Yes, that's safe.
So what are the minimum safety evaluation procedures for a Ayurvedic preparation? Recently, we want to prepare turmeric drops for use.
Okay.
So like in the form of ethosomes. So this has to be taken 4 drops in 100 ml of water and drink. So any safety evaluation procedures or need to be performed before
For sure,
use?
for sure you have to do, for sure you have to do, acute toxic study. What is the route?
Okay.
What is the route? It must be oral. So you
Yes.
have to do the acute toxic study. Just
Okay.
prove that. You can also do the repeated dose toxic studies. Mainly for the Ayush or the Ayurvedic formulations, there are no clear-cut guidelines.
Okay.
This already known. These drops are these the products are already known to be safer. We have been
Okay.
using it. So if minimal toxic studies are required for the herbal.
What are the minimal toxicity studies that can be done for this particular turmeric drops? Like minimal.
You have to do the bacterial toxic studies, like you can prove the genotoxicity, you can prove the acute toxic study, you can do the 28 days repeat dose toxic study, you can do the local toxic study. I may have to see that exactly because it may be varying from the product to product actually, but
Okay.
simple toxic studies can be done in fact.
Okay. Right. Thank you. Thank you so much. So your lecture is very wonderful, very informative. Actually,
I don't know.
One day full webinar I need to keep. Definitely, I'm planning in my mind that I will have to keep one whole session of a day. Yes, it is never-ending process. Every aspect of your topic is very, very important, and depth of the discussion is needed, and you are discussing a lot. So thank you so much for that. Yes. Thank you so much for that.
Yeah, sorry.
Yes.
When I was making this talk, what is happening? I feel everything is important. That's why I could not able to remove my 50 slides, but I could not do that. So I thought
one day, a whole day webinar, like at least half day, 2 hours webinar we wanted to keep.
Probably I have bored you people. Looks like.
No, not at all. It is very interesting. Yes.
Thank you, madam.
Okay.
Thank you, sir, for your valuable information on how pharmacology plays an important role in drug discovery and development. And also you have clearly explained what are the various steps in drug discovery and development and briefly explained about cancer targeted therapy. Thank you, sir, once again for your valuable information.
Thank you so much, madam. Thank you for the opportunity and wish you all the best the rest of the conference. I know that must be waiting to join. Yeah.
Now I invite Dr. P. Harati to introduce Dr. Taluri Narendra Garu.
Good morning, everyone. Myself, Dr. Harati, Associate Professor of Tigal Krishna Reddy College, take pleasure to introduce Dr. Taluri Nadavendra Kumar Garu for the second day of two-day webinar of ACPS 2022. Dr. M.V. Narendra Kumar Garu, working as an Head AR&D Knowledge Management at Dysel Chiral Technologies India Private Limited, and previous positions held by him include Professor and Head Pharmaceutical Analysis, in charge LCMS, Central Analytical Instrumentation Facility at Naipur, Hyderabad, and Scientific Manager at Biocon, Bangalore. He received PhD degree from CSIR, IICT in Chemical Technology, Hyderabad. He has published 90 scientific articles in his credit. He successfully supervised 50 MS Pharmacy and 4 PhD scholars for their research works. He is recipient of CSIR Research Fellowship, Institution of Chemists Associateship Awards. He is member of Scientific Societies, Royal Society of Chemistry, London, Asian Council of Science Editors. Science Direct declared 4 times one of his articles published in Journal of Pharmaceutical and Biomedical Analysis was among top 25 hottest articles. The Indian Drug Manufacturers Association conferred prestigious Young Pharmaceutical Analyst Award 2011 for his outstanding research contribution in the field of pharmaceutical analysis and elected as Associate Fellow, AP, Academia of Sciences 2014. Best Research Scientist Award, NIPER 2016, Bharat Vikas Award 2017, Institute of Self-Reliance. Journal of Pharmaceutical and Biomedical Analysis, recognized by the JPBA Quality Award 2017 for the outstanding contributions in reviewing and contributions made to the quality of the journal. He is a reviewer for many international journals, including RSC Advances, Analytical Methods, New Journal of Chemistry, Journal of Pharmaceutical and Biomedical Analysis, Journal of Separation Science, Biomedical Chromatography, Scientific Reports, Chromatographia, Chemosphere, etc., and trained as GLP inspector. Good morning, sir. Over to Dr. Narendra Kumar Garu to present on topic Emerging Analytical Techniques and Trends in Achiral and Chiral Impurity Profiling in Pharmaceutical Industry and Research. Thank you, sir.
Yeah. Thank you very much for a nice introduction. And good afternoon.
Thank you, sir.
Yeah. So just I will give a brief introduction about some of the emerging analytical techniques and trends, especially in pharmaceutical industry as well as research. So both achiral and chiral impurity profile. So to give a brief introduction about impurity profiling, quality, safety, and efficacy, these are the three important terms we used to hear from the pharmaceutical industry when you are talking about the drugs or pharmaceuticals. Safety and efficacy of pharmaceuticals are two different issues of importance in drug therapy, and the safety of the drug is determined by its pharmacological and toxicological profile, as well as adverse effects caused by the impurities. So the impurity drug often possesses unwanted pharmacological and toxicological effects by which any benefit from their administration may be outweighed. So it is quite obvious that products which are intended for human consumption must be characterized as completely as possible. So that's the reason we are getting good number of hyphenated technologies to characterize the impurities in trace and outer trace quantities. So quality and safety of drug is generally assured because of these impurities controlling parameters. So that the reason analytical activities related to the monitoring and controlling of impurities is one of the important activities in drug industry for day-to-day monitoring of impurities in trace and outer trace concentrations.
There seems some problem.
Showing the screen, I think, sir. Sir, are you trying to share the screen, sir? Is there any
Yeah, it was in between disconnected, it seems.
Yeah, correct.
So I think now you are able to see my screen.
Yes.
Yeah. So the analytical activities related to the monitoring and controlling of impurities important in modern pharmaceutical analysis. That's the reason we'll try to see some of the important technologies, how these are emerging in the pharmaceutical industry to find the trace quantities of impurities, not only general achiral impurities and chiral impurities, what are the technologies we can commonly use. So quality guidelines says about the importance about the stability, analytical validation, impurities, pharmacopoeia methods, good manufacturing practices, quality risk management, analytical procedure development. There are different number of guidelines are developed by the ICH, International Conference for Harmonization for technical requirement of registration of pharmaceuticals for human use. So stability is talking about Q1 guidelines and Q1A to Q1F, like analytical validation, Q2, like what is accuracy, what is precision, what is linearity, what is limit of detection, limit of quantification. All these parameters are there in analytical validation Q2, and impurities, like what are the organic impurities, are there guidelines, inorganic impurities, residual solvents, all this comes under category of Q3, Q3A, Q3B, and Q3C, and as well as Q3D is introduced recently for the elemental impurities determination. Trace quantities of residuals, elemental impurities means inorganic impurities we can monitor by using different technologies like ICP-MS, intercoupled coupled plasma mass spectrometry. From the last 2 years, this made it mandatory in industry to use it for the monitoring of trace level inorganic impurities. Like this, good number of guidelines are coming based on the safety as well as efficacy as well as quality requirements. So if you go to the website of ICH and if you're able to see the different threshold limits are prescribed. Say, for example, your dosage is just 2 grams per day or less than 2 grams per day. Reporting value is just 0.05%. So you need to report if it is present 0.05%. Need to identify this like impurity, it's if it is a 0.1% or 1 mg per day intake. So our qualification threshold, you can qualify the impurity also once it is non-toxic, but the maximum permissible limit will be 0.15% or 1 mg per day, whichever is lower. So this percentage totally depends upon the doses, how much dose we are going to expose per day. So these guidelines are further, you can see here, what is the minimum quantity of minimum daily doses and their threshold limits, all prescribed in the ICH guidelines very well. So coming to the pharmacopoeia and regulatory aspects, the international conference, yes, it is giving these guidelines, which was initiated in 1995 in Japan. Of course, nowadays, like throughout the world, either USA, European countries, as well as India, and all countries are respecting the ICH guidelines. And analytical procedures should be able to separate all the impurities from each other. For example, HPLC method was developed. Once you develop the HPLC method with the drug and you are able to separate not only drug, all the impurities also. Not only just bulk drug and trying to see the impurities, even the final dosage forms also need to be see the actual active ingredient as well as minor impurities also. Once you develop method and transfer to the quality control lab, need to ensure that this method is really fit for the requirement. Like it should be accurate, it should be precise, it should be specificity should be passed, and limit of detection, limit of quantification, linearity, range, all these parameters need to be identified. So, and you can say what are the ranges permittable for each methodology for accuracy, what is the near to the true value. You need to take a reference standard material and compare with that. Is it 99.99%? So precision, if you repeat the results like 5 times or 6 times, what is the relative standard deviation? It is less than 2% or 1%. Like that, we need to specificity, like you need to spike the impurities which are possible potential impurities and ensure that these impurities are not interfering with your active ingredient. Like this limit of detection, minor quantity which can be able to see minor quantity, that is limit of detection, and limit of quantification which can able to visible to us with the required precision and accuracies, limit of quantification. Like this, all these parameters must be well studied before transferring method from one lab to another lab. So validation of analytical procedure, that it is proof of its suitability for the intended purpose. It is important part of the registration application for a new drug. Once you want to register, unless there is no validation, it cannot be acceptable. So for example, additional peak tiling or peak resolution analytical recoveries are important in case of chromatographic methods, either you call it as HPLC or GC or SFC. So ICH guidelines serves as a basis for worldwide for both regulatory authorities as well as for industry to bring the importance of proper validation to authenticate that the DMF files which were going to be submitted. So the analytical R&D departments, analytical research and development units in the pharmaceutical industry are responsible for the preparation and validation of this test methodologies. So how these are coming, we are talking about impurities, how these are coming or originated from the different sources we can see here. So generally, you may be having 5 steps or 10 steps in your synthetic process of a drug. The last intermediate of the synthesis is generally the potential impurity, we can say, that can be carried forward to the drug substance, finally to the drug product. As well as product of incomplete reactions, the reactions are not 100% completed. Minor quantity of the starting material is left over. That is the incomplete reaction that is also carried forward. Product of overreactions, you maybe use some larger quantity of temperatures or the conditions what you used may give some overreactions that also lead to the another unwanted byproducts. So even the impurities which are associated with the starting material, even minor quantity, that is also will be forward towards this new drug substance which was prepared. If you are using the starting material itself having some impurity, that will be carried forward to the final drug substance. So even during the synthetic process, we need to use a good number of solvents to have good reaction mixture to complete the product. So in that case, the solvents, small quantity leftover solvents will be there. That must be controlled. What is the quantity left over? Is it like, for example, benzene, we cannot allow it is like 1 ppm or something like that. So the trace leftover quantity residual solvent analysis is also important, like GC, headspace GC, and GC-NMS technologies are very much suitable for the monitoring and controlling of these residual solvents. Once you know the monitoring, then you can able to control which step is giving this source. Then you can automatically modify that and able to get the good specifications. So impurities coming from the catalyst. Catalyst is important to complete your reaction. So like nickel, cadmium, palladium, all these metallic catalysts or reagents, what you're going to use during the synthetic process, minor quantity will be left over. So that quantity need to be monitored. So the ICH-Q3D, which is recently introduced, is talking about inorganic impurity profiling. The technologies like ICP-MS, inductive coupled plasma mass spectrometry, is one of the best choices to monitor trace quantities of these leftover metallic elements. So parts per billion, even parts per trillion analysis is possible with this hyphenated technology. You can able to find the trace quantity of leftover the elements. So we know that palladium or platinum, especially like lead, mercury, arsenic, and cadmium, these are the toxic elements considered. So once you are accumulating with the drug minor quantities, it is slowly accumulated into the like body fluids as well as into the kidneys and damage the system. Even minor quantities, if it is dose is taken continuously months together, definitely there will be accumulation of the metallic impurities in your body fluids. So that's the reason we need to monitor that also. Product of side reaction, of course, if a drug is exposed to the different atmospheric conditions, definitely it will be degraded into the different forms. So that degradation products need to be monitored. Stability studies, what we are going to do is to ensure that what are the degradation products possible, and we need to monitor and characterize that degradation products also along with the active ingredient to ensure that if that is present, how much that was present. So to monitor that, we need to use the degradation studies also. So enantiomeric impurity is one more important concept. We know that yesterday, I think there is a class about the like residuals of these enantiomeric drugs. So even a minor quantity of like one of the enantiomer, which is toxic, one may be therapeutically active. So in that case, like if it is non-toxic, it's fine. Small minor quantity you can allow. If it is toxic, even minor quantities, maybe 0.1 or 0.05% is also not allowed. So enantiomers or mirror images, non-impossible. So need to be, it's not so easy to monitor those enantiomeric impurities. What are the technologies available? We'll try to talk about that also, try to cover that also in this presentation to see what are the chiral chromatography, how we can use that monitoring these chiral impurities. Residual solvents, yes, mentioned that the leftover minor quantity of solvents like class A, class B, class C. Based on toxicity studies, these solvents are classified as class 1, class B, and class. Say, for example, benzene, highly toxic, class 1. Like that, the classification is carried out based on the toxicity profile of each solvent, which are commonly used with the drug preparation process. So impurities which are associated with the excipients, bulk drug is taken from that you are adding some excipient to make it a bulk final formulation. So in that case, So the excipients what you are going to use may also try to interact with your drug substance. Sometimes if it is metals are present in the excipients, that will interact with your drug substance, and drug substance may degrade also because of the metals present in the excipients. So like that, so we need to monitor all the possibilities to ensure that the drug is pure enough without having any degradation products, any contamination during the synthetic process of the drug substance as well as drug product. So I'll try to give some brief introduction about the chiral chromatography. Anisomeric impurities need to be monitored. That we know that it is not so easy like achiral impurities. So try to understand about this with a brief introduction about the chromatography and going to the chiral chromatography. What is the difference between these two? Try to understand from that. So chromatography is a physical process. We know that there is no chemistry actually change. You're injecting your sample and trying to interact this sample with the stationary phase and as well as mobile phase. After some time, it is get eluted out. So there are two important components in any chromatograph system. One is the stationary phase, another is mobile phase. Static one is nothing but stationary phase, which is generally considered saying as a column. So next one is mobile phase. Moving phase is nothing but mobile phase. So in gas chromatography, gas is the mobile phase. In HPLC, liquid is the high performance liquid chromatography, liquid is the mobile phase. If you are using supercritical fluid as a mobile phase, that is nothing but supercritical fluid chromatography. So in that way, for example, stationary phase, for example, silica you're using, you're derivatizing with the different bonding phases. So bonded phases, if your reactive groups you are introducing, like phenyl group or diol group or cyano group, that is like a bonded phase. So you can introduce some chiral selectors also as a bonded phases, then it is considered as chiral chromatography. The stationary phase is only different. The remaining system will be same. The HPLC you can use it as well as SFC you can use it to make it suitable for the chiral separations of drugs and pharmaceuticals. So just to go back to the basics about the HPLC, there is a solvent reservoir and of course degasser to remove the dissolved gases which are accumulated in the mobile phase, and the pumping will flush your mobile phase like required flow rate 1 ml or 2 ml per minute. And of course, you have the sample injection system. So you can inject your sample, say 10 microliters or 20 microliter, make it dissolve in the mobile phase. The mobile phase, which is going with a constant flow of 1 ml per minute, for example, which was said by you, will go to the column and there is interactions takes place in the column and separation takes place. After that, it is going to the detector gives signal. That is nothing but chromatogram. It is happening. That is the output is nothing but chromatogram. So the outlet of the detector is you can send if it is analytical HPLC waste to the waste tin, you can send it. Otherwise, if it is a preparatory HPLC, you are injecting larger quantities. You want to see or characterize and identify the fractions which were collected here or impurities which were you are collecting or one of the enantiomer you want to collect it. Then fraction collector will be there, and that is known as preparatory HPLC. Like that, these are different components will be there. You can see here different connecting tubes between the pump and injection port, and injection port to the, again, like from the pump to the detector. So all these lines will be there. So these lines must be ensured with the clean between the analysis. You need to flush your like a system with the suitable solvent and ensure that all the previous samples are washed out, or previously used buffers are washed out, all the previously used additives are washed out, and then start your analysis. So these are the things we need to understand. For example, normal phase to reverse phase, you want to convert, you have to see the suitable solvent to wash in between and go to the next stage. So this is the basic HPLC instrument, easy to understand, modulated one. And so from this easily we can see here. So there is like top side, there is like bottles, reservoirs, we can say, mobile phase reservoirs. You can keep one as for dedicated water, one for the methanol, one for the instrumental, like that you can dedicate. And there are two pumps you can see here. And of course, open the valves and flush the mobile phase to ensure that all the previous dissolved gases as well as gases which are accumulated in the lines will be totally washed out. Now it is totally filled with the mobile phase what we are going to use it. So this is the rheodynode injector. This is a manual injector. If you want to use syringe, you can use it. And of course, from that, you can able to inject 20 microliters or 10 microliters based on syringe. Then the next one box is nothing but a auto sampler. You can keep the samples like say 90, 100 samples, and so you can give simply command vial number 10, vial number 20, or vial number 30, like that automatically that will be taken out. by the injector, auto injector, and able to inject the required volume, say 10 microliters or 20 milliliters, and get the chromatograph. You can see here the monitor, there is a 4 chromatograms simultaneously. That is PDA detector, photodiode array detector, able to show a different wavelength monitoring in a single time. That is the beauty of the photodiode array detector. If you are doing the method development, definitely photodiode array detector will be helpful for the method development. You are looking for sensitivity analysis already. Your method is ready. In that case, you can use the HPLC UV detector too. So like that, this is the basic components present in the HPLC, and so these are the columns. One is top one is analytical column, and bottom one will be preparative column. The larger volume you can inject in case of preparative. The loadability will be there will be the preparative column. So that the reason you can purify the compounds by using preparative HPLC. Of course, like HPLC will be something different. The larger quantities you should handle. That the reason the hardware component will be a bigger in size. That is the difference between the analytical and preparative HPLC. That the mechanism of separation and operation all will be same, except the volume of injection will be different. So that the reason the hardware components are bigger in size in case of preparative HPLC. So this is the chromatogram. X-axis is like a time, and y-axis you can see detector signal. Milliabsorbance units will be the units for the y-axis. So this is like manual injector. Generally in colleges, try to use this manual injector to inject your sample. Of course, you ensure that there should not be any air bubble in between, so that you will get the good chromatograph. So chromatography categories you can see here: analytical, if it is analytical HPLC or analytical chromatography, compound identification and concentration determination is the major aim there. Means you are injecting your standard first, followed by the sample, and what the area given by the standard for the specific concentration. From that, you are able to calculate the concentration of the test sample which was injected. That is the analytical HPLC. Quantitative or qualitative identification can be carried out by using analytical HPLC. But coming to the preparative one, either semi-preparative or preparative, in that case, you are injecting more quantity for purification purpose. You know all the compound already, so you want to purify one of the compound. In that case, you need to inject more quantity and isolate that compound. So that process is followed in case of semi-preparative or preparative. In case of semi-preparative, up to gram level, you can purify it. In case of preparative, you can go up to kgs. If you want to do the purification at the manufacturing site, yes, sometimes you need to purify it. It is not possible during synthetic process to purify all the compounds. During that time, chromatography is the choice for you. Then industrial scale, metric tons, kilograms, if you want to purify, yes. SMD, simulated moving bed chromatography. I'll use some brief introduction about that also. So process scale separation is possible with that. So if you go inside the column and see what is the size of the particle size. Particle size is important to say. It can be used for the preparative, or it can be used for analytical, or it can be for the UPLC or HPLC separation. That depends upon the particle size what you're going to use. Column chemistry may be the same, but what is the particle size inside is important to ensure that where it will fit. So for example, 20 micrometer particle size is there, then you can use it for the preparative HPLC. If it is 10 micrometers, yes, in case of like chiral chromatography, you can use it analytical, but in chiral chromatography, generally it is for only semi-preparative or preparative only, not for the analytical purpose. So 5 micrometers to below to that is very common to use in the analytical purpose. So 5 micrometers, 3 micrometers, sub 2 micron particle size columns are also there. For example, we are saying the UHPLC or UPLC technology, where you are using the columns which are less than 2 micrometer particle size, generally 1.7 or 1.8 micrometer. Based on the manufacturers, they will give more less than 2 micrometer particle size columns. So where your system will be, like UPLC system will be having that withstanding capacity of high back pressure. That is the beauty of designing, and so that's the reason you are getting better separation in a shorter period of time with good intensity peaks. That is the ultra high performance liquid chromatography. So if you are using less particle size columns, always you get the high intensity peaks and lesser runtime with good peak to peak separation resolution, which is possible with the small particle size columns. That is the benefit. So what the mechanism of separation will, based on the chemistry, what you're going to use as a stationary phase or column, that depends upon the mechanism. I'll give outlines about how mechanism is happening. So based on the interaction, for example, one of the analyte is interacting more with the stationary phase, that the reason it is staying with the stationary phase. Another is not interacting with the stationary phase, that is eluting first. So in that way, the weaker interaction, stronger interaction plays key. What are the chemistries behind that is the different story. But in general, we can say, see, the compound which is eluting fast is not having that much interaction with the stationary phase and able to dissolve very happily with the mobile phase. That the reason it is eluting as first. Next one is coming as a second peak or third peak based on their interactions. So we'll try to see here, for example, if you cross section, take it and say, for example, this is C8 column, and in between these TMS, trimethylsaline was used as a end capping material, which are leftover silica material, which is base material here to just end capping. You are not allowing these residual selenols to interact with the analytes. If that is interacting, you will get with the different tilings. To avoid the tiling factor more, you need to see that column is end capped or not. It is packed with the end capping process done, means leftover selenol groups which are not reacted with the actual reagent to get the specific column, that leftover groups should be packed with the suitable reagent known as trimethylsaline or dimethylsalines are very commonly used to pack this leftover silica groups. So in that way, of course, you see this mobile phase is moving and stationary phase is trying to interact with the molecule. Of course, this is having the hydrophobic chain, and this is also hydrophobic chain, C8 material. So that's the reason you can get the hydrophobic interaction is possible. In addition to that, it is also dissolved with the mobile phase. Say ethanol, it seems like another one is water. It is also dissolved inside. So the distribution is there between the stationary phase as well as mobile phase. That is possible with the HPLC. So that's the reason selectivity will be there. But in case of GC, there is no mobile phase is liquid. Mobile phase is there in the gas counter is a gas. Gas is generally inert in nature, so that the reason there is no interaction with the gas. So only stationary phase plays key role to interact with the analyte. That's the reason we can see here the length of the column will be like 25 centimeters, not 25 centimeters. In case of HPLC, 25 centimeters is maximum, but in case of GC, 25 meters is the minimum requirement. 25 meters or 30 meters length columns will be there to get the required selectivity. Of course, GC is suitable for the only volatile samples to analyze. That is the prerequisite. So effect of stationary phases, we can see, for example, C18 is very commonly used column in many HPLC labs. Of course, next one is C8 and next one is C4. So once the hydrophobic chain length is decreasing, the interaction with the nonpolar compound is also decreasing. That's the reason retention time is coming down or elution will be fast. So if you want to elute your compound very fast, use C4. Or larger molecules like peptides you want to analyze, then you can use C4 instead of C18 or C8, which may take longer time. And of course, it may not be eluted sometimes also. So that's the reason. So the length of the chain, the hydrophobic chain is also important to have the good selectivity as well as good methodology. So for example, we take here the example of phenol and other substituted phenol, which is methyl and propyl group substituted. So you can guess that when there is a hydrophobic chains or substitutions are more, definitely the interaction with the hydrophobic C18 material will be more. That's the reason that is the eluted as a second peak compared to the phenol, which is a polar function, which is not having good interaction with the C18 column. That's the reason that is eluted first. So in that way, we can guess that is known as hydrophobic interaction theory, which is well known in case of method development reverse phase HPLC. People know it very well. So based on the chemistries, what we are going to use, different chemical interactions with the solutes are possible. So these are known as basic interactions, hydrophobic interactions. What we discussed is hydrophobic interaction, dipolar interactions, pi-pi interactions, hydrogen bonding, and acidic interactions. These are the different interactions are possible. It was reported and literature is available in counter of EA about different stationary phases which are having different interactions. So coming to the chiral stationary phases, coming to the chiral structures, once you are injecting a racemic mixture to the chiral stationary phase, one is interacting more with the stationary phase compared to another one. That's the reason eluting. One is fast, another is slow. What are the reason behind that? We'll try to understand. One is diastereomic complex will be formed between the ionomer as well as the stationary phase. That is the one reason generally we'll say. But if you go to the structure and see what are different interactions, we'll try to understand in coming slides about what are different interactions possible by looking at the structure of chiral structures. On the different chiral structures also I'll try to show it. So just go to the literature before going to that details. So which technique is very much used in case of chiral chromatography? So from 1999, you start searching. 40% is the occupancy for the HPLC. From slowly coming to 2012, 2012, the occupancy is more than 70% chiral HPLC. And you can find from 2010 onwards, the SFC is also occupying some percentage, around 7 to 9%. So HPLC followed by SFC are the techniques of choice for all the researchers or throughout the world to use HPLC, chiral HPLC, chiral SFC, or the suitable ones to analyze different chiral compounds. So what are chiral stationary phases then available? Are chiral columns types of chiral stationary phases available? And what are their loading capacity? Our aim is not only just develop the methodology. So sometimes impurities will be there. You need to isolate that and do offline characterization sometimes. That the reason loading capacity is also one of the parameters to consider. And of course, once the loading capacity is there, definitely the stability of that selectivity will be better. We can able to use it for a longer time. So in that point of view, we'll see here a loading capacity of different types of chiral stationary phases. So per kill type is a maximum of 50 milligram for a gram of chiral stationary phase. You can load it. Polysaccharide derivatives are up to 150 milligram. You can load for a gram of a chiral stationary phase. So that is the advantage of polysaccharide derivative stationary phase. Either amylose or cellulose majorly used. So those are the highly loading capability chiral stationary phases. So that's the reason we'll try to discuss about the polysaccharide stationary phases, which are very widely used. Again, you can go back to the literature and see why we want to go to the polysaccharide stationary phases to understand more. So from 1999 to the 2012, again, the review article says that 94% occupancy is there for the exclusive HPLC. We are saying SFC also we are saying, but in that, what are the columns they are using? They are using the polysaccharide based chiral columns. So those are suitable for analyzing and separating the enantiomeric compounds. So what are these polysaccharides? One is amylose, another is cellulose. The derivatives of amylose and cellulose are considered as chiral columns here. So we'll see how they will do it. So one is like 1,4-glycosidic linkages, alpha-glycosidic linkages. Another one is beta-glycosidic linkages. Based on that, the cavities between the glucose units are slightly different. Of course, you need to add a chiral selector. This is a just basic material which will carry your chiral selector. So that is nothing but polysaccharide chiral stationary phase. So next one, if you go and see, this is a chiral selector. So phenyl carbamate, phenyl group is there, NH and CO and O. So oxygen is there, nitrogen attached, hydrogen is there. Hydrogen is like a chance to give the hydrogen bonding because nitrogen is electronegative. Hydrogen is tendency to escape from the electronegativity and trying to interact with the other electronegativity atom which is present in the analyte. So that is a hydrogen bonding is formed. So otherwise, if it is like the analyte itself contain hydrogen, the oxygen which is present, the keto group present in this phenyl carbamate is trying to interact. That is also one more chance for the hydrogen bonding. So hydrogen bonds plays a key role here. In addition to that, phenyl group is there. This phenyl group having pi electrons. This pi-pi interaction, if the analyte is also having the aromatic ring, then their pi-pi interactions will be taking place. So dipole, so if there is a keto group, again, like negative and positive dipoles will be formed. This dipoles with the dipole formed in the analyte, dipole-dipole type of interactions. All these are different type of interactions possible. Of course, you can use some like substitution here on the phenyl group, either methyl group, chlorine group, so to change the selectivity. So to get better selectivity, you need to use better electron withdrawing and donating groups to have different selectivity for different compounds. You have a number of compounds, which compounds require donating and which compound withdrawing, we don't know. That's the reason good number of stationary phases made it available, and we can use them for the enantiomeric separation of the compounds. So initially we'll see that the amylose polysaccharide here, the alpha 1,4-glycosidic linkage will be there, axial and equatorial position of this alignment. That is nothing but alpha 1,4-glycosidic linkage. In this beta-glycosidic, the alignment is something different. So that the reason it is known as beta-glycosidic linkage. So this is amylose, and the second one is cellulose polysaccharide stationary phases. So the R group can be replaced with one of the chiral selector, phenyl carbamate. So here the example was like 3,4-dimethyl phenyl carbamate was used as a chiral selector. Here we can see all I concise as a single slide to show that what are different chiral selector made it available and to the market, and we can readily use them by looking at the names. Like for example, dimethyl means that is chiral pack IA. Say somebody says chiral pack IA means that consists of dimethyl, 3,4-dimethyl phenyl carbamate is nothing but chiral pack IA. So 3,5-dichlorinated phenyl carbamate is nothing but IE. So like that, the different chiral selectors are made it available for both. Here we can see here for this is amylose. The second one is for the cellulose. Cellulose also consists of because cellulose or amylose, which is a base material, that is also plays some important role to separate your compound in addition to the phenyl carbamate. So that the reason both materials plays important key role, and different chiral selectors have different selectivity for the different analytes. I'm not talking about method development that takes some more time. So especially the method development is slightly different compared to the chiral separations. So, but one thing I want to give here is the immobilized chiral stationary phases made it available in market. All this IA, IB, IC, like this, like I2, IK. So all these 10 columns are suitable for all the reverse phase as well as normal phase. We know that normal phase is normally used mobile phase like hexane with ethanol or isopropyl alcohol is the normal phase. Reverse phase is you are replacing the hexane with the water and you are adding buffers, and in addition to that you are using methanol or acetonitrile, that is reverse phase. Polar mode is you are exclusively using the polar organic solvent like methanol or acetonitrile and combination of all this organic solvent, that is nothing but polar organic mode. Helic mode is some percentage of water you are adding, and remaining is organic solvent, that is helic mode. And SFC is one more supercritical fluid chromatography. In place of hexane or in place of water in case of reverse phase HPLC, you are using a gas CO2, and remaining organic solvent you are using, that is known as SFC, supercritical fluid chromatography. What is that? We'll try to understand. That is slightly different than the HPLC, but it is and its own importance for the separation of either achiral or chiral compounds. So supercritical fluid chromatography is like we can see here, when a substance is brought about the critical temperature and pressure, coexisting of both the phases disappear and a new state called as supercritical fluid is formed. Now there is no gas or liquid, it is a fluid. Because of temperature and pressure, highly it was applied. So the physical properties of supercritical fluids are intermediate between those of typical gas as well as liquid. So it consists of the good viscosity as well as diffusivity. So which are good solubility. So liquids require liquids generally having good ability to dissolve the samples. Gases are having good dispersion. So those two properties will be there with this supercritical fluid. That is the beauty of SFC. So for example, you are taking gas CO2 here. Supercritical fluid is one of the best one is CO2 is one of the example. So first condense it. Once the distance between the gas molecule is coming down, that is condensation process. Then compress it further and then heat it, and it will become supercritical fluid. That supercritical fluid will be transferred along with the organic solvent like methanol or acetonitrile or isopropyl alcohol to go to the column, and where the interactions takes place with the analyte with the column, and then it is coming out from the column. Once it is coming out from the column, it will be removing temperature and high temperature, high pressure and all. It is going back as a CO2. In that way, recycling process is possible. So especially in case of preparative HPLC or preparative SFC separations, so we can use this supercritical fluid to get the purified fractions. So here we can see here how the preparative SFC works. So CO2 gas is there, and to that you are adding co-solvent, either methanol or acetonitrile or isopropyl alcohol, and remaining say you can add 5% to 40% of the co-solvent. Remaining say 60 to 95% you can keep as a CO2. That is the beauty of this technology. And then condensation, compression, heating it, and then send into the column. And once it is given the detector the signal, chromatogram is completed, then you are collecting back as a fractions. Fraction collector will be there, and the CO2 will be sending back to the again cylinder. That is a recycling happening. So what are the applicability, complementary selectivity? What HPLC which is not separated, you are getting it here separation because of the mobile phase composition is different. The selectivity definitely change, especially in case of chiral separation. The small difference is also important. Mobile phase composition. So then you get the good selectivity, faster separation, high productivity. The flow rate is here at least 3 to 4 times more than the HPLC because you're using gas, supercritical fluid. So that the reason you can use more pressure also to have the good chromatography in lesser runtime. So ground to multi-case separation is possible. It is a greener technology and easy evaporation of the fractions once, because it is carbon dioxide, again going back to it once it is exposed to the atmosphere, it is, you can collect back into the cylinder. Low cost of operation, all these are important here with this SFC. So the CO2 itself, like a acidic in nature, that the reason acidic K2 is generally not required to add. Required to use that TFA and try to get good big shapes. So anyway, neutral analytes also not require any A2s, and basic analytes like diethylamine, ethylamine, isopropylamine, butylamine, all these A2s you can use as less quantity as possible, like 0.1% or maximum 1% you can use it and able to get separations. So in that way, like recently published one of the article, antiretroviral drugs, I think more than 8 antiretroviral drugs are separated by using the SFC condition, and simultaneously also it was separated by using SFC to see how the SFC is acting for separation of this enantiomeric separation of all these antiretroviral drugs. So again, like not only achiral or chiral columns, all columns are having less particle size. So instead of 5 microns, if you're going 3 microns, then you can see the efficiency is enhanced. At least 40 to 60%. Once the particle size is reduced, then surface area increases. Once surface area increases, the interaction efficiency increases. Once the interaction efficiency increases, good resolution is coming in a lesser runtime. So that's the reason you are getting at least 40 to 60% efficiency when compared to the 5 micrometer particle size column if you have started using 3 microns. In achiral, 3 microns are very not common, 3.5 will be there. And of course, 5 microns is very standard size. In chiral, 3 microns are also made it available. So if you want to transfer method from 5 micron to 3 micron, yes, you can transfer it. And of course, generally the resolution will be enhanced, but not decreased, and sensitivity also enhanced. So for example, in this case, 5 micron chiral pack IA column is not 100% separated, for example, here in this case. Then if you're using instead of 5 micron, 3 microns, you started using, the resolutions still increased because even the mobile phase composition is same, but by changing the particle size, you are getting good separation. That is the importance of having the lesser particle size columns. So that's the reason UPLC is having the good resolution than the HPLC, we can say. So sub 2 micron, in case of chiral, we can see all these chiral pack IA columns and IB, all these columns you see here. In seconds, we can able to separate the compound like enantiomeric mixtures. So less than 1.2 minutes, 20 seconds, 30 seconds, 40 seconds. So once you stabilize the instrument, in that case, yes, you can definitely get the rapid separation in a shorter runtime. So in that way, cost reduction. So 20 minutes of runtime, you are using 2 minutes. Definitely, so especially definitely need to have the good separation in a shorter period of time. So next one is a counter current counter. This is also an important topic where
Some disturbance in your audio system, sir.
I think the screen has frozen, sirs.
Okay.
Some internet issue, I think, Vijaya.
Yes, madam.
He is coming in.
Hello.
Yes, sir. Now you're in.
Yeah. The slides are visible.
Yes. No, slides are not yet visible, sir. Sharing is gone.
I think it is just a few minutes back, or it is more than 5 minutes. So how much time it was? Like
few minutes, sir. I think 1.5 minutes to be precise. 1.5 minutes.
That's
if you
okay.
put the slides, we will tell you.
Yeah.
Let's see where it is. Transfer from 5 micrograms to microns to 3 microns. The slide.
You can ask me to stop anytime, not an issue. So I will save the time of Chandraya. Chandraya already, I think, consumed some of my time. So
no, sir, even you can consume some of the lunchtime.
Okay. Okay, I will try to complete that the earliest to about the. Yeah. Again, I think there is a poster presentation too. I think. Okay, so
Yes.
this like small particle sizes are also important. That was discussed. After that, we are going to the So countercurrent chromatography is also one of the important technique which is commonly used in case of industry, especially into the production level, larger quantities. So, for example, briefly to understand this, there are two animals. One is reptile, another one is rat. Both are kept on the center of this conveyor belt. So, both are moving forward direction from left to right, but conveyor belt is moving from the right to left. Definitely, the reptile, which is slow moving animal, definitely will come back, and of course, the rat will escape from the belt. So, in that way, we can say the feed is kept the center, and the extract is like reptile, and of course, raffinate is the one which is like a rat escape. So, the same principle was used for the simulated moving bed chromatography. We cannot move actually stationary phase. It is a simulation process. So that the reason it is known as simulated moving bed chromatography. So the serially we are connecting different columns. For example, here in this case, 8 columns. The inlet of the one of the column will be outlet of another column. Like that, serially one after another, all the columns are connected. So inlet of one will be going for the eluent, another will go to the feed, and outlet of one will be coming as extract, another will be coming as raffinate. So this raffinate again you can use it and get the extraction done. That is for larger quantity of purification process. Yes, this is required. So that is the where there also you can use these stationary gauges, especially parallel stationary gauges, to have the bulk quantity of separations. Of course, ordinary columns are also ordinary parallel columns are also you can use if it is manufactured in that size to purify the compounds. So actual how it looks in case of industry, you can see here at least 30 centimeters, 80 centimeters general width of these columns. You can keep how much length you want. So like that several centimeters length you can keep it and able to purify the compound, whether it is a chiral or chiral compound. So that way we can say the columns like analytical columns are nothing but generally 25 centimeters is the length, and the 5 micrometer particle size is very common standard size. Of course, for UPLC is less than 2 micrometer particle size or 3 micron particle size columns are in between, you can use it. And of course, that is for the discovery or general analytical R&D labs, you can use it. So, or once it is method is developed, you can transfer to the quality control lab. But if you want a quantity, some minor quantity for the like work, for example, say nimbolide or something, which was explained by the Chandraya, we are also part of that. So then the purification is done by the preparatory HPLC. Then we tested for the other activity studies. So in that way, the clinical studies can be carried out by using the preparatory SFC or preparatory HPLC. Then, of course, if it is still the synthetic chemistry is unable to synthesize a chiral compound purified 100%, or because of the cost matters, or because of the different number of steps to reduce, if some chiral impurities coming to the final mixture, then yes, production batch you can SMB, simulated moving bed chromatography, which was shown to you, that can be used to remove the chiral impurities. So like that, that is about chiral chromatography. I will give brief about the LCMS also, which is very commonly used technology nowadays. So you know that HPLC is working at high pressure condition, liquid, and of course mass spectrometry is working at high vacuum conditions. There is no even gas, total gas is also removed. So the coupling these two techniques is not so easy. So one is totally working in liquid in case of HPLC I'm talking about. This figure is showing a GCMS. So why I'm showing here GCMS is like you see the three different components present in the mass spectrometry. One is ionization source, the second one is analyzer, third one is detector. So all these three will be there in a vacuum chamber. So you have to keep total mass spectrometry in vacuum chamber. We are using EI, electron impact ionization. But if you are using HPLC and connected to the mass spectrometry, the liquid is also will be there. That is also passing to the mass spectrometry. That is not allowed. So to avoid that issue, instead of sending to the mass spectrometry, we are using the ionization chamber different. That is known as atmospheric pressure ionization, where sample will be ionized there in the atmospheric ionization chamber. Then it is sent to the analyzer directly and forward to the detector. In that way, the problem was solved. That is the history behind the LCMS technology. This is all explained with the like a graphical representation. Ionization at atmospheric pressure is the concept introduced to couple this LC with the mass spectrometry. So then the HPLC, which is high pressure, is able to connect with the mass spectrometer with high vacuum, and there is a chance to give the high penetrate technique known as LCMS. So that is about brief. Then coming to the this atmospheric pressure ionization, like there are three components in this, or three types are there in this. One is electrospray ionization, the second one is atmospheric pressure chemical ionization, the third one is atmospheric pressure photo ionization. So each one having its own limitation. For example, ESI, electrospray ionization, is suitable for the larger molecules like proteins, peptides, and able to get the better ionization with the electrospray ionization, that is for polar molecule. Next one is atmospheric pressure chemical ionization is for the nonpolar compounds. Atmospheric pressure photo ionization is for the highly nonpolar compounds. So in that way, each ionization methodology is specific for the specific analysis only. So we can see here the comparison. So ESI is occupying more high molecular weight compounds, but it is able to ionize the compound very polar to the polarity. ABCA is from the polar to the nonpolar, it is intermediate, and APPA is towards more nonpolar compounds to ionize. So molecule must be ionized first to reach the detector to get the detection. So that is prerequisite for mass spectrometry to ionize. A simple molecule without ionization will not be, without charge will not be detected by the mass spectrometry. So in that way, like analyzer is important, which mass to charge ratio will be sent first, followed by another one. So for that quadrupole, time of flight, ion trap, different analyzers will use different names to the mass spectrometry. So ion trap mass spectrometry is for the MS power n type of analysis. You are fragmenting further and able to get the information about one like impurity and get the structural elucidation. That is ion trap. Time of flight now it is replaced that time because high accurate mass measurements have to trace minor molecular masses. At that I will try to give more information. And a quadrupole is for quantitative analysis. So here time of flight mass spectrometry is so a uniform electromagnetic field is applied to all the ions at the same time and causing them to accelerate down to fly. Say, for example, there is a group of people asking them to run, and then you ask them to start it. So once the person who is having thin and lighter weight person definitely will move fast compared to the person who is having higher weight. 100 kgs person may be going late when compared to the 40 kgs person will reach a goal very fast. Because the people already taken the good quantity of food, good exercise, good training given to all the people, but all the people are not having same masses, all are having different weights. So in that case, definitely if the conditions are same, the less weight person definitely reached first compared to the middle weight person compared to the high weight person. In the same way, the time it reaches the ion to the detector, that determines what is its mass value, m by z value. So that is the time of flight mass spectrometry, which can able to give accurate mass measurements. So fourth decimal mass accuracy, you can get it by using time of flight mass spectrometry. So with that, we can able to do the impurities characterization. So how it looks, yes, I shown. So you can see here, this is known as soft ionization. In case of GCMS, that is hard ionization. In case of LCMS, known as soft ionization, the ionization methods are ES electrospinionization, atmospheric pressure, chemical ionization is very commonly used. So here in this case, a protonated molecular plus hydrogen, M plus H, deprotonated molecule, M minus H, or adducts like sodium adducts or solvents what you are used like acetonitrile methanol will form as adduct. So small loss of small molecules like the loss of water, loss of ammonia will be happening with the molecular ions in the soft ionization LCMS technique, which is not common in case of GCMS. So we'll see one example here. This molecule, which is having 184 molecular weight. So if it is done in the positive mode, 184 plus 1, 185 is the like you can able to find here. In case of negative mode, 184 minus 1, 183 is the peak you can able to get high intensity. So like that, you can able to distinguish and able to get the information with the LCMS, which is hyphenated technique. You can use the column like Carroll columns again, or HPLC columns again, or UPLC columns again, based on the front end HPLC, which is connected to the mass spectrometry. You can use in the LCMS. So like this, like different work was carried out with the HPLC, LCMS, of course some SFC also, based stability or impurity profiling. So these are some of the literatures you can go through and get the information about this. So this is the one I want to, I think, yeah, can I conclude here? So, yeah, so, madam.
Yes, sir. 10 minutes
Yeah.
you have, sir. 10 more minutes.
Oh, okay. I think then yeah, it is good to me to continue. Okay, so I think the concept of quality by design is also important going on in industry nowadays. So if you have a methodology which is flexible to transform from the R&D department to the quality control department for regular activities, that is known as quality by design. You are designing your methodology initially, and during your practice, there will not be any issues. So that is known as quality by design. I'm being just brief about that. And a systematic approach to the pharmaceutical development. Nowadays, we are applying for the analytical method development also, starting with the pre-defined objectives with the emphasis on product and process understanding controls is the definition given by the ICH Q8 guidelines about this concept of quality by design. You can use a design of experiment, multivariate analysis, Six Sigma approaches to follow this QBD methodology. So, which is started initially about 2002, and from January 13 onwards, it fully adopted in pharmaceutical industry. Not all industries, some of the industries. So, FDA CGMP is also saying about this, follow the like QBD, and there is no errors in that case. There is no need to go back and revalidate your method once you have the flexible conditions. What are the flexible conditions? That is known as design space. So how the design space can be carried out, those points we can understand by looking at this publication. Quality by design is systematic and rapid liquid chromatography mass spectrometry method. Eprocet and mesylate and its related substances carried out by using superficially porous particle column, which is like a UPLC separation HPLC. That is also novelty here in this case. So the objective of QBD is nothing but a rapid method, accurate method, and precise method, and robust method. That should be there. All the validations, what you are expecting, like highly accurate, highly precise, highly rapid in addition to all this. And the interaction of effect of all variables, pH effect, temperature effect, bubble phase composition effect, how these are happening simultaneously, and a space is designed, and with that design space, that values only if you keep automatically, there will not be any issues when you are operating. So the flow rate is 1.2. You are flexible. The regulatory agency will not be asked you. So because you developed a method such a way that it is not having impact in changes, like happening, even change from the minor quantity of flow rate or minor quantity of mobile phase composition or mobile phase like a gradient system or pH changes 3 to 3.5, like that. You are ensuring with the QBD approach and saying that design space is developed, so that the reason within that design space I can use my methodology. So variables like pH, temperatures, and gradient programs, and you make a design space. How you are doing it is a little bit, you need to take help of software, like a design expert is one of the software we used to view the some of the experimental requirements. Once you are doing that, and from that data, you can ensure that design space is developed. So the process can be selected here. Like I think there are 6 impurities along with the drug. There are 7 compounds. You need to separate it. So we are taking like 4 different columns: C18, C8, and the phenyl column, and of course fused core technology column also. And the design expert software is also taken. And here the terminology we need to understand is, one is ATP, analytical target profile, where accuracy is 1 point we are considering here. Faster separation less than 10 minutes. Resolution is more than 2. Critical quality attributes, like resolution is important to critical quality attribute, and what is retention time of last compound is also important. So that is critical quality attributes we are taking. These are the parameters generally we do it method development, but we are classifying here as per the QVD requirement. And risk parameters like columns and the pH of the mobile phase, organic modifier, flow rate, these are considered risk parameters, because if you change something in this parameter, definitely there will be change in the retention times or resolution of the compounds. So in that way, 2 phases, like screening method, another is optimization. In screening, different columns and different pHs and different organic solvents. In optimization, different temperature programs, gradient programs of flow rates, and develop the methodology. So here, like we can see left side, what are the design experts says based on our input. These are the 4 columns I am using. These are the 2 pHs, 3 and 5 I am using. Methanol and isoniazid also I am using. Then give the design expert. Ask the design expert to give the experiment to follow. Then it is giving like randomly 5.5 pH and isoniazid, and we're using phenyl column. So next one is like 3 pH by using methanol, using phenyl column again. So another one is 3 pH and isoniazid. So you are getting some phenyl again like some cell column like that. So by taking a different proposition of all these to ensure that all the parameters will be involved here. Then that 16 experiments which are suggested by the design expert here calculated. Just we run that 16 experiments. Out of that, which is showing the good resolution and lesser runtime is shortlisted. That is experiment number 13. So then optimization phase we need to go. Here in this optimization phase, temperature, gradient flow, as well as flow rate, all the 3 parameters. Based on that, again, 8 experiments were studied. So runtime of 7 minutes, runtime of 10 minutes. With flow rate of 1 ml, 1.5 ml. Column temperature of highest side is 50 degrees, lowest of 30 degrees. So the range was taken. So this optimization phase gives other 8 experiments. From that also, we finally achieved the design space. Means, see, 15% is a starting gradient. It is going to 65% within 7 minutes. If you are going with this process, not 7 minutes, within 10 minutes also fine. So, for example, if you are getting another process related to impurity, new impurity you are getting, or degradation product is coming, instead of 7 minutes runtime, you can keep up to 10 minutes, then there is no issue of resolution issue. All the peaks will get separated. So that flexibility itself you are getting initially during method development itself. Again, pH 3 and 3.3. In between, you can play. There is no issue. No regulatory agents will come and ask you why you are using 3.3 in one time, another one 3.2, another one is 3, because it is functioning good with 3 to 3.3. The flow rate of 1 ml, 1.3 ml. For example, resolution is down, like more peak shapes are broad. You want to increase the runtime, or you want to reduce runtime. You want to reduce the runtime and then reduce the unnecessary resolution or like peak shapes. You want to improve the peak shapes. In that case, yes, you have to use the flow rate more, 1.3. So 1 to 1.3 flexibility available. And the column temperature is like 30 degrees to 35 degrees. 5 degrees temperature difference is allowed, because it was from these 16 experiments on 8 experiments followed by 8 experiments, ensure a design space and graphs will be generated. That data shows it is flexible with this. So in addition to that, you have to verify at least 5 experiments in between this data and ensure that it is working fine. Once you are doing that and verification is done, then the regulatory agency will not say no to use the quality by design concept. So one of the chromatograph we can see here, all these 7 impurities along with the drug separated within 6 minutes. by using core shell technology, HPLC. By using HPLC, is it possible like 8 compounds or 7 compounds operation within less than 7 minutes in HPLC? No. The technology is core shell technology. In addition to that, the QBD concept is applied to get the good result. You can see here there is no peak-to-peak coelution or something. So I'm able to get all the compounds separated. That is the beauty of QBD. So with this, these are some of the trends and going on and techniques using by HPLC. Thank you very much for your time, and thank you very much for like Krishna Reddy College as well as RSC for the opportunity given to me to share some of my work carried out both in industry as well as in the academics. Thanks a lot.
Thank you very much, sir. I'm sure they have some doubts.
Yeah, really. Near to lunch break, even though there are some doubts are there. Good.
Yes, yes, sir.
Yeah, people are listening. I think, yeah.
Hello, sir. I have a doubt.
Yes,
Tanuri, sir.
Yeah, may I know who is on the line?
This is Dr. Vijaya, Principal, Tigala Krishna Reddy College of Pharmacy.
Madam, Chapandi Vijay garu.
Sir, what is the difference between super fluid chromatography and supercritical fluid extractor? Is it the same?
Yeah. Extractor is for only extraction purpose. So for chromatography is for the purpose of separations. And
what is the difference, sir?
Yeah, that extractor, for example, you are using, say, for example, CO2 with high temperature and pressure, that is extractor. The instrument manufacturer may knowing what is the components present in that, but chromatography is not that. Chromatography should have at least 3 components. One is column, one is detector, another one is mobile phase. All the 3 components must be there. That is the instrument, HPLC, same instrument. You can use it with the additional like a pump, which is flushing the CO2, and a cylinder also needed. That is CO2 cylinder, and this is like known as SFC. So SFC instrument is manufactured by the HPLC manufacturers like Shimadzu is manufacturing, Waters manufacturing. So these people will supply the SFC technique. So which is the additional hardware component present in the HPLC to have the good separation by using the supercritical fluid as part of your mobile phase.
Okay. Actually, this SCFE technique is commonly used nowadays, sir, for separation of some of the.
Commonly means it is emerging. I can say it is because the cost of this equipment is definitely more than the HPLC because you need to have additional features. And for example, if you are going for some of the chiral separations which are not separated in the ordinary modes like normal phase, reverse phase modes, then SFC modes definitely gives good separations. Especially if you want to go for purification purpose, larger quantities if you want to purify. So in that case, so wasting the solvents like large quantity of organic solvent is not permittable. So, and greener technology if you want to use, yes, extraction by using SFC is the best alternative, especially like the active ingredients from the plant materials. So pharmacophores, you want to isolate, yes, then SFC if you are using even the residual solvents, contaminants also you can reduce. Okay. So I am talking about residual solvent analysis that anyway you are using only CO2 that is coming back, and that is 90%, remaining 10% will be like methanol or acetylene, you can use it, that can be easily evaporated. So that's the reason. So the safer like isolations, purifications possible with high yield is possible with the SFC compared to the ordinary HPLC separations. HPLC is very good in case of analytical, but preparative, yeah, normal phase is considered as first choice, then the reverse phase, but SFC is considered first than even the other modes of operations to have purified fractions with less consumption of solvents and we can say rapid analysis that the greener technology we get the benefit out of all this.
Okay, sir. Thank you so much. You have very beautifully explained various analytical techniques like counter current chromatography, this hyphenated technique like MS, LCMS.
Yeah.
Yes, sir.
Yeah.
You have touched QBD also, sir.
Yeah.
Thank you so
Yes.
much for your time. Thanks a lot. Very informative session, sir.
Thank you very much. Thank you, sir.
Wonderful session. we had.
Thank you,
Thank you
Taluri
so
garu.
much.
Namaskaram, sir.
Namaskaram, thank you. You maintain the time also.
Yes,
Yeah,
sir.
We appreciate that. We appreciate that.
Part of RSC I should maintain.
No, because
Otherwise you will punish me.
no. Please don't say that. We are happy. You have accepted our invitation to address the students. I'm sure the students will benefit it.
Yeah.
And the connection, the topic I had yesterday, and the importance of chiral columns. There is a lot of connection in there.
Yes.
Without chiral columns, it's very difficult. Some of the compounds to separate them.
Yes. Yeah, both are like mirror images, same structure. Yes, only spatial difference is there.
Yeah.
It's not so easy to get the selectivity with ordinary columns.
Yeah.
Like HPLC and chiral HPLC. Which one is more costlier, sir?
No, there is no difference between HPLC and chiral HPLC actually. The same HPLC
The
you can
only
use
thing
for
is
the columns you have to use
columns,
chiral columns.
yes.
Yeah, that is the only difference. But we generally chiral
like
HPLC
normal columns and
Only-
chiral columns. What is the cost
chiral
difference?
column, yes, cost will be definitely more. For example, if it is like say 40,000 or 50,000 in case of a chiral columns, like chiral columns will be 1 lakh plus.
Double.
Yeah, minimum double, minimum
Okay.
double. Of course, there will be like discount price for the academics definitely.
Okay.
Yeah.
Yes, sir. What is the difference between analytical column and preparative column, sir?
Yeah, the preparative column, you can able to load more quantity. Means for example, analytical HPLC, for example, we are injecting 20 microliters or 10 microliters is standard injection volume. In preparative, you can start with 100 microliters and go with the in 5 ml at 20 ml also you can inject based on the column capability. So the purpose of preparative is there for the purification purpose, not for just quantitative analysis. You want to get the fraction, like how you are using regular silica gel column in the organic chemistry labs or material chemistry labs. So the same job can be done with the preparative HPLC. Instead of offline TLC, you can get the online detector like UV detector and able to separate the fractions required. So minor quantity of fractions you can isolate and able to go for characterization if it is unknown compound. That is the purpose of preparative HPLC in general.
So flash column chromatography, they use
Yes, flash also a similar principle, but flash columns are they're for offline purpose, not for hyphenation like HPLC purpose. Instrumentation will not be there that much rugged flow rates and all in case of flash. So, but the principle will be behind same. So, based on the particle size of the column chemistry as well as the pumps what you are going to use, the classification we can say generally the flash column like HPLC, SFC, or as well as UPLC. For example, HPLC and UPLC difference is the particle size of the column is definitely less. Yeah, 5 micrometer is for HPLC, less than 2 micrometer is for UPLC. But the hardware is suitable for that. The same pump you can use it, but the pump capacity flow rate is something different than you cannot use 5 ml or 10 ml per minute in case of UPLC. 0.2 ml, 0.5 ml, 1 ml is maximum in general in case of UPLC. But HPLC 1 ml is starting and go up to 2 ml is very common in case of even analytical HPLC. For preparative HPLC, you can use 10 ml per minute is the minimum starting for the preparative purpose. In that case, the pump size are different. So, some of the manufacturers are giving the flexible semi-preparative instruments where you can use both for the HPLC and the semi-preparative purpose, not the preparative purpose. Minor quantity you can isolate and do the research work. But industry required preparative and SMBs, but based on the quantities of sufficient.
Dr. Talluri, even though it's not the platform, you can mention that the 18th program, the DICEL program, because this is a right platform to students to. I think you better inform them if somebody is interested.
Yes. Actually, thank you very much, sir, for making me remind that program. We are going to conduct one program with again with RSC, Register of Chemistry Deccan section, on skill development aspects, exclusively for the HPLC this time. So this is 7 days course. 4 days will be totally workshop, theory classes. Most of the sections will be taken by me, and of course, some of the eminent faculties from the industry as well as academics. Professor Pisapatigar will be also there, one of the lectures. So, and followed by the hands-on section. So 4 days theoretical course means online classes. After maybe COVID issue completed, or depending upon batch size, 4 people will be called each batch for doing the practical section. That is for 3 days, Thursday, Friday, Saturday, for example. Like that, maybe monthly 2-3 batches will be run. So who is attending these theoretical classes will be eligible to get the practical sections once they are active and once they are interested. We'll be having some quiz and evaluation section. Based on that, we'll select the people for the practical section also. Of course, theory courses will be open to all the people mostly. Of course, we are last date we are keeping 12th of January is the last date for that. So encourage all of your students to attend the this program. So you will get it is free. There is no registration fee for that. That is one more, I think. So
yeah, sir.
Yeah, one more added value for the students. Even I think when we were students, I think we are not able to get this type of chances. So now with the encouragement of the industry as well as from the digital industry as well as RSC, we are able to give the free training for all the 7 days. So 4 days will be online, 3 days will be offline. So that offline will be declared later, but 4 days online will be starting from the 18th of this month. The last date for the registration is 12th of this month. So
any limit for the registration, sir, from any college? Limit of students, number of students from college?
Yes.
Yeah, we want to limit because it is open, depending upon application also. Again, we will sit with all the committee members and able to shortlist. If the number of applications are more, and I think definitely we will restrict the number. In that case, we'll ask you the merit candidates or something, like you have to screen some of the candidates. But the first section will be open to all. First day section will be open to all, which is again like a chiral chromatography will be there with more detail. Of course, different analytical techniques which are commonly used in the industry. One of the eminent faculty from the industry is coming, is going to talk about that. So those sections will be open to all the people, irrespective of they are selected or not. But the other 4 days will be at least 50 to 100 members we want to shortlist for the online training, and only 20 members will be finally given chance to work with the offline hands-on, because we cannot accommodate 100 members in industry, right? So there are so many restrictions. Yeah. Thanks a lot for the opportunity again. Sir, thank you very much. If you want to add something, you can add, because you're also you are the person behind this program also.
No. Even those who have finished their pharmacy degree, they are also eligible. Not only those who are studying, but those who are looking for a job.
Yes.
Okay.
Once they have skill development.
Yes.
And once they have skill development, this is a great opportunity for them also.
Okay.
The motto of this course is to make the people suitable for the job opportunities. Yeah, that's the reason. This is a skill development program we initiated. Thanks. Thank you very much.
Thank you, sir.
Yeah.
Thank you very much, sir. Thank you for sparing your time.
Thank you. Thank you so much.
Thank you.
Thank you so much, sir. The session was very informative and spending valuable time with us and providing the students also to get opportunities. Okay, thank you so much, sir. And now the poster session will start by 1:30, madam.
Yeah.
And
We are starting early.
yeah, as we have very less time, so many poster presentations are pending.
Okay.
So we'll join again by 1:30, madam.
1:30. So 15 minutes break for us.
Yeah.
Yes.
Okay,
Yes,
no
ma'am.
problem. Madam. No problem.
Sorry for inconvenience.
No, it's not at all an inconvenience.
So all the participants are requested to
All the
reassemble.
participants be ready by 1:30 for poster presentations. Thank you all.
Okay, ma, thank you.
Thank you.
ఒక బుక్స్ అని అలా పెట్టడానికి నాన్న.
అట్లా ఇప్పుడు
వీడియో ఆన్ చేస్తున్నా. ఎవాల్యుయేట్ చేసే
Good afternoon, ma'am.
Speaker, please.
Healthcare professional.
Good afternoon, everyone. Myself, M. Vijayalakshmi, Associate Professor, T.L. Krishna Reddy College of Pharmacy. And welcome to e-poster presentations of Day 2, ACPS 2022. So, a brief instructions for presenters of e-posters. So, presentation is limited for time for presentation is maximum of 5 minutes, ma'am. Okay, and please see that your speech is audible clearly, and any queries raised that must be posted in chat box that can be clarified by the presenters. Okay, so all the best for all, and shall we start the presentations? Yes.
Yes, ma'am.
Yeah.
Yes, ma'am.
So J Nikita. Postal code PGEN 604.
Yes, ma'am.
Yeah, you can share the screen to present the e-poster.
Yeah, okay, ma'am. Ma'am, am I audible, ma'am?
Yeah, it's clear, but your poster is not seen clear, ma'am.
Okay, ma'am. Ma'am, is it clear now, ma'am?
No, ma'am, you just rotate your mobile.
Yeah, yes, ma'am.
Are you presenting through the mobile?
Yes, ma'am.
You just rotate the mobile.
Is it clear now, ma'am?
A bit. Okay, you can start your presentation.
Yeah. Good afternoon, everyone. I'm Nikita from third year B Pharmacy from T.L. Krishna Reddy College of Pharmacy. I'm here to present a poster on the role of vitamin E as an anti-aging compound. Vitamin E is an fat-soluble antioxidant and it has been in use for more than 50 years in dermatology. Generally, vitamin E is of white types and it includes both tocopherols and tocotrienols. Vitamin E is found in variety of foods like almonds, avocados, broccoli, and wheat germ oil and sunflower oil, and many more. And we can also take supplements for vitamin E. And there are so many dosage forms are available in the market, like vitamin E capsules and tablets. And vitamin E is also used in many cosmetic products like moisturizing creams, oils, and in anti-aging creams. Generally, vitamin E is popularly known for its antioxidant property, and it is also considered as an anti-aging factor. Aging is the process which is characterized by the wrinkles and loosening of the skin. The main cause of this aging is exposure of skin to the UV rays. This UV rays damages the proteins of the skin and also damages the connective tissue of the skin, which leads to the aging of the skin. So this vitamin E helps in the biosynthesis of collagen fibers in the skin, which is a connective tissue. By this, we can overcome the aging process of the skin. Generally, aging is considered as a oxidative reaction. Vitamin E, being as an antioxidant, acts against this oxidative reaction and prevents the aging process. And vitamin E is also known as free radical scavengers. Free radicals are the particles which are responsible for the breaking down of our skin collagen, which results in the wrinkles and spots on our skin. Vitamin E is a free radical scavenger. It has ability to scavenge the radicals, which causes the aging of skin, and it breaks the chain propagation. Not only in the aging process, it also helps in the many more other related issues like skin related issues vitamin E prevents the skin cancer, and it also enhances the immune system. It also helps in the treatment of skin diseases like psoriasis, cutaneous ulcers, wound healing, sunburns, and melasma by different types of mechanism of actions. And the usage of this vitamin E has very less side effects, and there are no contraindications also. So I would like to conclude this by saying vitamin E have been found to play a vital role in anti-aging process and in other skin related problems also. Thank you, ma'am.
What is the chemical name of vitamin E, Amma?
Tocopherol, ma'am.
How many tocopherols are available?
4 tocopherols are available, ma'am. Alpha, beta, gamma, and sigma.
What is the use of sigma? The epsilon. What is the use of this vitamin E? Use.
It is an antioxidant, ma'am. It helps in the reducing oxidative stress in the body, and it is also used as antioxidant.
Apart from these qualities, so vitamin E has got one special quality. It is also called as one vitamin. It is called by the other name. Its special importance.
Don't know, ma'am.
Okay. Anti-sterility vitamin.
Okay,
you know what is sterility? If the vitamin E is deficient, sterility happens.
Yes, ma'am. Okay.
Impotence. It leads to impotence in males.
Okay, ma'am.
So do we have any formulations?
Any
yes, ma'am.
Preparations of vitamin E which are available in the market?
Capsules, ma'am, and vitamin E tablets and vitamin E serum, ma'am, for hair oils and all, and in moisturizing creams. Mirac is a supplement, ma'am, which is used in the psoriasis disease, ma'am.
Okay.
And it is also used with zinc and vitamin C combination for wound healing and sunburns, ma'am.
So what is the source? What are the sources for vitamin E?
Almonds, avocados, wheat germ oil, and sunflower oil, peanuts, kiwi fruit.
That means the seeds are more rich, contains more amount of this vitamin E. Seed, any seed contain vitamin E. All seeds contain vitamin E.
Okay.
Okay.
Right. Yeah. Very good presentation, Nikita. All the best.
Thank you, ma'am.
Now, poster code PCHEM 101. PCHEM 101. Hey, Nikita. New approaches in drug treatment for tuberculosis. Next, PCOL 214.
Yes, ma'am. Can I audible?
Yeah, you can raise your hand, ma. First.
Okay.
Chandrika.
Yes, ma'am.
Yeah, your postal code, ma?
PCOL 214, ma'am.
214. Okay. You can start your presentation.
Okay. Is it visible, ma'am? Ma'am, can I audible?
Yes, ma, your screen is visible, but you go to your poster.
Yeah, ma'am.
Yeah.
Is it visible, ma'am?
Yeah, clear, clear. You can start the presentation.
Good afternoon, one and all. My name is Chandrika from Nalla Narasimha Reddy College Group of Institutions, School of Pharmacy. I'm here to present the topic about the artificial blood and alternative approach on current red blood cell and platelet substance. In many recent day in first of all, we have to know what is the role of blood in our body. The blood is in viscous nature and it is alkaline and cellular fluid which carries oxygen to several parts of our body. It in the substance of blood we use the artificial blood in the blood transfusions. The artificial blood is made from plastic and
Chandrika, am I audible to you? Chandrika, we could not hear your voice clearly, ma. A lot of disturbance from your end. Just try to sort it out and present for next after presentation, next presentation, okay? Chandrika, am I audible? Next. PCOL 208. Nishita Harini. Are you there?
Yes, ma'am. Yes, ma'am.
Are you ready with your e-posters?
Yes, ma'am. Ready, ma'am.
Yeah, you can share screen and present the poster, ma'am.
Okay, ma'am.
Okay.
Good afternoon, everyone. I'm P. Nishita and we, Harnipriya, are here to present our e-poster on oncolytic virotherapy. The word oncovirus is onco related to the
Display your poster, ma'am. Your poster is not yet displayed.
1 second, ma'am.
Okay.
Is it visible, ma'am?
Rotate your phone, ma. Now again it's gone. We don't have much time. You just share the screen. Okay. Open the mini window of your mobile. Directly focus. Nishita.
Yeah, ma'am. Harini is sharing the screen.
Okay.
Is it visible, ma'am?
Your screen is visible, ma. But your poster is not displayed yet. Yeah. Okay.
Is it visible now, ma'am?
Yes, it's visible. Just rotate it. Rotate your mobile to get full screen.
Taekwondo.
Ma'am, now it's clear, ma'am.
No, ma, it's opening in edit mode, ma.
If I'm rotating, it's not clear, ma'am.
You zoom in maximum, ma, because we could not see other ads also. Close the ads in your mobile. Who is Akila?
Ma'am, now is it clear, ma'am?
Oh, yes, but these are the things you should do before itself, ma. Okay?
Yeah, sorry, ma'am.
It's okay. Start your presentation.
Good afternoon, everyone. I'm P. Nishita and we Harni Priya from NNRG School of Pharmacy are here to present our poster presentation on oncolytic virotherapy. The word oncolytic virotherapy is a therapy to cure the cancer in based on the selection of the tropical tumor topical tumor virus or the genetic of replication by selecting vector, that is by direct killing infection tumor cell or increasing the susceptibility to the death. Apoptosis, the apoptosis is a process called as programming death of the cell in the addition to the radiotherapy or chemotherapy. In addition, viral vectors can be modified to promote more potent tumor cells, including toxicity of the profile or generate host anti-tumor immunity. Firstly, the word cancer means growth of the abnormality of the cells. That is unwanted cell growth in the organ is called as cancer cells or tumor cells. Accumulating, generating or epigenerating alteration. And coming to the moderate technology of curing the cancer is surgery method, radiotherapy method, and chemotherapy method. And emerging treatment virotherapies are using an oncolytic virus to lyse the cancer cell, and advantages are high specificity, mean few side effects, self-regulating drugs, lack of contradiction. And over to you, Harini. Harini? Harini?
You can hear me. You can hear.
Harini, are you audible?
Am I
Yes,
audible, ma'am?
Yes,
Yeah,
yes.
continue.
How does oncolytic virus works? Actually, they occur naturally or by genetically engineering, transcription targeting, and alterations. Viruses can be currently being explored like parvovirus, SV40 virus, myxoma virus, adenovirus, mumps virus, influenza virus, vaccinia virus, and much more, etc. The activity of oncolytic virus it occurs on healthy cells and as well as the tumor cells. In a healthy cell, same it undergoes undamaged cell, same as the healthy cell. But in tumor cell, virus replication takes place, which in turn leads to the lysis, which means the breakdown of the cell. Local inflammation takes place, destruction of tumor cells takes place, and not only breakdown of cells, it also releases virus progeny. In fact, more tumor cells were released, and with the lysis, release of tumor antigens also takes place. Systemic anti-tumor immune response is also happen. Coming to case studies in India, till the year 2020, the projected incidence of patients with cancer deaths are males 6,79,421 and females 7,12,758. In females, most common cancer were observed is in breast cancer and uterine cancer. 1 in every 15 Indians will die out of cancer. And finally, coming to conclusion, further investigation is needed to characterize the better clinical impact of oncolytic virus, but there are increasing data demonstrating the potential promise of this approach for the treatment of human and animal cancers. Hereby, I'm ending this session. Thank you for giving me this opportunity. Thank you, ma'am.
Okay, ma. All the best. And next. Sneha, postal code 205, PCOL 205.
Yes, ma'am.
Yeah. Start your presentation. Yeah.
Ma'am, is my screen visible?
Yes, ma'am. Rotate your mobile, ma'am.
Okay, ma'am. Is this okay, ma'am?
Start.
Okay. Hello, everyone. Good afternoon. I am Sneha of third year studying in Tigala Krishna Reddy College of Pharmacy. My presentation is about melatonin, which is used as a potential adjuvant treatment for COVID-19. So melatonin, it is basically a hormone which is secreted by pineal gland in the brain. It is primarily responsible for regulating the circadian rhythm, that is to manage our natural sleep cycle. It is often used as natural supplement to overcome issues like insomnia, sleeping disorder. So how this melatonin, we are using it in COVID treatment. So we know that COVID patients have symptoms like fever, dry cough, fatigue, diarrhea. In some severe cases, we can see lung injury, acute lung injury, or acute respiratory distress syndrome (ARDS), and in severe cases, we can see sudden cardiac arrest within few days of the person testing positive. So how this COVID-19 develops is the virus SARS-CoV-2 attaches to the angiotensin converting enzyme 2 receptors in the epithelial cells, in the airway epithelial cells. Then they trigger a pro-inflammatory response, which results in cytokine storm. So what this cytokine storm is, it is nothing but cytokine release in excess amounts, which leads to hyperinflammatory condition in the person. This leads to acute respiratory syndrome. In addition to this, the virus also causes the reactive oxygen species, which damages the alveoli. So what this melatonin does is, the main category or action of melatonin is anti-inflammatory action. So we know that this virus causes uh, inflammatory in the in the alveolar cells of the lung. So this melatonin, it binds to the glycoprotein called CD147, which is responsible for the cytokine storm, and it reduces the cardiac damage by blocking that inflammatory action. This melatonin is a strong hydroxyl radical scavenger, so it protects the cells from the oxidative damages and also enhances the activity of some antioxidant enzymes like glutathione and all. So thus we can say that melatonin is a crucial compound of antioxidative system. Besides this, it also have immunomodulatory action, and when it is administered exogenously, we can see increased antibody production in the body. And when the immune system is suppressed due to this virus, it shows immunostimulant effect by increasing the T cell activation. Coming to the dosage of melatonin, we can take it about 1 mg to 10 mg per day for a month. Short-term doses, there are no side effects and all, but if it is prolonged use or taken in high doses, we can see that headache, nausea, dizziness, and sleepiness are the adverse effects. And besides this, melatonin also shows other beneficial properties like antidepressants, anxiolytics, neuroprotective, and antihypertensive effects. Um and trade name, the trade chemical name of melatonin is N-acetyl 5-methoxytryptamine. In some of the countries, it is a OTC drug, over the counter drug. We can get it anyways. Coming to the conclusion, at the end of the day, we can say that melatonin can just provide relief from clinical symptoms of COVID-19. It cannot eliminate or totally stop the viral transcription in the body, even though it has antioxidant effects, though there are no harsh adverse effects. The study of melatonin in animal models and in humans has shows efficacy and safety. So finally, we can tell that melatonin is predicted to be highly beneficial in COVID-19 treatment. That's it, ma'am. Ma'am.
Okay. Can you mention any discomforts due to excess presentation of the melatonin?
Melatonin
This
is actually a
production.
hormone present in our body, no ma'am. So if we
Yeah.
take it in a high doses, we can see occasional dizziness and a
Hmm.
nausea, headache,
That's all.
sleepiness.
That's all.
Yes, ma'am.
Okay, good presentation.
Thank you.
Next, Sainath.
Yeah, good afternoon. Yes, ma'am, ready, ma'am.
Yeah. Please share your screen and present the post.
Okay, ma'am. Ma'am, the screen is visible, ma'am.
Yes,
Hello.
your screen is visible.
Yeah. Yeah, good afternoon, ma'am, and I wish a warm greetings to every panel members present over here and my fellow participants. This is Sainachari Ganoji, PharmD 4th year from St. Paul's College of Pharmacy, and I have been created the flyer based on the specifications given by the organizers in the brochure. So the topic is motor-driven sperm hybrid, a shrewd ingenious cargo-targeted drug delivery for amelioration of female reproductive tract cancers. So I have been presented over here with the abstract. So the presentation of the topic says that the micromotor-driven sperm hybrid, which has the ingenious and the innovative technology for the targeted drug delivery in order to increase the efficacy in the cancers in the mainly female reproductive tract. The Shreve technology involves the loading of the sperms with the anti-cancer drugs like doxorubicin hydrochloride and coated with a nanometric layer of the iron for enabling and guiding and releasing the sperm at the target by the external magnet. This experiment has been done in the in vitro studies for the clinical trials in order to overrelate the more efficacious results for the treating the female reproductive tract. And the introduction of the topic says a sperm-driven micromotor is presented as a target drug delivery system which treat cancers in the female reproductive tract. Why? Because the current scenario has the many challenges, include the unspecific uptake by the other organs, limited tissue penetrations, and also decrease the effective concentration due to the dilution in the body fluids. These limitations or these challenges are of the chemotherapy. So hence there was an alternative therapy is being needed in order to increase the efficacy. So the scientists or the researchers have thought that why can't to increase the biocompatibility, why can't a sperm cell can drive the force in order to treat the diseases? Because in the recent history, the liposomes are also have been used in order to treat certain cervical cancers. So the researchers have thought that sperm can useful for the treating the female reproductive cancers. And here is the image which have been presented. We can see a sperm which has been attached with the tetrapod in order to guide the sperm towards the cancer towards the cancer cells. Means this is the in vitro presentation where in order to travel the sperm, there has been attached with the magnetic harness with the tetrapod and which drives the sperm towards the cancer cells. And the objectives of the presentation include this presentation of the topic include the system can demonstrate an efficient drug delivery vehicle by loading a motile sperm cell with anti-cancer drug like doxorubicin hydrochloride, guiding it magnetically. And materials and methods. In the materials and methods, they include motile sperm cell and magnetic tetrapod. As I've mentioned already in the introduction, magnetic tetrapod is being used in the in vitro analysis. So the method we can see the drug loading will be coated with the sperm tetrapod and will be driven into the tumor spheroid with the magnetic guiding. Hence it reaches, and with the recapsulation and the propulsion, the sperm releases the drug into the spheroid, that is a tumor spheroid. And next, coming to the results of this particular experiment include 90% reduction in the living cancer cells after the 72 hours. We can see this is the particular image of the results of the fluorescence, where we can see in the 0 hour, there was no change in the spheroid, the tumor spheroid, and of the 24 hours also, there was no change, but after the 48 hours, we can see the red color, which is being showing that is a doxorubicin, and after later 72 hours, we can see the drug has been increased by reducing the tumor spheroid. So the figure says cell killing effect of doxorubicin hydrochloride loaded sperm. And the advantages. So what are the advantages are being there by using sperm in order to deliver the drug towards the tumor includes, because sperm has the membrane fluidity, long life run, and high biocompatibility, can avoid the dose dumping. When we compare to the chemotherapy, we see the biocompatibility is very less and also has the high protein binding, so which can cause the dose dumping and also toxicological effects. In order to avoid that, sperm cells are the excellent candidates to operate physiological environments which neither express any pathogenic proteins nor proliferation, unlike other microorganisms. Because this one of the technique which also used before in a history by the like by the bacteria which is used to treat certain cancers, but the bacteria can cause the autoimmune disorders. Hence, sperm cells are already has the physiological environment suitability. Hence, this can work out for reducing the female reproductive tract cancers. And my conclusion includes it is one of the potential biocompatible cargo delivery in order to deliver the drug towards the targeted tumor tissue, and which is also a scaffold for diversified biomedical pertinence, conspicuously gynecological healthcare. And motor-driven sperm drug delivery could be envisaged for the idolization in the therapy or female reproductive tract cancers in the near future. And these are my references. Through this presentation, I want to conclude that together, let's give women a better chance at life so that the society being developed in a very right way. And thank you so much for this opportunity. Thank you, ma'am.
Okay. Okay, sign up your. Is there any methods for drug loading?
Drug loading, ma'am. Yes,
Yes.
ma'am. Generally, they have been incubated this sperm with the drug for the more than pre incubation, ma'am, for more than 38 hours.
Okay. What is the lifetime shelf life of this formulation? If
No, ma'am.
if you select it for drug loading, how
Yes,
much time we can give assurance for elimination?
So yes, ma'am. Generally, sperm cells have the life for I mean 15 to 30 minutes outside of the human body, ma'am. But when it is being frozen, it has so many days to years to be there in order for the study. So as per the formulation, it has been not approved, ma'am. It is being in the clinical trial process.
Okay.
So
once it is given to
yes,
after drug loading, once it is given the formulation, any
yes,
kind of route of administration.
Yes.
Okay. Oh, what is the efficiency of therapy can we expect?
Yes, ma'am. As per the research studies which have been reviewed, there was a point like there was more than 75 to 80% of the higher efficacy when compared to without any side effects, without
In
any.
drug loading?
Yes, ma'am. Through the drug loading through the sperm hybrid, there was a high
Okay.
efficacy, ma'am. Nearly 75 to 80%.
Okay.
Compared to chemotherapy, ma'am.
Other than
Oral
chemotherapeutic agents, is there any other drugs are loaded?
drugs.
Any
I think,
studies?
ma'am, as per my knowledge through the studies which have been there, they have choose the doxorubicin because doxorubicin have been I've been administered through the body through liposomes, ma'am. So they have been doing study through the liposomal drug suitable for this doxorubicin. Yes, ma'am. Already it is been FDA approved, ma'am. FDA approved, that is Doxil, ma'am. So where liposomes have been used. So through that inspiration, they have took the sperm cells.
Even amphotericin
So
also can be given, right? Through liposomes.
Yeah, ma'am.
Antifungal, amphotericin B can also be
Amphotericin
given.
B, yes, ma'am, is an antifungal.
Yes.
Yeah, fungal.
Even antimonial drugs can be given. Anti-leishmanial
Yes, ma'am.
drugs, antimonial
Yes, ma'am.
drugs.
And this doxorubicin is anthracycline antibiotics, ma'am.
Okay.
So obviously the class drugs can be have the compatibility to this pump. Yes.
Okay, okay, right. Well, presentation, Saina.
Thank you.
All the best. Yes.
Thank you.
Next presentation, Chandana.
Yes, ma'am.
Yeah. You can present the poster.
Okay, ma'am. Ma'am, is it clear, ma'am, my poster?
Yes, ma'am.
Thank you, ma'am. Good afternoon, everyone. My name is A. Chandana, final year student of Tigala Krishna Reddy College of Pharmacy. The topic that I am presenting today is natural herbs as anti-cancer drugs. Nowadays, cancer has become one of the most happening disease across the world. There are many reasons that causes cancer, like food habits, stress levels, pollution, and etc.
Cancer is a disease in which some of the body cells grow uncontrollably and spread to all the parts of the body. Well, herbs have a very vital role in the prevention and treatment of the cancer. In the recent, owing to the fear of side effects, people prefer to use plant products in the treatment of cancer. Several reports describe that report that anti-cancer medicine plants is due to the presence of antioxidants present in them. There are different plants that has anti-cancer agents in their plant parts, such as Vinca rosea, podophyllum, camptothecin, and etc. Vinca alkaloids have cytotoxic effect that arrests the division of cells and cause cell death. Podophyllum contains glycosides, flavonoids, saponins, terpenoids that help in the treatment of cancer. Podophyllum is poisonous when taken by mouth. Camptothecin, it is an alkaloid isolated from the stem wood of the Chinese tree called as Camptothecia acuminata. It is a quinoline alkaloid used as a chemotherapeutic agent in the treatment of leukemia. By this presentation, I want to conclude that plant-derived anti-cancer agents are effective inhibitors of cancer cells. They are naturally and readily available, they have less adverse effect, more potent when compared to the normal medicines. Thank you, ma'am.
Keep your poster displayed, ma'am.
Is it clear, ma'am? Is it visible, ma'am?
Yes, ma'am.
Okay.
What are the formulations, ma'am? What are the formulations can we expect from these natural herbs? Chandana?
Ma'am.
What are the formulations can we expect from these natural herbs
Tablets,
to treat cancer?
Tablets, ma'am, tablets, IV injections, and it is used in the chemotherapy treatments, ma'am, most of them.
Okay. What are the physicochemical factors you should consider to formulate it into tablets?
Ma'am?
What are the physicochemical properties of the drugs, herbal drugs? How it has to be improved?
I don't know, ma'am.
Okay, but well presented, ma. Good presentation. All the best.
Thank you, ma'am.
Next, Akanksha.
Yes, ma'am.
Ma'am, can I stop the screen share, ma'am?
Yeah, you can stop, ma. Chandana. Good presentation. Good. Akanksha, you can share the screen.
Is it visible, ma'am?
Yeah. Can you rotate? Are you presenting through mobile?
Yes, ma'am.
If it is so, can you rotate your mobile to get full screen?
Okay, ma'am.
Yeah, now it's perfect.
Good afternoon, one and all. I'm Akanksha from Nalla Narasimha Reddy Education Society's group of institutions. I'm here with the topic of pharmacy, always trusted for
Your
your
postal
health.
code, ma'am?
P call 209, ma'am.
Okay.
Pharmacy, always trusted for your health with pharmacovigilance. Firstly, what is pharmacovigilance? It is a practice of monitoring the effects of medical drugs after that has been licensed to use, especially in order to identify the evaluate previously unreported adverse reactions. If any drug or any chemical substance, if it is having any adverse effects, these effects can be reported by the pharmacovigilance department. Majorly, it has the capabilities of adverse effect case management, aggregate reporting, signal intelligence, and risk management. If any drug is causing any adverse effects or it is having any risk, that can be avoided and it is reported to the pharmacovigilance department so that these adverse effects can be reduced or they can recall the drug from the market. This can decrease the death rate of the people and also to provide good health to the people also. Firstly, not only this reporting not only can done by the people, but also it can done by the physicians, healthcare professionals, by the industry people, any regulatory authorities, anyone can do this reporting and report to the pharmacovigilance department in order to have good, in order to reduce the adverse reactions of the drug and have better health. Mainly importance of pharmacovigilance in patient care. The main aim of the pharmacovigilance is to monitor the adverse drug reactions of the drug and to provide safe and effective medicines to the patients, to promote rational use of medicines, to create national center for excellence at par with the global drug safety monitoring standards, to identify and analyze new signal for the reported cases, to know the benefit and risk ratios of the drugs, in order to know whether how much benefit is the drug providing and how much the risk it is causing to the people. This can be analyzed with the help of this pharmacovigilance. It has a major impact on KCD, which means chronic kidney disease and end-stage renal disease treatment. These all diseases can also be reported to the pharmacovigilance department. It also plays a major role in prescription drug monitoring program also. If a doctor or any medical professional has given any medicines to the patient, if the patient is having any adverse reactions due to those drugs, it can be directly conveyed to the doctor or the pharmacist who has dispensed the medicines, so that the adverse reactions can be reported to the pharmacovigilance center, so that these adverse drug reactions can be managed, or if these reactions continue, the drug may be recalled from the market. And I want to conclude that pharmacovigilance plays a major role in the healthcare department and monitoring the drugs, their safety and discovery of interactions. Every people have different interactions within them with the drugs, so effects on humans can be recorded, and more awareness can be spread regarding adverse drug reactions. Thank you. Hello, ma'am.
Is there any website to report adverse drug reactions?
Yes, ma'am. We have many websites.
Official websites.
Yellow card website, yellow card regarding UK, ma'am. We can report in that any adverse drug reactions can be reported in that.
In India?
In India, ma'am.
Recently, during Pharmacy Week and Pharmacovigilance Week, recently they have repeatedly mentioning that websites, right? And even toll-free numbers also.
What is the difference between side effect and ADA?
Okay. It's okay, Akanksha. And what is the difference between side effect and adverse drug reaction?
Adverse drug reactions means the reactions between drugs, ma'am. If we take 2 or more drugs, these drugs may have interactions between them, whereas side effects, it may be due to single drug also, ma'am.
Okay. Okay, ma'am. Good presentation. All the best.
Thank you, ma'am.
Okay, Akansha. Next, CH Pranay.
Yes, ma'am.
Yeah, you can start your presentation. Are you ready with your presentation?
Yes, ma'am.
Yeah. After Pranay, Pravalika is ready. Pravalika, you can raise your hand. Pravalika. Okay, it's okay. Pranay, you start your presentation now.
Okay.
Mention your postal code while presenting.
It's P-call 210, ma'am.
Okay.
Ma'am, is my screen visible? Hello.
Yes, ma, your screen is visible clearly.
Okay, ma'am. Okay. Good afternoon, everyone. My name is CH Challa Pranay. I'm studying in Nalla Narasimha Reddy College, 4th year, and my topic is about gene therapy. Gene therapy is a technique that uses genes to treat or prevent diseases by inserting a gene into a patient's cells as drug inserted of using drugs or surgery. Actually, the gene is inserted into the person with the help of a nucleic acid or like just on a drug through an injection or through a tablet. Approaches to gene therapy, including replacing a mutated gene that causes diseases with a healthy copy of a gene, inactivating or knocking out a mutated gene that is functioning improperly. The improper gene is normally replaced through this gene therapy. It is done majorly for cancer to eliminate any improper gene by using this therapy. Introducing a new gene into the body to help fight a disease. It is a promising treatment option for inherited disorders but remains risky. Conventional neonatal intensive care medicine is unable to treat severe inherited genetic disorders. Neonatal gene therapy is too late for an adult to treat effectively, but it appears potentially useful against inherited disorders in newborn infants. Here we present preclinical neonatal gene therapy data for two inherited disorders, metachromatic leukodystrophy and hypophosphatasis. Oh, sorry, ma'am, I can't spell it out. Adeno-associated virus-mediated gene transfer to neonate. A vector is a DNA molecule used as a vehicle to carry foreign genetic material into another cell is termed recombinant DNA. Among the numerous viral and non-viral vectors, adeno-associated viruses vector has emerged as a particularly promising tool for gene delivery. Virus vectors. An AVV is not pathogenic and it has ability to transduce non-dividing cells. AVV vectors as serotype depending on a target after intravenous injections into a neonatal mice of AVV vectors. serotypes 1, 8, and 9 harboring a luciferase gene. Expression of luciferase was detected within 3 days and continues for more than 16 weeks with no disease in expression. Advantages of neonatal gene therapy: penetrates the blood-brain barrier, induces immune tolerance, prevents early onset genetic diseases, enables the use of smaller amount of vectors. Applications of the neonatal gene therapy is in neonatal gene therapy for metachromatic leukodystrophy and in neonatal gene therapy for hypophosphatasia. In neonatal gene therapy for metachromatic leukodystrophy, it is an inherited autosomal recessive lysosomal storage diseases. Yes, ma'am.
Yeah, and good preparation of your poster, ma, and practice at least 2 or 3 times for next presentation, ma. Okay?
Thank you.
Okay, ma. Thank you. Next, Chandini, start your presentation, ma.
Okay, ma'am. Visible, ma'am?
No, not yet. Chandini, I could not see your screen.
Share చేశారా?
Ma'am, is it visible now, ma'am?
Yes, ma. You can start. Yesterday you started this, right?
Yes, ma'am. Good afternoon, everyone. This is Chandini. So my presentation is about acetyl glucose. Acetyl glucose is also known as N-acetyl beta D-glucosamine, whose molecular weight is 221.21 and molecular formula with C8H15NO6. It is widely distributed throughout the world, and its aqueous solution is colorless and clear. It is soluble in 25% of water and 1% of aqueous solutions. The acetyl glucose is produced chemically by the process based on the acid hydrolysis of crude chitin. The chemical degradation of chitin can be performed through hydrolysis using a strong acid such as HCl. Conditions include 15 to 36% of HCl about 40 to 80 degrees Celsius. In this procedure, up to 6.42 grams per liter acetyl glucose can be produced. Production of N-acetyl glucosamine is also seen in mammals, where they are present in components of glycoproteins, proteoglycans, glycoaminoglycans, and other connective tissue building blocks. Biomacromolecules of acetyl glucose seldom exist in free form except in human milk at 600 to 1500 mg per ml. Using chitin as feedstock, acetyl glucose can be prepared through a process that is based on chitin hydrolysis. Chitin is produced in two forms, alpha chitin and beta chitin. And the applications of acetyl glucose is for tissue repair and anti-inflammatory, and in cosmetics, and in the production of salicylic acid. N-acetyl glucosamine is a monosaccharide that usually polymerizes linearly through 1,4 beta linkages. N-acetyl glucosamine is taken for osteoarthritis and inflammatory bowel diseases, including ulcerative colitis and Crohn's disease. Mainly N-acetyl glucosamine is used for the treatment of inflammatory diseases in alimentary tract and gastrointestinal tract, where the inflammation can be reduced by using this chemical. Glucosamine sulfate has high permeability in cartilage, but no tangible effect, while it is opposite is true for N-acetyl glucosamine. It is a secondary amide between glucosamine and acetic acid, and it is a basic component of hyaluronic acid and keratin sulfate on the cell surface. In industrial production for industrial production, we use chemical methods by using acid hydrolysis of crude chitin, and for production and for the production in mammals and for production of biochemicals, we use it through mammals or animals, through chitin or through human milk. So the main application of acetyl glucose is for the tissue repair and to avoid the inflammation which can be which are due to other which are due to other parasites like which are formed due to parasites like osteoarthritis and inflammatory bowel diseases. The acetyl glucose is generally considered to be the agent which can be used for diabetic patients to acetyl glucose is used in the diabetic patients medicines too. Glucose when compared to glucosamine sulfate, acetyl glucosamine is more applicable in usage, ma'am. Thank you, ma'am.
Okay, Chandani, can you tell me the safety profile of acetyl glucose?
Safety profile of acetyl glucose, ma'am. It's generally safer to use, ma'am.
It's completely safe for use, right?
Yes, ma'am.
Is there any profile, any instructions while handling?
Acetyl glucose is not generally while making in chemical while in chemical methods, acetyl glucose should be treated with hydrochloric acid, where it may produce some of the reactions, ma'am, which can cause bruises. So acetyl glucose should be taken in appropriate proportions, ma'am.
So you mentioned that it is used for production of sialic acid.
Yeah, ma'am.
In applications, it's there. It is used for
Yes,
production
ma'am.
of
Production
sialic
of sialic acid. Yes, ma'am.
What is the role of sialic acid?
It is a preservative, ma'am.
Preservative. Okay. Okay, ma'am.
Okay, ma'am.
Yeah, good presentation.
Thank you, ma'am.
M. Sandhya. You can unmute yourself, ma'am.
Yes, ma'am.
Yeah, are you ready with your presentation?
Yes, ma'am.
Yeah, you can share the screen and present the poster. While presenting, please mention your poster code, ma'am. All of you.
Okay, ma'am.
Okay.
Good afternoon, everyone. I'm Esvaya from Nalla Narasimha Reddy College. Today, my poster topic is application of nanotechnology in cancer diagnosis and therapy. First of all, what is cancer? Cancer is a disease which is characterized by uncontrolled proliferation of cells, which starts from initial point to the and can spread to other parts of the body. Nanotechnology has been uh, applied in development of nanomaterials such as gold nanoparticles and quantum dots used for diagnosis of cancer in molecular development. Nanoparticles are promising tools for advancement of drug delivery, medical imaging, and diagnostic sensors. Nano devices are used in the drug delivery, which are used are micelles, vesicles, dendritic polymers, liquid crystals, nanocapsules, and nanospheres, etc. First of all, why we need to use nanomaterials for the treatment of cancer or for diagnosis of cancer means these are small in size and have higher surface area and controlled and target drug delivery, reduced side effects, and low doses required for diagnosis and therapy. What are the nanomaterials used in diagnosis of cancer treatment is liposomes, carbon nanotubes, quantum dots, nanoshells, gold nanoparticles. These are used. Now, talking briefly about the applications of in therapy of nanoparticles, those are liposomes. These are bilayered membrane and water phase nuclei. These are amphiphilic, means they can allow both hydrophobic and hydrophilic drugs. Now, talking about carbon nanotubes, these are single walled, multi-walled, and these absorb light near infrared region and affect the target tumor cells. Colloidal gold nanoparticles, these are good contrast agents because of small size, good biocompatibility, and have higher atomic number. These act by both active and passive ways. In passive ways, how it works means it gathers the gold nanoparticles and enhance imaging because of permeability tension effect in tumor tissues. And in active phase, it works by coupling of gold nanoparticles with tumor specific targeted cells. And I would like to conclude my topic by saying nanotechnology based method is best method for diagnosis, drug delivery, and treatment of cancer and other disease because of novel properties and advantages. Thank you, ma'am.
Okay, Ma, you mentioned carbon nanotubes also one of the drug delivery systems.
Yes, ma'am.
Right? So even you have mentioned these nanoshells, quantum dots, right? Dendrimers. Yeah. And here, can you tell me the difference between organic and inorganic drug delivery systems?
No, ma'am.
No.
No idea.
It's okay. And what is the purification techniques for carbon nanotubes? You said multi-wall, single-wall carbon nanotubes are available.
Yes, ma'am.
Yeah. Any purification techniques?
Don't know, ma'am.
It's okay. It's okay. But your topic is really very good. So here liposomes also is there. Can you tell any information about the stealth liposomes? Stealth liposomes.
Ma'am, can you-
STEELTH. Stealth liposomes. Or pegylated liposomes or long circulatory liposomes. It's okay. It's okay, mom. It's okay. But your topic is really good. Okay. Okay, ma'am. Good presentation. No problem.
Thank you, ma'am.
Yeah. And one more thing, it's a small announcement to you all. You must submit feedback link immediately after completion of the session. Okay. And one more thing, certificate can be generated only the persons who submitted feedback link. Okay. Submission of feedback link is mandatory. Okay. Next, Sakshi, are you ready?
Madam,
Sakshi.
Madam, ఏమైంది?
Sitaram. Mr. Sitaram. After Sakshi, Mr. Sitaram, you present the poster.
Ma'am, this is Fardana, ma'am. Okay, ma'am.
Sakshi, keep displayed your poster. Ma? See, in your poster, you mentioned it is lack of data, right? Noisy social media, big data algorithms. Huh? So, in these conditions, how can we take artificial intelligence as one of the promising tools? Am I audible to you? Sakshi, am I audible to you, Ma? Yes?
Yes, ma'am, you are audible.
Yeah. Tell me the answer.
Ma'am, we have that is why we need to furtherly advance these kind of technologies, ma'am, so that we can share more like we can more share data in those things, ma'am.
Okay. So works has to be need to work. Okay,
Yes,
to
ma'am.
abolish all these problems.
Ma'am, I didn't present my poster, ma'am.
You have started and presented, no?
No, ma'am, I asked that am I audible, but no one replied. I thought I'm not audible. I was seeing for that, ma'am.
Okay. There may be some technical issue in this one. So you can, no issue, no problem. You can continue.
Can I start my presentation, ma'am?
You can continue. Don't start. We are running very short of time.
Okay, ma'am.
Okay. You present main advantages of artificial intelligence and conclude the topic.
Okay, ma'am.
Yeah.
Artificial intelligence is used in research and drug discovery, ma'am. We also can repurpose these existing drugs. That means the drugs which are already known, they can be again studied so that we can find what are the uses that can be useful for the COVID treatment. They also reduces the workload of radiologists, ma'am. They also reduce the workload of radiologists by diagnosing the disease. They also track infected persons and reduce the possible contamination in the nearby areas. We can see many apps nowadays like John Hopkins dashboard, Nexus Train, and Blue Dot. These are in this first one, John Hopkins dashboard is used to confirm generally shows confirmed cases versus recovered ones, and the Next Train updates us how the virus is spread, and the Blue Dot also predicts infectious diseases among others. Blue Dot is reported in 65 languages. And if we furtherly study on these kind of technologies, we can succeed in very good level. And if we combine artificial intelligence with clinical studies and chest imaging, then we can further expect good things in technologies, ma'am. Thank you, ma'am.
Okay, Sakshi, well presented. What is the main advantage of artificial intelligence, ma'am?
Ma'am, it can speed up the process, ma'am.
Speed up the process of what?
Of it might be of drug discovery, tracking the data so that the virus does not spread, and also it can diagnose and detect the disease earlier when compared to human.
So how can we expect applications of artificial intelligence in future?
Ma'am, we should, as I said earlier, we should combine artificial intelligence with clinical data, ma'am, so that more already we have data that like diagnosis and laboratory tests and exposure symptoms, exposure histories and symptoms. These can be correlated with the artificial intelligence for further accuracies, ma'am.
Okay, ma'am. Okay, very good. Good presentation. Next, Sitaram.
Yes, ma'am. I am ready, ma'am.
Yeah. Start your presentation.
Other participants and ma'am, is it clear, ma'am? Ma'am, is it visible, ma'am?
Yes, I can see your screen, ma'am.
Yes, ma'am. Now, good afternoon, everyone. This is Farzana studying before.
And display your poster. Display the poster. I can see your screen, but I can't see your poster.
Display అవ్వట్లేదు. కనిపిస్తుంది కానీ డిస్ప్లే అవ్వట్లేదు.
Your screen sharing. Your screen sharing అని వస్తుంది. Is visible అవ్వట్లేదు.
డిస్ప్లే అవ్వట్లేదు అని చెప్తున్నాను సార్. కనిపిస్తుంది. Ma'am?
Now it's okay, ma'am.
Good afternoon, everyone. This is Farzana studying B Pharmacy final year in Madhya Pharmaceutical Education and Research. Now, I would like to share monoclonal antibodies available on COVID patients. Monoclonal antibodies has more effect on the COVID patients. Like it is a very effective antibody. It is affected on the antigen which is caused by the Omicron and COVID line. An antibody produced by a single clone or cells and cell lines consisting of identical antibody molecules. A monoclonal antibody consists of monoclonal antibody infusion treatment. It can be given by the IV infusion. These antibodies are similar to one you would be naturally make it response to the infections, and monoclonal antibodies mass produced by a laboratory. These monoclonal antibodies are also called as monoclonal antibody infusion treatment. It is mainly treated with COVID patients. It is less harmful to the COVID patients because these monoclonal antibodies enter into the body and attach with the antigens. These antigens can be protected by the monoclonal antibodies, and this is not be attracted by the other viruses. The first figure is introduced that monoclonal antibodies bind to the target cells and once attacked, there is a harmless. This table number 1, monoclonal antibodies treatment, which is given by the IV infusion, and IV infusion enter into the human body. They easily attach it with the antigen, which is responsible to attach the antigens, and it is very less harmful effective from protected from the COVID patients. These monoclonal antibodies methodology is monoclonal antibody therapy is given by intravenous infusions. These infusions are 1 hour department infusion enter and required about hour to administrated. One possible effects of monoclonal antibody therapy is allergic reaction. When allergic reaction is produced, administered suddenly administration of monoclonal antibodies, please consult the doctor and reduce the allergic reactions. These monoclonal antibodies having more side effects like nausea, vomiting, hypersensitive reactions, allergic reactions. But it is a application of monoclonal antibodies is it is very effective against the COVID patient and Omicron. This is our imivibab. Discussion as the name itself suggests that monoclonal antibodies, it is target one specific site of antigen. It is COVID-2 virus, blocking and the entry of healthy cells and protecting of the body from the same. Most of the people who recover from COVID-19 develop antibody against the laboratory and bind to the spike proteins of the SARS-CoV-2. SAR means serum acute respiratory syndrome COVID patients. And finally, conclusion, monoclonal antibodies are effectively work against an COVID patient administered by systemically and nebulization routes or intravenous routes. More side effects of monoclonal antibodies is allergic reaction and hypersensitive reaction. Thank you.
Okay, ma'am, moving further now.
Yes, ma'am.
Okay. So, can you differentiate monoclonal versus polyclonal?
Monoclonal means different types of spike proteins attached to the antigen, ma'am. Different antibodies going to attach with antigens which is present in the human body.
That is?
Different types of antibodies, ma'am.
That is polyclonal.
Monoclonal, ma'am.
Polyclonal?
I don't know, ma'am.
Okay, okay. Okay, ma'am.
Thank you, ma'am.
Any examples, ma'am, for monoclonal antibodies?
Irmivibab, ma'am. Irmivibab IV infusion, ma'am.
Okay.
Yes, ma'am. Thank you, ma'am.
Next, Silpa. Vijaya Silpa.
Yes, ma'am.
Yeah. After that, Anand, P. Anand. After Silpa, Anand. Okay?
Okay, ma'am.
Silpa, you can share the screen.
Yes, ma'am. Is it visible, ma'am?
Okay, ma, rotate it. Yeah, now it's clear.
Okay, ma'am. Good afternoon, everyone. Respected sirs, madams, and my fellow members. I'm R. Vijay Shilpa. I'm from TKR College of Pharmacy. I'm going to present on the topic colon targeted drug delivery system. So it is a system designed to deliver the drug to target site, that is colon, and it is specially designed to deliver the drug which are degraded by the digestive enzymes in the stomach, such as peptides and proteins. And it is also used to deliver the drug of lower molecular substances to treat colonic diseases like Crohn's disease, ulcerative colitis, and other problems. It can be used as sustained or controlled release delivery system when administered orally and can be used for local therapy. Coming to the drug absorption, due to the morphological and functional aspect, drug absorption is done by carrier-mediated transport system, which is rapid and effective. And the main advantage of colon-targeted drug delivery system is drugs which are poorly soluble in GIT can be designed through this system. Coming to the advantages,
we can reduce the gastric irritation caused by the NSAIDs. And we can, as I said, we can treat the affected areas with smaller doses. Coming to the approaches, there are, as we know, the pH of the colon is 6.1 to 7.5. The key point of this approach, the first approach is to restrain the acidic environment of the stomach, by use of polymers such as Adragit L100, Adragit S100. And coming to the next approach, pressure controlled drug delivery system. As digestion occurs due to the peristaltic movements of the stomach and intestine, the pressure is created, which is a key point for this kind of approach. And by this approach, we can deliver the drug into the colon by using polymers such as ethyl cellulose. And coming to the third approach is co-diestem. This approach is mainly used to minimize the problems related with the pH and the time, and it consists of two layers, acidic layer and a HPMC layer, and the drug is covered with these two layers. And coming to the next approach is pulsing cap system. In this system, the formulation is developed in a capsule form and a plug is placed in the capsule, which controls the release of the drug. Polymers such as HPMC and polymethyl methacrylate can be used. The main advantage of the system is the capsule, when comes into the contact with the dissolution fluid, the drug is released at the target site. And thus I conclude my topic by saying that the colon drug delivery system offers benefits for local and systemic effects. Thank you, ma'am. Hello, ma'am. Hello, ma'am.
Tell me the applications of pulsing cap systems.
As I said, it delivers the drug at the target site. And
what kind of drugs can be delivered to the colon, especially?
The drugs which are poorly soluble in the GIT, like ciprofloxacin, ibuprofen, which causes gastric irritation, theophylline, can also be delivered.
You mentioned about pulsing cap systems, right?
Yes, ma'am.
Is there any example for pulsing cap systems can be delivered to the colon?
General capsules can be.
Pulsing cap systems.
Yes, ma'am.
Pulse in cap. Yes.
No, ma'am. I don't remember.
You didn't remember. Okay, but your presentation and poster, way of preparation of poster is very good, ma'am.
Thank you, ma'am.
It's clear. Your poster code is PCEU 404, right?
Yes, ma'am.
PG Pharmaceutical Analysis, PKS, right?
Yes, ma'am. PCEU 404.
Okay, okay. All the best.
Thank you, ma'am.
Next, next Anand, get ready. After Anand, like Unisa, are you there in the meeting?
Yes, ma'am, I'm here. Yes, ma'am, I'm here in the meeting.
Yeah, means previously your earphones are not connected. That's
yes,
why.
ma'am, not.
Yeah.
Yes.
After this, Anand, you present your posters.
Okay, ma'am.
Are you
I have
ready
to share
with your
the
new
screen.
poster?
Yes, ma'am. I can share, no, ma'am, with the screen.
Yeah, you can share. You can share.
Okay.
After this presentation, you present the poster.
Okay, ma'am.
Okay. Anand, start the presentation.
Okay, ma'am.
Ma'am, is the poster visible, ma'am? Ma'am, am I audible?
Yes.
Hello, ma'am.
I can hear you and your poster also visible. Just zoom in, ma'am.
Is this okay, ma'am?
Okay, okay. Start your presentation.
Thank you, ma'am. I wish you a very happy afternoon to all the participants and the panel members who are present in the session. My name is Anand and I'm from third year B Pharmacy from the prestigious Tigara Krishna Reddy College of Pharmacy. Today I'm here to present my views on the topic role of steroid in COVID treatment. First, let me say what is a steroid. Steroid is a man-made version of chemical known as a hormone. It is good at reducing the inflammation. First, what inflammation? It is nothing but it is a natural immune response that our body fight against a toxin, infection or injury, etc. It is an attempt to heal itself. It is not like inflammation. Steroids are used has a dramatic effect in reducing an inflammation or reducing a pain. Steroid has a great effect in reducing the pain or inflammation in 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 a therapy. In this short communication, I'll discuss the recent literature regarding how steroids are employed in COVID therapy. These corticosteroids are employed to treat allergies, inflammation, and other autoimmune disorders. These steroids are used to treat critical illness patients in COVID therapy with low doses of corticosteroids. These corticosteroids are given in low doses, like for 7 to 10 days. The dose of the corticosteroid depends on the administration of corticosteroid to the patient. Now I request Kavita to continue the session. Hello.
Hello, am I audible?
Yes, you are audible, ma.
Good afternoon to one and all. My name is P. Kavita. I am from Tigara Krishna Reddy College of Pharmacy from third year. As my friend said earlier, steroids were being employed in the COVID therapy for their anti-inflammatory properties. So the steroids mainly reduce the inflammation by suppressing the pro-inflammatory mediators produced by the immune system. So the main drug which has been employed to treat the COVID is dexamethasone. It has been tested by the scientists at the University of Oxford and found that dexamethasone reduces death by 1/3 in ventilated patients and to 1/5 in a patients who require only oxygen supply. So these corticosteroids can be given in various forms such as pills, vial injections, depend upon their formulation. The alternative regimens other than dexamethasone are hydrocortisone, methylprednisone, and prednisolone. So these corticosteroids do nothing to prevent the replication. They will not prevent the replication of the virus, but they increase the ability of the host to fight against the infection. So I would like to conclude that these corticosteroids have been emerged as a life-saving medications which are recommended for the patients with severe or critical COVID conditions. During the severe COVID conditions, these corticosteroids are mainly prescribed by the WHO because at the time of COVID, the patient will develop a overstimulation of the immune system, which can be harmful to their health. So these corticosteroids act to suppress this overstimulation. So further well-developed design clinical trials are urgently needed to evaluate the safety and efficacy of the steroid therapy in COVID-19. So thank you for everyone giving me this wonderful opportunity.
What is the steroid used in COVID therapy?
Dexamethasone, ma'am.
Dexamethasone. Other than dexamethasone?
In pregnant women, it can be a prednisone, hydrocortisone.
Dexamethasone is the what is the marketed name? No?
Don't know, ma'am.
Yeah. Okay. Good presentation. All the best. Next,
Thank you, ma'am.
Laikunisa.
Yes, ma'am.
Yeah. Start your
Okay.
presentation.
Okay. Once again. My screen is visible to all of you. Hello, good afternoon, ma'am. My screen is visible to you all.
Yes, ma'am. You can
Okay,
start.
ma'am. Okay, ma'am. Good afternoon, everyone. My name is Laikunisa. I'm a M.Pharm first year pharmaceutical student. I joined this year, and my topic is COVID-19 vaccine development. It's a global challenge. I just want to say that my topic I have chosen because it is the burning topic, and we need a lot of efforts from our sides, from all the countries globally. We all are working on the development of COVID-19. So it is a very different. There is a difference in the development of COVID-19 vaccine comparing with that of typical development. And now what is the I have given the compare and contrast between the typical development of vaccine and COVID-19 vaccine development. So preclinical stages for those typical vaccines were it was about 18 to 30 months. Now it has been reduced to 0 months, means without a preclinical trials, means the trials which are before testing on the human bodies, before testing or before giving to the human patients, trialing on another animals, or testing the safety data, etc. This all coming at a preclinical stages. So it is a zero, means how could we find the effectiveness of this vaccine? Okay, that has to be improved in order to develop a perfect vaccine. Compared to them, phase 1, phase 2 also, there's a lot of difference in the month. So based on the severity of the condition, how it is worstly affecting the global human beings. So based on that, the vaccine is developed, but without proper efficacy, finding proper efficacy of the drug, and without proper clinical and preclinical testing on the drug, because the time limit what we are taking for the development of a vaccine, okay, comparing to typical and now what we are giving, it's a lot of difference, means there's a months of differences there, okay, which we are lacking. So friends, we should all focus on the development, and where we are lacking back, okay, the testing, everything what we are taking, the time what we are giving is way less. That's why I think that's why, because of this reason, we are not able to get a proper results for the vaccines what we are getting. Okay, and it is as we all know that it is a COVID-19 is caused by corona. It is COVID-19 is coronavirus 19, and it is now it is called as COVID-19, but it is also called as SARS-CoV-2, like a severe acute respiratory syndrome COVID-19 2 strain. Okay, and the possible mode of transmission is it's airborne route. And what is the challenges we are facing for the development of the vaccine is like limited understanding of pathophysiology, and targeting humoral or mucosal immunity. Okay, and also the lack of suitable animal model, like what we are testing for on rabbits and that rats, similar to the human body. But this virus is giving a deceive to all of us, and also pure success of human severe acute respiratory syndrome. So we can see I have shown the graph also, that in that graph there is a lot more resources are available in our country compared to any other countries, China, UK and Germany. Like we have more resources available. with the utilization of those resources, we can make a vaccine. Okay, our country can make a perfect vaccine to treat this disease. Okay, ma'am, my topic is over.
Okay, Unisa. Here, your presentation is really good and very informative as per current situation, what we are
Yes.
facing so many challenges now.
Yes, ma'am.
But it's good. But thing is, I could not find your details on the poster, ma. Just to verify the poster.
Just check, ma'am.
There is no poster code and even your name
I have,
and
yeah,
nothing is, I could not find. No problem, no issue.
on the
Just,
poster.
yeah, on the, what are the
Okay,
poster
okay.
you
It's
displayed?
optional. It has been given, so I didn't mention. Optional, they have
It's not
mentioned.
there. It's not there. It's not there.
Okay.
It is there like MC University, Hamilton, Canada.
Okay, ma'am.
So can I find the author of this poster?
Yes,
Yeah,
ma'am. I have
Akam.
been, yeah, from Mac University.
Is Akam is there in the group
Yeah.
now in the meeting?
No, ma'am.
No, it's okay. No issue. No issue.
Mm.
Okay, right. Good presentation, Anisa.
Okay, ma'am. Thank you so much.
Anand? అయిపోయిందా కదా మానస
Ma'am.
మానస
Yes, ma'am.
డి మానస
Yes, ma'am.
PMAN 502.
Yes, ma'am. Shall I start?
After that, Manidipika, get ready, ma.
Okay, ma'am.
Right.
Ma'am, shall I start sharing?
Yeah, you can start.
Okay, ma'am.
Manasa.
Just a minute.
Ma, if it takes time, I give chance to Manidipika.
Okay, ma'am.
Ma, as per our instructions we received, session will be closed within 15 to 20 minutes, ma. That's why we are speeding up. So we should not delay. Actually, the session supposed to close by 3:30, ma'am, but we extended even 15 more minutes poster presentation session.
Ma'am, is it visible now?
Yeah, I can see your poster. You start the presentation, Manasa.
Okay, ma'am. Good afternoon, everyone. I'm Dimansa Reddy from 4th year, Tigala Krishna Reddy College of Pharmacy. Today, I'm going to present about computer-aided drug delivery system and drug design. Generally, when we introduce a new drug into the market, it is a very complex, risky, and costly process. Generally, it takes 10 to 14 years and more than $1 billion dollars of capital is invested. During the drug discovery phase, 10,000 compounds are identified. But when it comes to preclinical phases, it is shortlisted to 250 compounds. And during the clinical phases, that is phase 1, phase 2, phase 3, and phase 4 studies, only 5 compounds are analyzed. And then FDA approves only one drug. That is it taking 10 to 14 years for approval of one drug. So here, computer-aided drug delivery system and drug design system helps in various stages of drug design, thus reducing the cost of research work and development time. Majorly, computer-aided drug delivery system is of two types: structure-based drug design, ligand-based drug design. Structure-based drug design is further classified into binding site identification, docking, and scoring. Docking is nothing but it is a technique used to predict how a protein is interacting with a ligand molecule. And ligand-based drug design is further classified into two types: pharmacophore modeling and qualitative structure-activity relationship. Pharmacophore is nothing but it is a part of a molecule that is responsible for particular biological interaction. Then the virtual screening can be done and a compound is selected. And it is further lead is identified and a new drug is approved. Main parts and main aspects of computer-aided drug design is modeling, screening, target identification, validation, optimization, database, etc. Major applications of computer-aided drug design is it can reduce the synthetic and biological testing efforts. It gives most promising drug candidate by elimination of poor efficacy rate, poor ADMET. ADMET is nothing but adsorption, distribution, metabolism, elimination, and toxicity levels. It is cost-effective process, time-saving, rapid, and automatic process. Finally, I want to conclude my presentation on a keynote that computer-aided drug design and drug delivery system is an effective tool in the area of drug discovery and development. It is a cost-effective way, and in the past years, it has shown impressive researches, and it will play an important role in future also. And it played a very important role in vaccine discovery also. Thank you, ma'am.
Manasa, can you tell any software techniques for development of CAD? Hello, can you hear me?
Yes, ma'am. I don't know, ma'am.
Don't know? Okay.
It's okay, ma. Very good presentation, Manasa. It's Manidipika. Start your presentation, ma.
Ma'am, I think she's not there, ma'am.
Manidipika, am I audible to you, ma?
Ma'am, she's not there in the meeting, ma'am.
She's not there in the middle. Very good, very good. Next, Rakesh. Third year.
Yes, ma'am.
Now, start your presentation, Rakesh. After Rakesh, Sana, are you ready?
Is it audible, visible, ma'am? Screen?
Yes, yes, yes.
Can I start now, ma'am?
You have only 3 minutes for presentation.
Okay, ma'am. Good afternoon, good afternoon, one and all. My name is D. Rakesh. I am studying B Pharmacy third year in Tigala Krishna Reddy College of Pharmacy. Today I am presenting my e-poster, mucormycosis. Mucormycosis, it is a rare and deadly fungal infection which is also called as black fungus. It is infecting to the patient of novel coronavirus diseases in India. On May 9, 2021, the Indian Council of Medical Researchers and Union Health Ministry issued an adverse screening, diagnosis, and management of black fungus.
Rakesh, your poster is not visible clearly, ma'am. Make it full screen.
Ma'am, it is full screen, ma'am.
Full screen. No.
Is it okay, ma'am?
Okay. No, just rotate your mobile.
Okay, ma'am. The causes of the mucormycosis are: first, the black fungus. It is caused by the group of molds. It is also known as mucormycosis. It is present in soil, plants, and decaying substances. There are different types of mucormycosis which can affect to the body parts. There are different types of mucormycosis are rhinoarbitrocerebromucormycosis, which is affected to the nose, eyes, and brain. There is pulmonary mucormycosis where it is affected to the lungs. Gastrointestinal mucormycosis, which is affected to the stomach, and cutaneous mucormycosis, which is affected to the skin. Among these, the most infected one is rhinoarbitrocerebral mucormycosis, where it is affected to the nose, eye, and brain. And once it is black fungus, it is enters to the nose, it is affected to the eyes, and then it is transmitted to the central nervous system. When it is affected to the brain, once the black fungus enters into the brain, it causes the life-threatening condition. So early detection is very needed before it is entering into the brain. The symptoms which is include are headache, nasal congestion, facial pain, loss of vision. If it is left untreated, it may result in the blindness, removal of the nose, jaws, or even death. First, we have for diagnostic test, we have to take the sample of fluid from your nose and sending into the lab. Or other diagnostic methods which are involved is tissue biopsy, MRI scanning, CT scanning to diagnose the mucormycosis. For treatment part for the mucormycosis, it is we are giving the medications like amphotericin B. The amphotericin B, which is act as an antifungal agent, which is an act against the black fungus. The role of this amphotericin B in fungal cell, the squalene, it is converted into the lanosterol and it is converted to the ergosterol. This amphotericin, when is giving to the patients who are suffering from the black fungus, this amphotericin acts on the ergosterol and it is binds to the fungal cell membrane. As it is binds to the fungal cell membrane, the fungal cell membrane should be damaged. It damages the cell membrane. As it damages the cell membrane, the cellular contents which all lost and the cell becomes shrinkage and the cell become dead. So in this way, this amphotericin which is act on the fungus and it is acting as an antifungal agent. Now, how it is spreading? The black fungus, how it is spreading? The fungus, it is found throughout the environment and most people breathe them without any effect. The people with weak immune system can develop infections in the sinus and lungs. It is doesn't spread from person to person. As we see in COVID, it is spread from the one person to another person. But black fungus, it is not spread from one person to person. It is only spread it is spread to the persons who has low immunity levels. As there is a low immunity levels, the black fungus are the favorable condition where there is a low immunity levels. And it is rare skin, it is also presented to the skin infection only when skin is injury. To overcome this one, we have to keep the blood sugar levels normal. During COVID-19, most steroids are using to the patients. As you're most using of steroids, there is a decrease in the immune system. As there is a decrease in the immune system, the antifungal, this black fungus is majorly, it will cause into the patients. Before COVID, the black fungus, it is very rare. It is present only once in the 1 lakh of people. Now in COVID-19, in second wave, the most of the peoples are suffering because in COVID-19, the most of the patients are suffering. Most of the patients are having less immune system. This black fungus is developing where there is a less immune system. So giving steroids is maintain the steroids levels normally. That's ma'am.
Rakesh, can you give any other antifungal agent which can be used or only amphotericin B for black fungus?
No, ma'am. Other than the amphotericin B, we have the other medicines, ma'am, to cure that one.
Which one? Can you tell?
Posaconazole, isavuzanazole, lysosomal amphotericin B injection. It can be given through injections or the pills, ma'am, for swallow.
Rakesh,
Yeah, ma'am.
what are the formulations through amphotericin B can be administered? Through what kind of formulations?
It is an injectable. It is given through a IV injection, IV route, ma'am.
IV injection in which formulation? Any carriers or directly drug is dissolved in a vehicle can be injected?
It is giving to the vehicle.
But amphotericin B, if it is exposed to healthy tissues, is there any issue or it's okay?
Ma'am?
Amphotericin B, if it
Yes, ma'am.
exposed to healthy tissues,
Yes, ma'am.
what is the effect?
If it is damages to that tissue, ma'am, only it is
Then-
effects on the
then if it is-
antifungal tissues and it will
Yeah.
damages the fungus cells.
So how we can make that amphotericin B, as you said, it can be injectable. By injection, how it can reach the target site?
It will have some detectors. It will only binds with the fungal cell which has the ergosterol, ma'am. It will detect amphotericin and it will binds to the ergosterol and it will binds with that ergosterol, ma'am.
So amphotericin B without ergosterol, it cannot bind to any tissue?
Only it will it acts on fungus cell, no, ma'am. Because we are dealing with fungus, black fungus. In ergosterol, it will binds with an amphotericin and it is destroying that fungal cell.
Okay, good. Good, ma'am.
Thank you. Thank you, ma'am.
PMAN 504 postal code. Nandini, are you ready?
Yes, ma'am.
Yeah, start your presentation.
Okay, ma'am. Ma'am, is it visible?
Yes, ma'am.
Good afternoon, everyone. This is Nandini from Tiga Krishna Reddy College of Pharmacy. I'm presenting the topic of drug waste management. Nowadays, we know that drug waste management is an alarming issue nowadays. So I need to give presentation on that because drug waste contain all the atoms that people no longer have any use, for which they either intended to get rid or have already discarded. Drug waste is caused for the concern because it poses threat to human and environmental health because of the dangers. Drug waste cannot be disposed like conventional waste. It requires a special handling, whether it comes from a hospital, pharmacy, or private household. There are several concerns with the drug waste management. Some drugs contain heavy metals, endocrine disruptors, and to many other compounds that are dangerous for animals and environment too. These problems are mostly occurred in river streams that the normally prescribed doses, and there is concern that chronic exposure to the prescribed dose, chronic exposure to the cases causes serious health problems that can act and synergistically and to cause adverse health effects. There are some methods for drug disposal. The most obvious way for an individual to dispose a drug is landfilling. This landfilling helps in many ways to dispose of drugs. It is in two types. It is in open dump or engineered landfill. In open dump, untreated drugs discarded into discarded. This method is suitable for less toxic drugs because these may not lead to less pollution of soil and environment. Whereas in engineered landfill, it consists of final site that allow solid waste in a secure manner by minimizing the impact on the environment. There are some other methods for drug disposal that are incineration, autoclave, microwaving, deep burial, secure landfilling, waste immobilization, reuse of drugs, recycling of drugs, and bioremediation. There are some special for special guidelines to dispose formulations of drugs like tablets and capsules, where some short tablets like 50 to 100 tablets are collected in a polythene bag containing tea coffee grind. They have to be sealed and have to put in the trash, like NSAIDs, antidepressants. Whereas like oral liquids and intravenous fluids, these liquids should be diluted with the water before before disposal of those liquids, whereas injectable and ampoules have to be braked before dumping, whereas steroids, the small quantity of steroids to be taken in NaOH before disposal, whereas large quantity can be disposed in an incinerator. There are some risks to minimize drug waste. Take back program and by drug donation. I conclude that there are many advantages of pharmaceuticals through treat humans and animals, but it also have disadvantages in case of improper use of medicine by looking at the amount at which the pollution of the environment due to pharmaceuticals or various drugs. This make classification and proper disposal of expired and unexpired drugs without causing any harm to humans, animals, and environment. It may help to reduce the danger today and years to come. I conclude that we have to be careful while disposing the drugs. Thank you, ma'am.
Okay, ma'am. Is there any regulatory approvals? Is there any SOPs are there for disposal of the drug, or we can
Yes, ma'am. There are some FDA guidelines to dispose the drugs.
That depends on based on
formulations.
Based on formulations and characteristics of the
drugs.
Okay. But if it is improper disposal of the drug occurs, what are the consequences we can face?
There would be water pollution, air pollution, soil pollution.
Water pollution in terms of?
In terms of, they are harmful for animals, aquatic animals.
Okay. Aquatic, okay. What about groundwater?
Groundwater, it would be harmful for the people who consume that water.
Okay, even crops.
Yeah, crops.
Okay. Good presentation, Nandini.
Thank you, ma'am.
All the best. All the best. Beulah, are you ready? Beulah. Next. Pranati. PChem 106.
Ma'am, yes, ma'am, yes.
Pranati is there?
Pranita, ma'am.
103. Pranita 106, 103 Pranati. 103 Pranati is there? No? Okay,
No,
Pranita, you start your
Yes,
presentation,
ma'am.
ma'am.
Yes, ma'am. Sana is sharing the screen, ma'am.
Okay, you can share the screen. Nandini, you stop share the screen. Okay. Pranita, 106.
Yes, ma'am.
Yeah.
Yes, ma'am. Is my screen is visible?
Yes, ma'am. Start your presentation.
Okay, ma'am. A very pleasant good afternoon to everyone. Here I am, Sana, going to present the e-poster on the topic of named reactions. Named reaction is a chemical reaction named after its discoveries or developers. A chemical reaction plays a crucial role in organic chemistry, which continues to grow in its ability to construct ever more complex and diverse chemical molecules. The reactants in the reaction may undergo oxidation, decomposition, elimination, addition, rearrangement, substitution. Named reactions are the chemical reactions which are used in our daily life. Some of the named reactions such as Scott-Bowman reaction used for the synthesis of vanillin and capsaicin. Now coming to the Scott-Bowman reaction. It is developed by the Carl Scotten and Eugen Bowman, so that its name is turned as a Scott-Bowman reaction. Alcohol reacts with acid halides in the presence of strong base to form an ester. Is known as Scott-Bowman reaction. In this reaction, phenol reacts with the benzoate chloride in the presence of strong base, that is NaOH. It forms the phenyl benzoate in which ester functional group is present. Mechanism. Phenol, in the presence of sodium hydroxide, it forms the sodium penoxide ion. Phenol itself forms the penoxide ion, which is a resonance stabilized structure. Penoxide ion. In phenoxide ion, oxygen contains the two lone pair of electrons. Oxygen is an electron-rich species. The lone pair of electron will attack on the carbon and forms an intermediate compound. In intermediate compound, the oxygen contains the negative charge that is migrated to the between carbon and oxygen, and chlorine is eliminated. As a result, product is formed in which functional group is present. Ester functional group is present. Uses. It is used in the synthesis of N-vanillin, is known as a synthetic capsaicin. Next remaining topic is hand over to the Pranita.
Ma'am, this is Pranita from second year B Pharmacy from Tegal Krishna Reddy College of Pharmacy. I'm going to say about Cannizzaro reaction. This reaction was developed by Stanislav Cannizzaro. Here, this is a chemical reaction in which 2 moles of aldehyde with lack of alpha hydrogen, when heated in the presence of concentrated sodium hydroxide base, it undergoes disproportionation reaction. Here, 1/2 molecule of aldehyde is oxidized to carboxylic acid, and the other half aldehyde group is reduced to alcohol. And this is known as Cannizzaro reaction. Here, alpha aldehydes with alpha hydrogens do not undergo this reaction, whereas aldehydes with alpha hydrogens undergo aldol reaction, that is aldol condensation reaction. And coming to the mechanism, there are 3 steps in the mechanism. In the step 1, attack of hydroxyl ion on the carbonyl carbon takes place and forms an intermediate complex. In the second step, the hydride ion transfers from the step 1 intermediate, and this reacts with the another molecule of benzaldehyde and forms acid and another intermediate. In the step 3, acid-base reaction takes place between the intermediate and acid which are produced in the second step in the presence of base and forms sodium benzoate and benzyl alcohol. Coming to the uses, it is mainly used as raw material in the varnish industry, that is for acrylic paintings and all. And this reaction yields neopentyl glycol, which is being used in synthetic lubricants and plasticizers. And I conclude that named reactions are the main source to formulate any drug which is used for the treatment of many diseases. Thank you, ma'am. Ma'am. Hello, ma'am. Hello, ma'am.
Which type of aldehydes are used, ma, in aldol condensation?
Aldehydes which lack alpha hydrogens, that means which do not contain alpha hydrogens, ma'am.
In aldol condensation?
Cannizzaro, ma'am.
I am asking in aldol condensation.
In aldol condensation, aldehydes which contain alpha hydrogens, ma'am.
Okay. What are the applications?
Of Canizarum.
Aldol condensation.
No idea.
Applications of aldol. No idea. Okay, ma'am. Okay.
Next participant.
మళ్ళా ఫోన్ పట్టిండు
Bila, are you there? Faria.
Yes, ma'am.
Start your presentation, ma.
Yes, ma'am.
Good morning, everyone. I'm A. Srija, studying B Pharmacy second year in Tikala Krishna Reddy College of Pharmacy. And the topic I'm going to explain is nutraceuticals. To face present day situation and to maintain proper health, it is important to take good food and let that food be our medicine. And the only food that has medicinal benefits are nutraceuticals. Actually, nutraceutical is a term that combines the two words nutrition, which is a nourishing food component, and pharmaceutical, which is a medicinal drug. This term was coined by Stephen D. Felice in 1989. Nutraceuticals are classified on the basis of their natural sources, pharmacological conditions, as well as chemical constitution of products. Most often, they are grouped into four categories. They include dietary supplements, functional foods, medicinal foods, and pharmaceuticals. These nutraceuticals are used to improve health and prevent chronic diseases and postpone the aging process and in turn increase life expectancy. And remaining topic hand over to Beulah.
Hello. Is my voice is audible, ma'am?
Yes, ma'am. You can proceed.
Examples of nutraceuticals are ginger, garlic, and turmeric. The main application of the active ingredients that forms a ginger to participate and have a composition is ginger ale, and it is also a spice and medicine for treating nausea, dysentery, heartburn, diarrhea, loss of appetite, infections, cough, and bronchitis. And the application of garlic is it functions as a antimicrobial, anti-cancer, antioxidant, immune boosting, hepatoprotective, anti-diabetic, and prevent cardiovascular diseases. And the main ingredient present in the garlic is allicin. And next about turmeric, a wide spectrum of therapeutic effect, has an anti-inflammatory, antibacterial, antiviral, antifungal, and wound healing, antioxidant, and anti-cancer. The main active ingredient present in turmeric is curcuminoids. And next I hand over to Srija for remaining two.
Tomato. It is used as antioxidant, cardioprotective, anti-diabetic, antimicrobial, anti-cancer, and it is a photoreceptor. And the active ingredient present is lycopene. Carrot. Carrot is used as anti-mutagenic and it protects from stroke, high blood pressure, osteoporosis, and heart diseases and bronchial asthma. And the active ingredient it contains is beta carotene.
Then
What?
nutraceuticals are the products which that can be considered as non-specific biological therapeutics used to promote general well-being, control symptoms, and prevent malignant process. And the few of the nutraceuticals are categorized as categories that include as dietary supplements and functional food, medicinal food, and pharmaceutical products. Mainly, the few nutraceuticals are used as dietary supplements which contains and produce nutrients derived from food products. Like for example, like concentrated in liquid capsules, pills, and powders. And here I also conclude that about the topic of nutraceuticals, that to make use of nutraceuticals as they have potential nutritional, safety, therapeutic effect, and maintain normal physiological formula. Thank you.
Can you give any examples of marketed products, ma? Nutraceuticals.
Lycopene.
Any marketed products.
Lycopene is a product produced from stomach.
Lycopene is a product or chemical constituent?
It is also a product.
Any other?
Beverages.
Okay. Okay.
Thank you, ma'am.
Apurva, are you ready for the presentation? Apurva, not there? Then T Manasa is there? T Manasa. Close decision. Vijayalakshmi. Anna, Faria. Faria is there? Faria and Ravali.
Are you ready with your presentation, ma'am?
Yes, ma'am.
Start your presentation.
Our eyesight is one of the most important senses. 80% of we perceive comes through our sense of sight. By protecting our eyes, we can reduce the odds of blindness and vision. Good afternoon, one and all. My name is Ravali and my friend Faria are here to present a poster presentation on overview of current technologies in Oxford for the treatment of ophthalmic diseases.
Ocular drug delivery is one of the challenging tasks facing the pharmaceutical researchers. One of the major barriers of ocular medication is to maintain a therapeutic level at the site of action for a prolonged period of time. The main objective of present review is to describe different technologies used for the preparation of occult and importance of occult in treatment of ophthalmic diseases. So as we know, different categories of drug are used for the treatment of ophthalmic diseases such as antiviral, antibiotic, anti-glaucoma, antibacterial drugs. Methods involved in the preparation of occults like solvent casting method, film casting method, and kneading method. For ophthalmic active drugs, topical application is the most prescribed route of administration for the treatment of various ocular disorders. The intraocular bioavailability of topically applied drugs is extremely poor, which is due to flow of excess fluid by nasolacrimal duct. The advantage of ocular insert with solid device placed in cul-de-sac of the eye. Solid devices have an increased shelf life and the presence of additives such as preservatives is not required. Ocular delivery system like inserts, biodegradable polymeric system, and collagen shields are being developed in order to attain better ocular bioavailability and sustain action of ocular drug. Therapeutic efficacy of an ophthalmic drug can be greatly improved by its contact with the corneal surface. Ophthalmic inserts offer many advantages in dosage forms like increased ocular residence, accurate dosing, increased shelf life.
Physiology of eye. The eye is a spherical structure and with a wall consisting of 3 layers, the outer sclera, middle choroid layer, and the inner retina. The outer sclera protects.
Just you can conclude my topic.
Okay, ma'am. Okay. Here we conclude that oxides, those are the solid dosage, solid and the semi-solid forms which are instilled into the eyes. And oxides have wide variety of advantages to treat ophthalmic diseases. Here I conclude that the most important technique used for the oxides is film casting technique, and these are widely used for treating ophthalmic diseases, ma'am. Thank you.
Example, can you give?
Yes, ma'am.
Formulation example, marketed product.
Yes, ma'am.
Sodium diclofenac, ma'am.
Okay, ma'am. Okay.
Natamycin, sodium diclofenac, which are used in the treatment of conjunctivitis and inhibition of intraoperative meiosis and post-operative inflammation in cataract surgery.
Okay, ma'am. Okay, good presentation.
Thank you, ma'am.
Wow. Okay, ma. Good presentation.
Thank you, ma'am.
Shalia.
Please mute yourself.
Hmm? 5 minutes. 5 minutes.
వాళ్ళు ఎవరు?
ఒక్క నిమిషం. ఇంకా ఇద్దరు ఉన్నారంట మా వాళ్ళు. Yes. Is there any other need to present the poster?
Yes, ma'am.
Yes, ma'am.
Other than TKR?
Yes, ma'am.
Shirisha?
Yes, ma'am.
What is your code?
Ma'am, PMAN 503.
PMAN 503. What is the topic, ma'am?
Pharmacogenomics, ma'am.
Pharmacogenomics. You are in association with Sai Sudha, right?
Yes, ma'am.
We are keep on calling you. So many times we have called and even we have given time. You are not ready with the
No, ma'am.
your
I was
presentation,
texting
right?
I was texting
No,
you,
ma'am.
ma'am.
Sai Sudha, you asked you, I need 5 more minutes. I need 5 more minutes,
Yes,
right?
ma'am. That is before an hour, ma'am.
Then, okay,
Again,
now are you ready for presentation, Sai Sudha?
Yes, ma'am.
Okay, start your presentation. So other than Sai Sudha and Shirisha, is there any other persons need to submit? Sandhya, presented. Sriya. We call Sriya continuously, but she's not there, but Sandhya presented it.
Sandhya presented,
Yeah.
మరి ఇవన్నీ అయిపోయాయి 209 అయిపోయింది.
Yeah, she said carbon atoms, everything. Yeah, she completed.
excuse me, ma'am.
Yeah.
Ma'am, Sandhya and Swaya, we both are partners, ma'am.
It's completed, Nama.
Yes, ma'am.
Yeah. Yeah,
Ma'am,
you
is
only
my
know
slide
that
visible,
stealth
ma'am?
liposomes, everything we discussed about it regarding poster presentation.
Ma'am, is my slide visible?
Yes, ma, it is visible. You can start your presentation now.
Thank you, ma'am. Good afternoon, ma'am. I'm Sirisha, studying B Pharmacy 4th year from Nalla Group of Institutions. Today, I'm here to present a poster on pharmacogenomics, a revolution in pharmaceutical R&D and personalized drug therapy. Pharmacogenetics is a study of variability in drug response, which is determined by single gene. And pharmacogenomics is study of variability in drug response, which is determined by multiple genes within a genome. Coming to history of pharmacogenomics, it has first started when Pythagoras has observed that ingestion of fava beans resulted in potentially fatal reactions such as hemolytic anemia and oxidative stress in few individuals. This phenomenon was termed as favism. The other case is where administration of succinylcholine injection during anesthesia showed prolonged paralysis and fatal reactions. This was due to genetic variations in patients who lacked pseudocholinesterase enzyme. Coming to pharmacogenomics, it is a study of how an individual's genetic inheritance affects his or her response to drug. It can be simply said as study of influence of genetic variations on drug action. Pharmacogenomics is an approach to personalized medicine which is not based on average patients but on characteristics of individual patients. Goals of pharmacogenomics includes understanding the polymorphism of genes. Here, polymorphism refers to alterations in the genomic sequence. It is also helpful in maximizing the drug efficiency and minimizing the drug toxicity and also predict patients' response to drug. Here are few examples of altered drug responses due to genetic variations. First one is G6PD deficiency. Glucose 6-phosphate dehydrogenase is an enzyme which is involved in HMP shunt pathway. It is a good source of NADPH generation. It forms glutathione, which is present in RBC damage. In deficiency of G6PD, it leads to deficiency of glutathione, which makes RBC's membrane fragile and leads to hemolysis. The other example is thiopurine 5-methyltransferase deficiency. It is an enzyme which metabolizes thiopurine derivative drugs like mercaptopurine, thiaguanine, azathioprine, etc. And cytochrome P450 enzyme deficiency. Usually there are almost 50 cytochrome P50 enzymes in human beings. These are usually found in liver and small intestine. In people living in areas like Saudi and Thiofian, shows higher levels of cytochrome enzymes. This is the reason that they can cope up with the high toxic doses of different alkaloids. This is also the reason for them for being ineffective to certain class of drugs like antidepressants and neuroleptics. Successful therapies based on pharmacogenomics. Herceptin is a successful drug which is developed based on pharmacogenomics and is used to treat breast cancer. In normal breast tissue cell, HER2 gene is a cell surface receptor which is responsible for normal cell growth. In conditions like breast cancer, this gene is overexpressive, which is leading to cancerous growth. This is the same reason in almost 30% of breast cancer cases. R-Herceptin is an antibody which blocks cell surface receptor and prevents the abnormal cell growth, which can prevent the cancerous tumor formation. And the different techniques which can be used to identify the genetic variations in a genome is rDNA technique, that is recombinant DNA technology, and DNA amplification, DNA chip technology. These techniques can be used to identify the genetic variations in the genome. Coming to benefits of pharmacogenomics includes it helps in development of safer drugs, better vaccines, and also accurate determination of doses. Challenges to grow and expand pharmacogenomics. There are different challenges which are inhibiting the growth of pharmacogenomics, like lack of education regarding pharmacogenomics and ethical and legal issues, resistance to genetic testing, and it is also expensive. I hereby conclude my presentation stating pharmacogenomics has great potential to optimize drug therapy. It helps in development of drug to treat various problems like serious diseases, Alzheimer's, cancers, asthma, HIV, etc. Thank you, ma'am. Ma'am?
Yes. Keep your posted disclaimer.
Okay, ma'am.
What are the different advanced trends in gene delivery?
Ma'am?
Advanced trends in gene delivery.
Gene delivery.
Yeah. As you said, recombinant DNA technology.
No idea, ma'am.
Okay. But you presented very good.
Thank you, ma'am. Ma'am, can I stop my screen sharing? Hello?
Okay, ma'am?
Ma'am, can I stop sharing my screen?
Yeah, you can stop sharing.
Thank you, ma'am.
Okay. So I would like to conclude the poster presentation sessions of day 1 and day 2 of a national webinar of TKR COP and in association with RSC LSD India. So here I'm very happy to announce that the response of the abstracts we received, though it is very short duration and unexpected holiday circulars, everything, but very good response we received. We have, on behalf of management and staff and principal of TKR COP, I would like to thank Nalla Narasimha Reddy group of institutions for your active participations and for sending major number of abstracts, and Shatavahana University and Tigala Ramreddy College of Pharmacy and St. Mary's College of Pharmacy and St. Paul's College of Pharmacy and then Mother Teresa College of Pharmacy, Kurnool, Andhra Pradesh State.
Our presentation not completed.
Whose presentation?
Whose presentation, ma?
Akshita and Apurva.
Okay, you want to tell now?
Yes, ma'am.
Akshita.
Yes, make it fast then. Share your screen.
Okay.
Akshita, I have. I'm calling her, sir. I'm calling her, ma'am, continuously.
She's now ready. We'll listen to her. Don't
Discourage. Okay, Akshita.
Yes, ma'am.
Though you didn't even responded when we are calling you, but
Net problem, ma'am.
then now session is about to conclude. Okay, we are not willing to disappoint you. You start. Okay, we'll give time. Apurva
Okay,
and
ma'am.
Akshita, start your presentations.
Okay, ma'am.
You can share the screen.
Yes, ma'am. Good afternoon, one and all. I am Akshita from B Pharmacy, second year, from Tigala Krishna Reddy College of Pharmacy. Today I am going to tell about the recent trends in sustained release matrix drug delivery system. First, what is drug delivery system? It is a formulation. It is a formulation that enables therapeutic substance to reach its site of action without reaching the non-target cells, organs, or tissues. For preparation of the matrix tablets, we will combine the medicines norfloxacin 400 mg and tinidazole 600 mg. It is used to treat diarrhea and dysentery caused due to infection. They will kill the harmful microorganism which will cause the infection. The reasons the oral route achieved such popularity that the oral administration is the ease of administration and the drug is well absorbed. All the pharmaceutical products should be formulated for systemic delivery only. Next, oral drug delivery has been used still decades, and it is the widely used utilized route of administration. Among all the routes, it is the employed route of administration. It is for pharmaceutical products of different dosage forms. The reasons that the oral route achieved, such as it is the ease of administration and the drug is well absorbed. Next topic is continued by the Apurva.
Yes, Apurva, continue.
Yes, ma'am.
Are you there or not?
Ma'am, is the screen was not coming?
Yes, sir. The same Ankita can explain. If she's not in a position to explain, you just explain. Ankita.
Next, classification of the matrix system. It consists of the sum of the classification, mineral matrix, drug retained in the support, drug absorbed in the support, lipid matrix, delivery by diffusion, delivery by surface erosion, hydrophilic matrix, unlimited swelling, delivery by diffusion, limited swelling, controlled delivery through swelling, inert matrix, controlled delivery by diffusion. Modified release system. To overcome the problem of this drug therapy, modified release system were developed, and it is divided into 4 categories: delayed release, sustained release, controlled release, prolonged release, conclusion. It is used as prolong the release of drug for extended period of time to improve the patient compliance and to reduce the side effects, and it will increase the bioavailability of drugs. Yes, ma'am. Thank you.
Okay, ma.
Okay.
And very good presentation at this stage. Okay.
Thank you so much.
Very good. All the best for your next presentations also.
Thank you.
Shirish. Can we check this? Okay. Yes, ma'am. And thank you for all participants for your active participation. And we expect the same response for further conferences which are going to be held in Tigala Krishna Reddy College of Pharmacy. And once again, I would like to thank all the colleges who have participated actively in national webinar, especially Nalana Reddy Group of Institutions, Rathavana University, Tigala Ram Reddy College of Pharmacy, and St. Mary's College of Pharmacy, St. Paul's College of Pharmacy, and University College of Pharmaceutical Sciences, Kakatiya University, Mother Teresa College of Pharmacy, Kurnool, Andhra Pradesh State, and special applaud, not to mention, but all Tigala Krishna Reddy College of Pharmacy students for your interest and active participations in our college. So now I would like to hand over the session to Mrs. P. Sunitha, Associate Professor in Department of Pharmaceutical Analysis, Tigala Krishna Reddy College of Pharmacy. Thank you.
Thank you, madam. Dear delegates, welcome to the valedictory session. And it's my pleasure to introduce Mr. Mujibuddin, CEO, Clinosol Research Private Limited for this valedictory session. Mr. Mujibuddin did his MPharm in pharmacology and MSc in clinical research. Since 2005, he has been serving several top-rated pharma CROs as well as IT firms, and is a trailblazer with strong determination and wisdom to overcome adversity. He started his career in clinical research when not too many of the current younger generation knew about this industry. Possessing over 15 plus years of industry experience, he has managed every stage with utmost care and diligence. Of late, in the last 8 years, he instilled his skills into trainings and has trained around 5,000 plus professionals, among which many of them are reaping successful careers. As of now, he is striving to benefit medical, pharma, biomedical, and life science students with his teaching methodologies. He loves sharing actionable ideas and information about clinical research, pharmacovigilance, clinical data management, regulatory affairs, and clinical SaaS. Having a wide array of experience as an associate manager, an independent consultant, author, lecturer, and CEO of an IT-enabled pharma services training and consulting firm, Clinosol India Private Limited. As a subject matter expert, he has helped many professionals to strengthen their relevant work skills. Thousands of his students have already benefited from the practical knowledge shared by his online, offline, and corporate trainings. Now, I invite Mr. Mujibuddin Karu to present valedictory address. Over to Mujibuddin, sir. Good evening.
Thank you. Good evening. Thank you. Thank you, madam, for calling
Good evening,
me.
sir. Mujibuddin, sir.
Thank you, madam. Thank you. How are you? Thank you. So, warm welcome and happy to be here. I was seeing the slide presentation and it was super good. Thank you so much. Big round of applause for all the faculties and all the organizing members and all the students who have supported this event. Thank you so much. So before starting any of my session, I would like to tell all the young minds, you have a very good future. You have very good careers upcoming because we are hearing more about COVID and I'm sorry. We are hearing more about COVID and the research is needed in terms to give more scientific data, more scientific evidence about the new drugs or devices or any devices which are being used for COVID testing or any vaccines which are being used for COVID testing. So before I tell any of this wordings for young minds who are attending this, so never hold your horses back before you come into an existing saying that you don't have jobs and all those things. You have multiple arenas starting from clinical research, clinical data management, pharmacovigilance, medical writing, or clinical SaaS. You have n number of opportunities. I would like to share a couple of experience with you guys. So I think everyone knows about pharmacology and pharmacist. We are all proud to be a pharmacist. We are proud to be a pharmacist, and we intend to tell that we are doing enough research to give right drugs or right vaccines to the human beings. So being a pharmacist, I am very much proud to say this moment that I have been invited for giving sessions to MBBS and MD doctors and medical colleges to speak about clinical research. So let's be proud on that area that being a pharmacologist or pharmacist, we have been invited to speak to doctors about continual medical education, which some of the medical colleges host this. And we should be proud enough to say that I was reporting, or I was having a reportee of nearly 15 to 20 IIT engineers. So being a pharmacist, some 15, 20 IIT engineers were reporting to me when I was working in Oracle India Private Limited. So wherein we have to feel happy that being a pharmacist, we were able to show that ability to handle the IIT engineers also. We are not less than any of the engineering graduates, or we are not less than any of the doctors. We are into ecosystem of the healthcare professionals. We are part of ecosystems, and if you see any of the drug or a vaccine or any device which is coming into market, the whole and soul research is being done by most of pharmacists and most of healthcare professionals, including physicians and all those things. So the career oriented path for you guys is marvelous. I would say that it's very clear, detailed that due to increase of COVID, most of research is being outsourced to India. Either it may be a clinical research or data management or pharmacovigilance or clinical SaaS. So more jobs are coming to India. So the requirement from companies, I would like to share with you what is required from the industry, what industry is looking, because I come from industry and I speak in terms of industry, how industry is looking. So they are requiring the technical skills, which is most important in terms of the industry expertise, the technical skills, how you are good at regulation and guidelines, how can you analyze the regulation and guidelines and conduct the research studies. That's a point number one. Point number two, very good communication skills, either it is speaking or writing or listening or conversing or convincing. So this is very important point, and right attitude towards the work and dedication and discipline is most important aspects where companies are looking for it. So to reiterate it, to clear it, most of IT companies are hiring pharmacists, or most of CROs are hiring pharmacists, because we clearly states in clinical research that as per GCP, there should be a requirement who is a healthcare professional who has to work in clinical research. So there will not be any other person who is not a healthcare professional to work in clinical research. When you speak about research of new drugs or vaccines or any new molecule, we have to have a part of pharmacists or a doctors who have to work on these lines. So that's where more opportunities, more growth in terms of clinical research has came. So we are very fortunate to have a very much huge jump in the careers or jump in the number of jobs we are getting in terms of research. So I would suggest all the students to be more active on professional platforms like LinkedIn and start writing blogs and articles and start communicating and convincing the people about your resumes. Do not copy your resumes, have your own resumes where you can put the points what you wanted to speak in interview, because the resume is only one point where they will see for in terms of interviews. And the new jobs which are upcoming is medical affairs, which is giving a lot of opportunities, and HURs, health economics and outcome research. This is also giving lot of scope in terms of the research which we are thinking that how much drug should be costing, because the drug should be purchased by patients. Though we are doing enough research about the financial aspects, that how much it has to be sold for the patient so that it can reach to the right patients. So coming back to the performance of Clinosol, I was very happy with the performance. I worked in industries for 15 years and then I started Clinosol 3 years back, and tremendous performance for the last year 2021. We were very happy, all blessings of God. We were very happy to share this data. We have got nearly 300 jobs all over India. So this is wonderful aspect, and we have got students from TKRCP also who have got jobs from Clinosol, who got trained in the Clinosol, and we have got net worth of package of nearly 6 crores. On an average in 1 year, we got 6 crores of packages, and nearly 70 plus companies have hired from Clinosol. And we had industry expert interactions, nearly 100 plus interactions we had with company industry experts, and we gave nearly 4 lakhs plus cash awards for a number of events. Nearly 250 blogs were being published and 175 presentations were being given, and nearly company trainings we have conducted for more than 100. So I would request all the students to be more active in terms of learning and making yourself comfortable. Though we have gone into offline to virtual mode of learning, but still the learning should continue, and until the life is continuing, the learning should continue. So keep motivating yourself and keep working towards it. Maybe one more time lockdown may come or go. This all things will come and go, but learning should be very much active. We should not get distracted with our learning. So, and thank you so much, madam, for calling me. And I'm very delighted to talk on this topic. Thank you.
Thank you, sir. Now I invite Dr. Pintu Ganguly Garu, Vice Chairman, RFC LSD, India, to address the gathering.
Thank you, madam.
Good evening, sir.
Oh, fine. That means I'm audible.
Good evening, sir.
Yes, sir.
Oh, good. No, I'm extremely happy to be giving this taking up this concluding session on very interesting 2 days of learning. And as Mr. Mujibuddin very rightly said, it's all about learning. I think this intense 2-day learning that we had under the auspices of Tigala Krishna Reddy College of Pharmacy on this interesting topic of approaches and challenges in pharmaceutical sciences in association with RSC. I think it has been a wonderful 2 days of very good learning for the students. I would like to take this opportunity to thank Professor Jayant Kandare, who spoke about liquid biopsy in detecting cancer, onco-discovery technology. He has his own company. He is also a professor. Then yesterday we had Professor Pisapati, again our esteemed colleague and our guide and advisor in RSC. He spoke about importance of chirality in drug therapy. Thank you to Professor Jayant Kandare and Professor Pisapati for their wonderful lectures on very important topics. Today we had Dr. Chandraya on recent trends in pharmacology and toxicology. He spoke about And then we have our esteemed colleague, Dr. Narendra Talluri, who spoke about analytical techniques and its strength. And when we look at the list of the speakers that we had a very good mix of industry and academics. There were several veterans of academics, and we also had good industry representation. And the valedictory session by Mr. Mujibuddin, again, it's extremely useful, and the context that he brought in about employment or employable opportunities for pharma students. I think we should be all thankful to him to have given us hopes about what is there in future for all of you. So as we say in RSC, we say that let's advance chemistry together. And we do this by bringing in scientists, we collaborate, we organize the poster sessions. I think another, my thanks to also the students. I could see a variety of posters. I did join little late, but again, I could see the mix of name reactions, pharmacogenomics, nutraceuticals, a wide range of posters. Again, great learning opportunities for all of you. And some of those were excellent, but very good effort from all of the students. Before I say thank you and wish you all the best, I would want to mention on that we have another very important skill development program that we are organizing, RSC is organizing along with DICEL. DICEL is sponsoring it. It's a national skill development training program for academicians and job aspirants. It's basically for MSc and MPharm students who are there in their final year, or those who would have completed. It would help them get into some skill training that will enable them to get better jobs as they look forward to their future. So the topic is modern methods in pharmaceutical analysis. This is being held from 18th Jan to 21st Jan. And the last date of registration is 12th Jan. So all the MPharm and MSc students, or mostly MPharm students, that those who would be attending them, we would urge you to participate and get these trainings to help you with your future job prospects. With that, thank you again, the management of the Tigala Krishna Reddy College, the faculty, and for giving us this platform to have this national webinar. So thank you, all of you, and all the best for a great career and a great future. And stay healthy, stay good. All the best. Thank you.
Thank you, sir. The most awaited part of the national webinar is prize distribution. Now, I invite Dr. K.R. Radhika Garu to announce prize winners in e-poster competition. Over to Dr. K.R. Radhika Garu. Voice not coming. Voice.
Your voice, madam. Hari. Madam, your voice. No, madam. No, madam. Unable to listen.
Madam, have you connected any headphones to the system? Remove it and talk it directly. Headphones would have been connected. Remove that headphones, wires and all. Directly talk on the mic. No, I think there is, you are on unmute, but we are unable to hear you. You are on unmute. Or else you have to check your mic connections.
Should
Madam, we can do one more thing. We can exit. We can ask Radhika Madam to exit and join once again.
I think that would help.
Am I okay, sir, now?
Yeah, yeah. Now it is okay. Now it is okay.
Thank you so much. I was a little, when such things happen, especially with these electronic items, we really get, paranoid. Thank you so much, Mujibuddin. Thank you very much, Dr. Pintu Ganguly, Dr. Mujibuddin. All of you have made this program worthwhile. I hope that all the students who have been participating and all those who have very enthusiastically, presented the posters, kudos to all of them. Kudos to the college, Tigara Krishna Reddy College, their vibrant and dynamic principal and her extremely able team of lecturers and good students. I really thank you. And at our end to RSC and all the people who are there here, Dr. Ganguly, Dr. Pisapati, Dr. Abhiyan Prasad, then Dr. Sri Lakshmi, all these people are a great help to us, really. I should congratulate them, and then I will not stand in between the excitement and the students. I will start from the consolation prize, just to build up the aura. I will start from the consolation prize. We have given 2 consolation prizes. The first consolation prize goes to the title ADRs of ACE inhibitors, G. Nikita, Tigala Krishna Reddy College of Pharmacy.
3rd year, B Pharmacy.
Yeah, she is in the 3rd year. Okay. Now the next consolation prize goes to growing demand for plastic during COVID-19. It was extremely interesting, sir. It was yesterday and very well presented, sir. I just wanted to tell both of you, very, very well presented by K. Bhavana. Again, she's from Tigala Krishna Reddy College of
Pharmacy, B Pharmacy, 4th year.
Yeah. Hearty congratulations, Bhavna and Nikita. You have done an excellent job. Then the 3rd prize goes to Oncolytic Virotechnology. Oncolytic Virotechnology by P. Nishita. She is from Nalla Narasimha Reddy Group of Institutes. Nishita, hearty congratulations for your 3rd prize. Then the 2nd prize goes to role of artificial intelligence in tackling COVID-19. Very impressive topics also these students have chosen. Sakshi Sharma from Tigala Krishna Reddy College, she backed the.
Madam, again,
everybody got
Mute.
Muted.
I think host has muted everyone. Madam, you are on mute.
Make Radhika Madam.
No, you can unmute yourself.
Yes, sir. Thank you so much. Yeah, I didn't even realize. Did all of you hear the 2nd prize, sir?
No,
role
no.
of artificial. Yeah, the 2nd prize goes to role of artificial intelligence in tackling COVID-19 by Sakshi Sharma, Tigala Krishna Reddy College of Pharmacy. They have done
3rd year B Pharmacy.
Yeah, very interesting topics they have chosen actually. And the first prize goes to sperm hybrid, Sainath Chari Gannoji from St. Paul's College of Pharmacy. Hearty congratulations. Actually, I must now here tell you that there were more than 850 registrations for this. 850 registrations were there. 52 people have actually wanted to present papers, but I think they could not, some of them could not manage. So 35 people presented papers. And the most, good thing about the PR of Tigala Krishna Reddy College is 8 colleges participated in this event.
Wonderful. That's wonderful.
8 college participation. It's a wonderful thing, sir.
And very good learning for everybody.
Yeah.
Madam, thank you again for organizing all this for the student. Thank you, Radhika Madam. You have been
thank you, sir. Thank you so much. Thank you, sir. Hearty congratulations to all the prize winners.
Thank you, Radhika Madam. Thank you.
And speakers also. Thank you so much, Mujibuddin. Thank you.
Thank you, Radhika Madam.
Thank you, madam. Now, I invite the principal Tigala Krishna Reddy College of Pharmacy, Dr. Vijay Kuchana Garu, to deliver concluding remarks and vote of thanks.
Dignitaries, Mr. Muzubuddin, CEO Clinisan, Dr. K.R. Radhika, Honorary Secretary, Royal Society of Chemistry, Local Section Deccan, Dr. Pintu Ganguly, Vice Chairman, RSA LSD, Professor V. Sapati, Special Advisor, Special Advisor, RSA LSD, Dr. P. Srilakshmi, Treasurer, RSA LSD, Dr. R.B.N. Prasad, Chairman, RSA LSD. Good afternoon to you all. So we came to an end of the two-day national webinar, Approaches and Challenges in Pharmaceutical Sciences, ACPS 2022. Firstly, I would like to thank the management of Tigala Krishna Reddy College of Pharmacy for extending their support for conducting this two-day national webinar. I would like to thank especially the Royal Society of Chemistry for associating with the Tigala Krishna Reddy College of Pharmacy for conducting this two-day national webinar. I would like to thank all the speakers for spending their valuable time to share their knowledge in these lectures. Actually, really, it was a celebration of knowledge for 2 days. Wonderful celebration of knowledge this 2 days. So day one keynote speaker was Dr. Kandare, Jayant J. Kandare, co-founder Arctorius Innovations and Research, OncoDiscover, Pune, professor at MIT, the keynote speaker. So he explained about the prevalence of the cancer, different types of cancers, liquid biopsy, which is a non-invasive, rapid and precise method for detecting this cancer, isolation of tumor cells, cell DNA, tumor node and metastasis stages, types of epithelial to mesenchymal transitions. And he also explained about the strategies for effective anti-cancer drug delivery systems. So next speaker in the day one was Professor V. Sapati. He explained about chirality, importance of chirality, effect of chirality on the safety and efficacy, and the ADME of the drug molecules in the body. And next, there were e-poster presentations in the afternoon in day one. So next we have day two. The first speaker was Dr. Chandaya, Assistant Professor from Department of Regulatory Toxicology, Head Biological Sciences, Naibar, Hyderabad. So he explained about the role of pharmacology in drug discovery and development, various stages involved in drug discovery and development, role of antibody drug conjugates for cancer targeted therapy, GLP toxicology guidelines, various cancer biomarkers, drug repurposing, omics, proteomics, genomics, and he also explained the latest approaches and strategies for drug delivery systems for cancer. So next we had Dr. Talluri Narendra Kumar lecture. So he was he's head AR&D, knowledge management, diacell chiral technologies. So he explained about origin of impurities, analytical techniques like he also enlightened different techniques like HPLC, supercritical fluid chromatography, supercritical fluid extractor, UPLC, LCMS, and other techniques like counter current chromatography, chiral chromatography, and he has given more emphasis on columns used in chiral chromatography and explained about quality by design and its importance. So I also thank Dr. Muzibuddin, CEO Clinisal, for being as a special speaker for this valedictory. So even we had e-poster presentations in the afternoon, just now we finished. So I'm happy to share that about 850 registrations were made by different students and research scholars, staff from the college and other colleges. We received 52 abstracts from 8 different colleges. 8 colleges were participated. And among 52 abstracts, 34 presentations were made by the students. Just now, Madam already announced. The first prize winner will be given a cash prize of 1,000 rupees. The second prize winner will be given a cash prize of 750 rupees. And the third prize winner will be given a prize of cash prize of 500 rupees. And we have 2 consolations. Each prize is 300 rupees. So they will be given 300 rupees towards this consolation. 2 consolation prizes were made. Most importantly, I would like to thank all the delegates and participants for spending their time. I also thank my faculty members and students of Tegal Krishna Reddy College of Pharmacy for their support for conducting this webinar and made it a successful one. I would like to thank Dr. Padmaja, HOD, IT Department, Tegal Krishna Reddy Engineering College for her technical support. So with this, I conclude this 2-day national webinar, which is served as a platform for students and research scholars, faculty members to interact with experts from both the industry and academia. As Ganguly sir was rightly pointed out, we have speakers from industry and academia and share their knowledge towards development of skill in multidisciplinary areas of research in pharmaceutical sciences. Hope all delegates are satisfied with this 2-day national webinar. And I thank everyone on behalf of Tegal Krishna Reddy College of Pharmacy who directly or indirectly involved in this webinar and made this 2-day celebration of knowledge a wonderful and successful one. Thank you, one and all.
On behalf of Royal Society of Chemistry, London, also India Deccan section, I thank all the participants, everyone who was named by your principal, Dr. Vijay Lakshmi, for having participated enthusiastically and making this program a success. Thank you very much.
Thank you. Yes, madam.
Can we end the meeting?
Yes, madam. We'll end the
and
meeting.
yeah, please thank all your faculty on my behalf. Yeah, they have
Yes,
really
ma'am.
done, and those 2 male staff members also who were,
male staff also.
Yeah,
Yes,
everyone. Anyhow, we always, men will be
Keep
in
in
front of
touch.
the camera. Yeah, men will be behind the screen. So thank everyone, all the participants and all the staff members of your college. Thank you.
Thank you, madam. Thank you for you also, especially. So you made this webinar, supported a lot for this webinar.
Thank you, ma'am.